तरन्नुम।
हेलो हेलो।
हेलो।
तो आप लोगों के पास अगर कोई भी क्वेश्चन कुछ होगा, कुछ मन में कुछ दुविधा हो, जो आपने अभी पिछले 2 घंटे से जो लेक्चर सुने हैं, और अगर कुछ विचार आ रहे हैं आपके मन में, तो आप लोग हमारे एक्सपर्ट से पूछ सकते हैं। ओके सर।
So, good afternoon, friends. Are you comfortable or you are exhausted? Those who are exhausted, they can raise their hand like this. And those who are comfortable, they can raise their hand like this. So, most of you are comfortable, energetic. We welcome you to National Brain Research Center, the apex body in the field of neurosciences. And we have got panel of scientists, clinicians, special educators, psychologists, psychiatrists to talk to you. उद्देश्य क्या रहेगा हमारा बात करने का कि जो सुबह से हम बात कर रहे हैं ऑटिज्म के बारे में या फिर जो न्यूरो डेवलपमेंटल डिसऑर्डर्स के बारे में बात कर रहे हैं, उनके बारे में मन में बहुत सारी शंकाएं होंगी, बहुत से सवाल होंगे और डाउट्स ऐसे होंगे जिनके बारे में हमें लगता है कि हमें क्लियर कर लेना चाहिए। एजुकेशन एक ऐसा फील्ड है जिसका ना केवल पूरे देश से बल्कि हमारे भविष्य से सीधा संबंध है, देश के भविष्य से सीधा संबंध है। तो अगर हम कोई गलत चीज यहां से सीख के जाते हैं तो वो गलत चीज ही वहां पे एग्जीक्यूट हो जाती है। इसीलिए ये डाउट क्लियरिंग सेशन भी है, सवाल जवाब भी है, पैनल डिस्कशन भी है। छोटी सी एक स्टोरी से मैं शुरू करना चाहूंगा। मैं साइकेट्रिस्ट के तौर पे काम कर रहा था गुड़गांव सिविल हॉस्पिटल में। तो हमारे जो सीएमओ थे उनका मेरे पास फोन आया कि ऐसे ऐसे करके किसी सेक्रेटरी हेल्थ ने एक केस भेजा है आपके पास में, इसको देखो और इसका डिसेबिलिटी सर्टिफिकेट बना दो। तो उनका डिसेबिलिटी सर्टिफिकेट के लिए जब वो बच्चा आया तो वो बहुत ज्यादा चुस्त कूद रहा है, कभी इधर चढ़ रहा है, कभी उधर चढ़ रहा है, वो टिक के नहीं बैठ पा रहा था। तो मैंने कहा कि ठीक है, इसको थोड़ी देर असेस किया। असेस करने के बाद में अंदाजा यही हुआ कि ये शायद इसको अटेंशन डेफिसिट हाइपर एक्टिविटी डिसऑर्डर है, जिसके बारे में कुछ चर्चा अभी हुई होगी, एडीएचडी। लेकिन क्योंकि स्कूल के अंदर टीचर्स ने या पेरेंट्स ने कह दिया था इसके बच्चे के नंबर नहीं आ रहे हैं, ये तो मंद बुद्धि है, वो मंद बुद्धि का सर्टिफिकेट बनवाने के लिए आए हुए थे। हमने उसको असेस किया, लगा कि मंद बुद्धि तो नहीं लग रहा है। तो जैसे ही उसको बोला कि हम इसको दवाई दे के देखेंगे और उसके बाद में सर्टिफिकेट बनाएंगे, तो उनकी फैमिली को, क्योंकि वो बड़े सिफारिश से आए हुए थे, उनकी फैमिली को लगा कि ये टाल मटोल कर रहे हैं जैसा कि सरकारी अस्पतालों में होता है। उन्होंने दोबारा से फोन कराया सीएमओ को कि ये डॉक्टर थोड़ा अजीब है, ये काम कर नहीं रहा है। सीएमओ ने मेरे को बुलाया कि क्या बात भाई, डू यू नो हु हैज सेंट दिस पेशेंट। मैंने कहा सर मैंने तो ज्यादा ध्यान नहीं दिया इस बारे में। कहते शी इज द सीनियर मोस्ट ऑफिसर इन द स्टेट, सेक्रेटरी हेल्थ, जिसको हम सब लोग रिपोर्ट करते हैं, उन्होंने भेजा है। तो टांग क्यों अड़ा रहा है काम के अंदर, काम कर। तो मैंने कहा सर ये तो होएगा ही ऐसे कि वी विल गिव सम मेडिसिन। हम पूरा उन्होंने जोर लगाया लेकिन देयर वाज नो चॉइस, हमने उसको दवाई दी। एंड द मैजिक ऑफ न्यूरो साइंसेज और द मैजिक ऑफ मेडिकल साइंस इज लाइक दैट। जादू इस प्रकार का है, वो बच्चा ठीक हो गया, अटेंशन डेफिसिट ठीक हो जाता है। वो बच्चा अगले महीने ठीक हो गया, उसके पढ़ाई में नंबर भी अच्छे आने लगे। उसके बाद में जो हमारे वो सेक्रेटरी हेल्थ नाराज थे, अगले महीने मैडम का फोन आया, वो कहते कि तुम तो जादू कर सकते हो, मैं मेरी जान पहचान के अंदर एक मंदबुद्धि बच्चा और है, उसको भी दवाई दे दो, वो भी ठीक कर दो। इस बार भेजा तो इट वाज ए केस ऑफ मेंटल रिटार्डेशन, जो कि दवाई देके ठीक नहीं हो सकता था। लेकिन मैं ये कहानी इसलिए बता रहा हूं कि कभी कभी इतने पढ़े हुए और इतने इंटेलिजेंट लोग भी गलती कर सकते हैं। तो उन्होंने कहा तुम्हारे हाथ में जादू है, तुम कुछ भी कर सकते हो। मैंने कहा मैम जादू कुछ भी नहीं है, वो बीमारी ठीक होने वाली थी तो ठीक हो गई, ये वाली बीमारी इसके लिए दवाई नहीं है। हाउएवर रिहेबिलिटेशन एंड अर्ली एजुकेशन, अर्ली इंटरवेंशन, फिजियोथेरेपी, ऑक्यूपेशनल थेरेपी, वो सब अभी भी हो सकती हैं। तो ये एक ऐसा पहलू है कि इतनी सारे न्यूरो डेवलपमेंटल डिसऑर्डर एक जैसे नजर आते हैं जब हम एज ए टीचर बात करते हैं। क्यों टीचर से वर्कशॉप रखी गई है? क्योंकि फर्स्ट कांटेक्ट या तो पेरेंट्स है या फिर स्कूल के अंदर आप हैं। तो आपका बहुत ही महत्वपूर्ण रोल है इसके अंदर, वो जो आप अदा कर सकते हैं। मैं फर्स्ट ऑफ ऑल रिक्वेस्ट करूंगा कृषाणु सर से फॉर हिज कमेंट्स, एंड देन आफ्टर हिज कमेंट्स वी वुड बी इनवाइटिंग द क्वेश्चंस आल्सो। सो ब्रीफ फ्यू मिनट्स ऑफ योर कमेंट्स सर।
तो आप सभी को नमस्कार। और आप यहाँ पे आए हैं तो हम बहुत आपके आभारी भी है, क्योंकि ये जो सिलसिला हमने चालू किया था पिछले साल से, तो ये सिलसिला अभी जारी रखना है हमें। तो अब आपसे मुझे एक प्रश्न पूछना है कि आप में से कितने आदमी दूसरी बार आ रहे हैं इधर? तो बहुत अच्छी बात है। तो मुझे लग रहा है कि आपको कुछ पहली बार अच्छा लगा, इसीलिए आपने दूसरी बार आया। तो ये ऐसा सिलसिला चलता रहेगा, क्योंकि ये तो एक बार में ख़त्म नहीं होने वाले हैं। हमको ये संपर्क संपर्क बना के रखना है, और बार बार बात और आपस में बात करना है। तो फिर उसी से हम एक दूसरे से सीखेंगे और ये सिलसिला आगे बढ़ाएंगे। तो मैं साइकियाट्रिस्ट नहीं हूँ, साइकोलॉजिस्ट नहीं हूँ, मैं ब्रेन साइंटिस्ट भी नहीं हूँ। मैं सिर्फ इतना जानता हूँ कि ब्रेन न्यूरॉन से बनता है और वो एक मशीन है। और एक मशीन जैसे उसको ट्रीट अगर करेंगे आप, तो फिर आपका जो जितने सारे गलत फहमियाँ हैं वो दूर हो जाएगी। क्योंकि हर मशीन की तरह ब्रेन का भी एक इनपुट आउटपुट फंक्शन होते हैं। तो हम जैसे उनको इनपुट देंगे वैसे ही उसके आउटपुट आएंगी। और जैसे हर मशीन एक जैसा नहीं होता है, तो हर हर दिमाग एक जैसा नहीं होता है। तो हर एक दिमाग अलग अलग होता है। तो ये जो संवाद हमने संपर्क किया है, अभी एस्टेब्लिश किया है, ये संवाद की चर्चा की विषय ऐसे ही होना चाहिए कि ये जो डाइवर्सिटी है, जो अलग एक एक मशीन अलग अलग जो बना है, और उसका मतलब एक ही इनपुट से दूसरी किस्म की आउटपुट आते रहते हैं, तो उसको कैसे समझाया जाए और उसको कैसे ट्रीट किया जाए, मतलब उसको कैसे मैनेज किया जाए। क्योंकि हम अ, हर एक बच्चे को अलग अलग से मैनेज करेंगे तो उसकी विकास भी अच्छी तरह हो पाएगी। और जब उसका विकास अच्छी तरह हो पाएगी तो वो बच्चे आगे जाके समाज में बहुत ही अच्छी तरह योगदान कर पाएंगे, यही हमारी आशा है। तो इसीलिए हमने ये पैनल आपके लिए बनाया है। तो मुझे यही कहना है कि आप आते रहिए, हमारे 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 साथ संपर्क बना के रखिए, और फिर आगे हम सिलसिला बढ़ाएंगे। थैंक यू वेरी मच।
Thank you, sir, for your opening remarks. Now, next in the panel is and डॉक्टर शिफाली गुलाटी, हमारे सबसे इम्पोर्टेंट जो इंस्टीट्यूशन मेडिकल साइंस में है, एम्स दिल्ली, वहाँ के मैम हेड ऑफ डिपार्टमेंट हैं, और इस फील्ड के अंदर इनका अंतर्राष्ट्रीय स्तर पर योगदान है मैम का। तो मैं तालियों के साथ मैम का सबसे पहले स्वागत करना चाहूंगा एज ए गेस्ट पैनलिस्ट, और मैम के ओपनिंग रिमार्क्स के लिए प्लीज मैम।
एक्चुअली हमारे को आइडेंटिफाई करना होता है जो हमारे पास स्कूल में बच्चे आ रहे हैं ना। कई लोग पढ़ रहे होते हैं अच्छे, हम उनसे खुश रहते हैं, और जो अच्छे नहीं पढ़ रहे होते हैं हम उनको दोषी ठहराते हैं कि इंटरेस्ट नहीं ले रहे हैं। पर हमारे को एज टीचर्स ये देखना है कि जो नहीं पढ़ पा रहे हैं अच्छे, है ना, उनको कोई और परेशानी तो नहीं है। कई लोग सिर्फ किसी को दिख रहा ही सही रहा है, सुनाई सही दे रहा है, छोटे बच्चों की क्लास में इंपॉर्टेंट है। फिर वो जैसे आपने तारे जमी पर देखा था, है ना, वो ना टीचरों को पता चला ना उसके पेरेंट्स को पता चला, वो बस ये था फादर उसको थप्पड़ ही मारता था हमेशा, है ना, और उनके बाकी बच्चों के साथ नहीं था। बच्चा कंफ्यूज था क्योंकि उसको अक्षर ही समझ में नहीं आ रहे थे, टिल वो उसको बोर्डिंग स्कूल में भेजा और आमिर खान ने रिसर्च करके समझा कि क्या प्रॉब्लम है। फिर अल्टीमेटली उसके बाद उनके जो एनुअल रिपोर्ट के बैक पेज में उसकी पेंटिंग आई। तो बेसिकली है हर बच्चे में पोटेंशियल होता है, उसको हमें देखना है और क्या परेशानी है उसकी गलती नहीं है। बहुत सारे न्यूरो डेवलपमेंटल कारण होते हैं, आपको एडीएचडी जैसे बताया सर ने, वैसे ऑटिज्म है, कुछ मानसिक का क्षमता में कमी होती है जिसको ग्लोबल डेवलपमेंटल डिले बोलते हैं, 5 साल से नीचे, और ऊपर इंटेलेक्चुअल डिसेबिलिटी, और भी स्पीच एंड लैंग्वेज डिसऑर्डर होते हैं, स्पेसिफिक लर्निंग डिसेबिलिटी होता है। तो क्या क्या जो भी बच्चा स्कूल में ठीक नहीं कर रहा है, हमें उस पर अटेंशन फोकस करनी है और देखना है। और उसके बाद आपको अगर किसी पीडियाट्रिशियन को दिखाना है, अपने हमारे स्पेशल एजुकेटर देखेगी ताकि देख के पता चले कि क्या है। तो हमारे को खुद ही चौकन्ने होना है, पेरेंट्स को भी देखना है और साथ में हमें भी, ताकि हर इंसान, हर बच्चा, जैसे हमारी पांचों उंगलियां बराबर नहीं है, हर बंदा भिन्न है, उसका जो भी पोटेंशियल है उस तक लेके जाएं। और उसमें अगर कोई परेशानी है, अवस्था है, कोई न्यूरो डेवलपमेंटल डिसऑर्डर है, तो उसको पकड़ें। तो आप सबका बहुत इंपॉर्टेंट रोल पड़ता है उसके अंदर।
थैंक यू मैम, संक्षिप्त रूप से बताने के लिए कि किस प्रकार से आप योगदान दे सकते हैं इस विषय के अंदर। हमारे बीच में कलकत्ता से मैम आए हैं, तो डॉक्टर मल्लिका बनर्जी मैम का मैं स्वागत करता हूं। और आपके मैम मैम को मैं रिक्वेस्ट करूंगा हिंदी इंग्लिश दोनों में मिक्स करके मैम बोलिएगा प्लीज, ठीक है?
And sorry, at first I apologize that I can speak properly in Hindi. It may actually create some love to you if I speak in Hindi fluently. I can't actually. The main point, what I'm telling, and I have heard the last, to some extent, the last lecture presentation. One point is very much important, particularly for any type of disability, not for only autism, but for autism, that is the emotional aspect. It is very much important. Because of that social and emotional aspect, autism are actually far behind. Whatever their abilities are, one point, they can't express it in socially accepted way, and they don't want to. For that reason, it is said that with all others, or person with autism alone, even with others, they within themselves. Why? That is, they are not bothered about the society. They don't want to. Whatever they have, their ability, etc., all things, they don't want to express, or they don't know to express. Whichever anyone may be prominent, whatever, but both are there. That is the first point of that. For that reason, they are emotionally not stable. They can't recognize others' emotion. They can't express their emotion in a sociable way. So I think, for as the teachers, at first, it is not at first, but a priority for autism is there. What the my previous speaker said, that what is actually what should not be actually the attitude, etc., all things by which they have that particular self-esteem less, and according to the social interaction, we have to be aware that self-esteem is not decreasing toward zero, but we have to increase it up to which they can express, they have that actually, they have that not the ability, wish to express something. They wants to. That want is actually in all of ours, and it is our duty to actually provoke them willfully to express. And for that reason, emotional, affective, and social aspect is very, very important. Cognitive aspect, all you know, you are the teacher, you know better than me how to increase that cognitive aspect. But of course, the attitude toward the society and attitude of you toward them, of course, it should be empathetic, not sympathetic. How much sympathy you will go, you are actually demeaning them. So it should be empathy. I don't know proper Hindi of it. In Bengali, Shomanubhuti, not Shohanubhuti. It is sympathy, not empathy. Again, I am telling, I don't know the Hindi term of it. But really, that is the first we have to acquire. And for that, after that, with us, if we really grow our empathy toward them, then we will be successful for a real socio-emotional attitude for them, by which we can increase their at least hope to want, wish, hope to express themselves. After this, thank you, and then a little.
धन्यवाद मैम, आपके साइकोलॉजिकल एस्पेक्ट के बारे में मैम ने बताया। तो मैं थोड़ा सा ब्रीफ करना चाहूंगा मैम ने किस तरीके से बताया। मैम बोल रहे हैं कि इमोशनल एस्पेक्ट्स जो हैं, या जो व्यवहारिक पहलू हैं ऑटिज्म के, न्यूरो डेवलपमेंटल डिसऑर्डर्स के, या इस प्रकार की मानसिक समस्याओं के, वो बहुत ज्यादा महत्वपूर्ण है। हमें उनके बारे में ध्यान रखना चाहिए, और ना केवल उस बच्चे के बल्कि वो बच्चे इतने प्यारे होते हैं कि उनके साथ में आप इमोशनली टीचर भी जुड़ जाते हैं, टीचर भी जुड़ जाते हैं, डॉक्टर्स भी जुड़ जाते हैं इमोशनली, और पेरेंट्स तो इमोशनली उनसे बहुत ज्यादा अटैच्ड रहते हैं। तो जो इमोशनल एस्पेक्ट्स हैं बच्चे के संबंध में, वो इम्पोर्टेंट है, हमें ध्यान रखना चाहिए इनके बारे में। और एक बड़ी अच्छी बात मैम ने बोली कि उन्हें सहानुभूति से ज्यादा समानुभूति की आवश्यकता है, कि उनको हम, समानुभूति वाला शब्द बड़ा अच्छा बताया। इट इज सेम मैम, हिंदी और हिंदी बंगाली सेम है, देखो। बंगाली में भी यही बोलते हैं। यस मैम, तो बंगाली में भी समानुभूति बोलते हैं, हिंदी में भी। सो हमारे पास एक्शन फॉर ऑटिज्म से 2 स्पीकर्स हैं, उनसे आप दोनों से आपका सुबह से संपर्क हुआ है, लेकिन फॉर द सेक ऑफ पैनल डिस्कशन मैं दोनों से 1 मिनट के लिए रिक्वेस्ट करूंगा। पहले दिव्या जी से रिक्वेस्ट करूंगा कि आप कुछ शब्द कहें।
हाय, मैंने तो ऑलरेडी बहुत बातें की है। इंक्लूजन की बात की है कि उनको साथ लेके चलना वो बहुत ज़रूरी है। तभी कुछ हो सकता है। जब उनको एज अ चैलेंज, एज अ प्रॉब्लम देखते हैं तो बहुत मुश्किल होता है। हमारा नज़रिया जो है वो होना चाहिए ऐसा कि हम उनको एज अ प्रॉब्लम नहीं देखें। हम ऑफ कोर्स सोल्यूशन ढूंढें जो चीज़ें हो रही हैं उसके लिए, पर ये जो आता है ना, ओहो मुश्किल है, सही नहीं है, ये फीलिंग आती है। इट्स वेरी वेरी नॉर्मल एंड देयर इज काइंड ऑफ, कोई ये नहीं है कि आप गलत कर रहे हैं। ह्यूमन बीइंग को आता है, जब डिफिकल्ट सिचुएशन आता है तो ये आता है इंटरनल फीलिंग हर एक इंसान को। भगवान नहीं है, आता है। लेकिन इसी को हमें थोड़ा अवेयर होते हुए समझना है कि ये जो फीलिंग है हम उसको मिनिमाइज करें एंड हैव पेशेंस, उनके साथ काम करें, उनको जो चाहिए वो देने की कोशिश करें, पहले समझें, देने की कोशिश करें, ताकि वो नॉट ओनली स्कूल क्लासरूम एवरीवेयर उनको हमारे जैसे ही अपॉर्चुनिटी हो बात करने का, कहने का, अपनी बात, सारे काम करने का, रेस्पेक्ट जो मिलता है, जो ट्रीटमेंट और अटेंशन मिलता है, वो भी हमारे जैसे वो ले पाए। ये बहुत ज़रूरी है। कैसे होगा? अनटिल एंड अनलेस हम उनको एक्सेप्ट करें, अपने में वो स्टेमिना लाएं कि ठीक है मुश्किल हो रहा है बट ठीक है, कोई बात नहीं। सो ये करना है। इट्स डिफिकल्ट बट इट कैन हैपन, हो सकता है। ठीक है, बस इतना ही।
धन्यवाद दिव्या जी आपके शब्दों के लिए, ब्रीफ। एंड फाइनली मैं रिक्वेस्ट करूंगा अमृता जी से फॉर हर ब्रीफ वर्ड्स अबाउट द पैनल डिस्कशन। बाकी आप सुबह से इनसे बात कर ही रहे हैं, प्लीज।
मैं सिर्फ एक कमेंट करना चाहती हूँ। हमारे जो इंडिविजुअल्स हैं, हमारे जो बच्चे हैं, अगर वो कोई भी चीज कर रहे हैं, अगर वो उछल रहे हैं, वो एक जगह पे बैठ नहीं रहे हैं, उनको मुश्किल हो रहा है हमारे एनवायरनमेंट में वहाँ पे बैठने के लिए या कोई भी चीजों को समझने के लिए, पहले हमें ये समझना है कि हमारा बच्चे को कहाँ मुश्किल हो रहा है, किस जगह मुश्किल हो रहा है, क्यों हो रहा है। जब तक हमें वो वाई नहीं पता लगेगा, हम कहीं पे भी, हम किसी भी तरीके से हम बच्चे को या वो इंडिविजुअल को हम हेल्प नहीं कर पाएंगे। सो इट्स ऑलवेज इंपॉर्टेंट कि हम ये अंडरस्टैंड करें द वाई, क्यों कर रहे हैं। इट कैन बी द डिफिकल्टी जो बच्चा फेस कर रहा है, या इट कैन बी कि उसकी फैमिली में ऐसे ऐसे चीजें हो रही है जिसके कारण उसके पेरेंट्स उनको हेल्प नहीं कर पा रहे। तो एज टीचर्स, एज एजुकेटर्स, वो हमेशा हमारा रिस्पॉन्सिबिलिटी होता है कि हम पेरेंट्स को या उस फैमिली को हम किस तरीके से हम उनको गाइडेंस दें और उनको हेल्प करें। थैंक यू।
धन्यवाद है आपके शब्दों के लिए। सुबह से हम इस विषय में बात कर रहे हैं, तो हम इस बात को मानने के लिए तैयार है कि ये एक महत्वपूर्ण पहलू है, इसके बाद इस विषय पे बात करनी चाहिए। और खास तौर से नेशनल ब्रेन रिसर्च सेंटर के जो ऑटिज्म के हम 3 के बारे में, इन्होंने न्यूरो डेवलपमेंटल डिसऑर्डर के बारे में अपने फाउंडेशन डे पे ये डिस्कशन रखा है या वर्कशॉप रखी है, हम इनका धन्यवाद कर देते हैं। सबसे पहले एजुकेशन डिपार्टमेंट की तरफ से। एजुकेशन डिपार्टमेंट एक तरीके का एक कोलैबोरेशन है बिटवीन साइंस एंड
साइंस। आप भी साइंस से ही आते हैं, एजुकेशन में भी साइंस है, आपने ही पढ़ा के साइंटिस्ट तैयार किए थे एजुकेशन डिपार्टमेंट ने, और आज एजुकेशन डिपार्टमेंट को वापस साइंटिस्ट अपना पहलू वापस करते हैं। पर्पज इज टू गिव बैक टू सोसाइटी, कि हम अपनी सोसाइटी को वापस क्या दे पा रहे हैं। तो आप में से कुछ ऐसे सदस्य होंगे जिनके मन में सवाल उठते हैं कुछ इस बारे में, या कुछ डाउट होते हैं, या कुछ ऐसी बात है जो आप शेयर करना चाहते हैं, तो आपका स्वागत है। वन बाय वन हमारे एक माइक, हमारे जो ऑडियो विजुअल कोऑर्डिनेटर है, मैं उनसे निवेदन करूंगा कि माइक आप प्लीज हैंड रेज करें। जी जी, धन्यवाद। आपका नाम बताइएगा। जी जी, प्लीज प्लीज।
जस्ट अ मिनट।
कहाँ पोस्टेड है ये भी बताइएगा जी। सब्जेक्ट कहाँ पोस्टेड, नाम, तीन चीज। साकरस से आए हैं जी,
प्राइमरी स्कूल पे।
बहुत अच्छे जी, स्वागत है आपका।
ऑटिज्म के बारे में सुबह से हम सुन रहे हैं, और ये बताया गया है कि अलग नजरिया होता है उस बच्चे का, दूसरे बच्चों से अलग नजरिया होता है। मैं ये जानना चाहता हूँ कि जो विकलांग बच्चे होते हैं, ये ऑटिज्म उसी श्रेणी में आता है या इससे अलग होता है?
जी जी जी, हाँ, धन्यवाद आपके सवाल के लिए। मैम से मैं रिक्वेस्ट करूंगा टू रिस्पॉन्ड टू द क्वेश्चन।
प्रश्न के लिए धन्यवाद। ऑटिज्म एक न्यूरो डेवलपमेंटल डिसऑर्डर है, हाँ, एक अवस्था है, बीमारी नहीं बोलते, अवस्था बोलते हैं, ठीक है। तो उसके अंदर ये है, और उसके अंदर जो है बहुत और सारी आती हैं, जिसमें आपको बताया था, जिसमें मानसिक क्षमता जो कम होती है, पुराने जमाने में मेंटल रिटार्डेशन बोलते थे, लेकिन वो बहुत नेगेटिव टर्म है, दूसरे को नीचा दिखाने का टर्म है, तो वो टर्म पूरे वर्ल्ड ओवर से हट गया है। तो इसलिए मैंने जो बताया था, 5 साल से नीचे ग्लोबल डेवलपमेंट डिले, और 5 साल से ऊपर इंटेलेक्चुअल डिसेबिलिटी बोलते हैं, ठीक है। और वो जो तारे जमी पर वाला जो स्पेसिफिक लर्निंग डिसेबिलिटी था, किसी को नजर कम है तो विजन इम्पेयरमेंट, हियरिंग इम्पेयरमेंट, इत्यादि इत्यादि, ये सारे जो है न्यूरो डेवलपमेंटल, जो बच्चे का डेवलपमेंटल पीरियड है, जो कि मैक्सिमम शुरू होता है फर्स्ट 1000 डेज, मतलब प्रेगनेंसी के भी पूरा टाइम और उसके बाद भी पूरा, तो मैक्सिमम उस समय होता है। उस समय कोई भी परेशानी होने से, जिसमें कुछ जेनेटिक होते हैं, अनुवांशिक, कुछ इन्वायरमेंटल होते हैं हमारे आसपास में, और कुछ इन्वायरमेंट में कुछ चीजें होती हैं जो, अब जो मैं जेनेटिक मेरे को पाया है, जीन्स में मेरे को है, वो तो मुझे मिल गया मम्मी डैडी से, राइट। लेकिन वो जब उसका जो, उसमें से वो जीन्स के अंदर से प्रोटीन बन रहा है, राइट, प्रोटीन बनने का प्रोसेस है, उस प्रोसेस जो है इन्वायरमेंट में हो रहा है, मेरी बॉडी के इन्वायरमेंट में, मेरी जो बॉडी की इन्वायरमेंट है, इज गेटिंग इन्फ्लुएंस बाय दैट एयर पोल्यूशन, बाय पेस्टिसाइड एक्सपोजर, स्ट्रेस, क्वालिटी ऑफ लाइफ, ये सारी चीजों से है। तो ये कॉम्प्लिकेटेड हो गया ना, कुछ अनुवांशिक है, कुछ जेनेटिक है, कुछ इन्वायरमेंटल है, तो आप समझ रहे हो, तो ये न्यूरो डेवलपमेंटल डिसऑर्डर्स हैं। अब आप विकलांगता की बात कर रहे हो, जो डिसेबिलिटी का एक्ट है सरकार का, वो आप बच्चों को, इन बच्चों को सपोर्ट देने के लिए बनाया गया है, तो वो 21 डिसेबिलिटी में से ये सारे उसमें इंक्लूड किए, पर आपको हम सबका नजरिया देखना है। आज पहले जमाने में जब थे, मैंने बोला जब मेंटल रिटार्डेशन बोलता, वो नेगेटिव टर्म था, पहले हम बोलते थे नॉर्मल बच्चे हैं, ये अब नॉर्मल बच्चे हैं, ठीक है, पुरानी बात है, 15-20 साल पुरानी। फिर हो गया न्यूरोटिपिकल्स और स्पेशल नीड्स, है ना, जैसे आप लोग हैं, हम लोग हैं, जो हम टिपिकल डेवलपिंग है, बाकी स्पेशल नीड्स है। अब वो बदल गया है, अब क्या कहा जा रहा है वर्ल्ड ओवर, सारी दुनिया में, न्यूरो डाइवर्स। हम आपके स्कूल में भी देखा, जब हम भी पढ़ते, 4-5 लोग हैं जो मरते, मर मर के पास होते थे हर बार, और 4-5 ऊपर हैं जो फर्स्ट पाने के लिए लड़ते रहते थे, सेकंड भी आ गया, वो दुखी हो रहा है, फेल होने वाला दुखी ना है, वो सेकंड वाला दुखी है, और मेजोरिटी वाले बीच में है। तो ये स्पेक्ट्रम है ना, हम इनको नॉर्मल मांग रहे हैं। तो अब ये कह रहे हैं कि जो ऑटिज्म क्या है, जिसमें सोशल इंटरेक्शन, सामाजिक अभाव, कम्युनिकेशन, संचार, इन दोनों में गुणात्मक हानि है, और साथ में उनमें कोई रिपीटेड दोहरावनीय व्यवहार होते हैं, फिक्स्ड इंटरेस्ट होते हैं, इत्यादि इत्यादि बात हुआ है, सेंसरी इश्यूज होते हैं, ऑल दैट, वो सब अलग है। पर पॉइंट इज, ये भी तो उसी स्पेक्ट्रम का पार्ट है। तो आज के लिए कह जा रहे हैं न्यूरो डाइवर्स है, ये हमें भिन्न लग रहे हैं, हमारे सोसाइटी के जो हमारे अभाव और जो सोशल इंटरेक्शन, जो सामाजिक अभाव और संचार के हिसाब से हमें वो भिन्न लग रहे हैं, उन्हें हम भिन्न लग रहे हैं, उनको हमें नहीं चेंज करना है, हमें उनको साथ लेके चलना है। आप मेरी बात समझ रहे हैं? अब सब चेंज हो गया है। और जो दिव्यांगन है जो बोला जा रहा है, वो एक आपको सपोर्ट देने के लिए बच्चों को और उनके पेरेंट्स को, वो डिसेबिलिटी एक्ट है। और डिसेबिलिटी शब्द एक्चुअली इस थोड़ा सा नेगेटिव है। और जो एक चीज है, हर इंसान का जो पैदा हुआ है, उसका इक्वल फंडामेंटल राइट टू लाइफ, एजुकेशन, डिग्निटी,
सेम बिजनेस से है, कोई व्हील चेयर में आ रहा है या कोई ऑटिज्म का व्यवहार ठीक नहीं कर रहा, उसका राइट है डिग्निफाइड होने का। आप किसी पे एहसान नहीं कर रहे, जो बच्चा ठीक नहीं कर रहा उसको देखने का आपका ड्यूटी है। जब आप मर रहा है, जो मर मर के पास हो रहा है, उसको भी तो आप मान रहे थे। हमारे बॉस होते हैं कई बार, वो बड़े हट में गुस्से वाले होते हैं, हम नौकरी थोड़ी छोड़ देते हैं, एडजस्ट करते हैं ना। तो हमें उनको नॉट आउट ऑफ ऑब्लिगेशन, बिकॉज ऑफ इट इज एथिकल, इट इज लीगल, आपका ड्यूटी है कि सबको साथ लेके चलना है। तो आपको उनको करना है, जो बच्चे ठीक नहीं कर रहे स्कूल में, आइडेंटिफाई करना है कि क्या कारण है, और जो भी उसका ट्रीटमेंट है, थेरेपी है, स्पेशल एजुकेशन के साथ चलना है। तो आप ये समझिए कि हर बंदे को, हर को वैसे ट्रीट करी जैसे मुझे है। मेरा कोई एक्सीडेंट हो जाए, मैं व्हील चेयर पे चले जाऊं, मेरे को बाद में डिमेंशिया हो जाए, तो मेरे को भी प्रॉब्लम हो जाएगी। तो ये लोग सोचते हैं ना, जिनके बच्चों को प्रॉब्लम है, कि भाई हमारे को तो है नहीं, तो ठीक है, और उसमें लुक डाउन करते हैं, बेचारा बेचारा, वो सब नहीं है। हमारे को उनकी प्रॉब्लम के ऊपर बेचारा नहीं बोलना है, उनकी प्रॉब्लम को समझ के उनको सपोर्ट करना है। आजकल सोसाइटी में इक्वालिटी की हम बात नहीं कर रहे हैं। इक्वालिटी है कि आपको ये मिला, रिसोर्सेज सबको इक्वली दे दिया। इक्विटी की बात हो रही है, जो हाइट में छोटा है उसके नीचे स्टूल नहीं लगाओगे। अब स्पीकर आता है, छोटे वाला आता है, स्टूल नहीं देते नीचे, नहीं, छोटी सी बात है। तो इट इज दैट, इट इज, आप किसी पे एहसान नहीं कर रहे, इट इज योर ड्यूटी एज ए टीचर। यू विल फेल अगर आप सबको इक्वल नहीं करेंगे। जो आपके टीचर बच्चे पास हो रहे हैं, जो अपने आप साइंटिस्ट बन रहे हैं, और जो है दैट दे आर डूइंग, पर आप जो नहीं कर रहे उनको भी उनके मैक्सिमम पोटेंशियल तक लेके जाना है। दैट इज योर ड्यूटी। आपके ये बहुत लीगली इंपॉर्टेंट है, एथिकली इंपॉर्टेंट है।
बहुत ही अच्छे शब्दों में मैम ने बताया। मुझे लगता है आपका डाउट क्लियर हुआ होगा साक्रेस वाले टीचर साहब का। देखिए, बड़ी अच्छी बात मैम ने बताई, वही समानुभूति वाली बात। यू वांट टू ऐड प्लीज?
I want to add one point, ma'am, that is, if we think the attitude that person with autism, person first, not autistic child, child with autism, person first, if we actually make our habit with that, we have to give them really proper respect what you are telling. So from today we think over, I and you all, if you for another 10% you tell that person first, not the disability, then it is okay. I'm 1 minute I'm telling, I'm short heighted. Already you perceived. I'm short heighted, and in my life there are so many demanding terms I have heard, so many. Once I think that I have to think over how to overcome it. I have a spondylosis, and someone tell me that, oh, you spondylosis, it is natural because always you have to talk with your, but because I am short heighted, with your facing up, for that reason spondylosis started. At that time I told, oh no, who are longer than me, who wants to talk with me, he has to come down, make eye contact with me, and then I will talk. I know it is a pride talk. I know it is a high pride, but it was needed. It was needed at that time. Really not. But I know that is needed to make pride. It is our duty to give them that pride. Thank you.
बहुत अच्छे मैम, बहुत ही अच्छे शब्दों में मैम ने कहा। मैं कहूंगा कि एजुकेशन के अंदर तो टीचर का प्राथमिक दायित्व यही बनता है कि जो लाइन में लास्ट है या जो पढ़ाई में पीछे चल रहा है उसको ज्यादा सपोर्ट दे। सपोर्ट उसको भी दे जो फर्स्ट आ रहा है, बहुत अच्छी बात है, लेकिन जो लास्ट लाइन में है उसे सपोर्ट देना सबसे आवश्यक है। और सबको समान व्यवहार करना वो भी जरूरी है, तभी हम इक्वल एजुकेशन दे पाएंगे, एक इक्वल सोसाइटी का निर्माण कर पाएंगे। एक छोटी सी, मेरे को ये नाम इतने बदले हैं, मैम ने जैसे बताया ना कि पहले हैंडीकैप बोलते थे, फिर डिसेबिलिटी बोलने लगे, अब नाउ न्यूरो डाइवर्स बोलने लगे, अलग अलग शब्द हुए हैं। हमने एक यहाँ विश्वास फाउंडेशन काम करती थी पहले, तो उन्होंने कहा कि हम इस पे डिसेबिलिटी, मेंटल डिसेबिलिटी सर्टिफिकेशन के ऊपर कैंप लगाना चाहते हैं। तब नया नया शब्द डिसेबिलिटी यूज होने लगा था, लगभग 15 साल पहले। उससे पहले क्या बोलते थे? हैंडीकैप। हमारे बोर्ड को हम लोग बोलते हैं हैंडीकैप बोर्ड, अभी भी ऐसे ही बोलते हैं। तो उस बोर्ड के लिए हम लोग वहां सारा फॉर्म लेके, उन्होंने कहा हम सोना में चाहते हैं। तो सोहना का जो एरिया है मेवात से मिला हुआ एरिया है और वहां पे बहुत ज्यादा जागरूकता नहीं थी। बहुत लोग आए, सैकड़ों लोग आए अपने सर्टिफिकेशन के लिए, पूरी भीड़ हो गई। और जब कैंप शुरू किया तो हमारे जो डीलिंग और हमारे साइकोलॉजिस्ट और डॉक्टर सब लोग इकट्ठे बैठे हुए हैं। जो फाउंडेशन की चेयरमैन थी उन्होंने कहा कि कब तक आपका ये कैंप शुरू हो जाएगा? कौन सा कैंप मैम? हैंडीकैप कैंप। उन्होंने जैसे ही हैंडीकैप बोला, कहते डोंट से हैंडीकैप, हैंडीकैप बोलना गलत बात होती है, हैंडीकैप नहीं है। वो कहने के बाद में उनकी लड़ाई हो गई। हम नहीं करेंगे जी कैंप। हमारे हेल्थ डिपार्टमेंट के जो डॉक्टर्स थे या क्लरिकल स्टाफ था, सबने कहा जी हम यहाँ पे सोना में आए, ये आके हमारे को कह रहे हैं कि हैंडीकैप ही नहीं, हमारी गलती निकाल रहे हैं, हमने कैंप नहीं करेंगे। सो वंस आई रीच देयर, जब मैं वहां पहुंचा तो ये लड़ाई काफी ज्यादा हो चुकी थी। तो मैं वहां गया और मैंने जाते ही उन सब लोगों को कहा कि क्यों नहीं कैंप शुरू किया, 2 घंटे हो गए, इतनी सारे लोग बैठे हुए हैं। तो कहते जी ये तो हमारे साथ बदतमीजी कर रहे हैं, हमारी गलतियां निकाल रहे हैं। जागरूकता, एजुकेशन, इट इज ए कंटिन्यूअस प्रोसेस। हमारी एक छोटी सी बड़ी बुक है, हैरिसन की बुक है। उस बुक का आखिरी पेज यही कहता है कि जब तक ये बुक लिखी जा रही है, ये बुक आउटडेटेड हो गई है। जिस दिन ये छपने के लिए निकलती है उसी दिन नई, इतनी रिसर्च नई हो चुकी है, इतनी स्टडीज हो चुकी हैं कि वो भी आउटडेटेड हो गई। जिस दिन छपती है उस दिन उसका शुरुआत का पहला पेज आउटडेट हो जाता है, उस बुक में लिखा हुआ है। तो ऐसे ही साइंस है, हर रोज साइंस में इतनी तरक्की हो रही है कि हर रोज नए आविष्कार हो रहे हैं, नई अपडेशन हो रही है। तो हम लोगों को अपडेट होने के लिए तैयार होना चाहिए, और अपडेट करने के लिए तो एजुकेशन वाले ही, टीचर्स ही तैयार कर सकते हैं। और एक जी, और एक सवाल के लिए आपका आमंत्रण है। किन के मन में सवाल उठता है जी? प्लीज प्लीज आप नाम, प्लेस ऑफ पोस्टिंग एंड सब्जेक्ट भी बताइएगा।
सर मेरा नाम जय भगवान है। मैं नगीना नो मेवात में प्राइमरी टीचर हूँ। बेसिकली प्राइमरी में 1 से 5 क्लास तक एक टीचर को ही सारे सब्जेक्ट्स पढ़ाने
जी
होते
जी
हैं।
जी जी।
सर मेरा मेन क्वेश्चन ये है कि हम लोग सुबह से बात कर रहे हैं, जो ऑस्टियम बच्चे हैं, डिसेबल जो बच्चे हैं मेंटली, उनको जैसे मैं क्लास में बच्चे को पढ़ा रहा हूँ, मेरे पास 2 स्टूडेंट या 1 स्टूडेंट है, उनको मेरे को अल्फाबेट पढ़ाना है, काउंटिंग पढ़ानी है, उन पे मैंने फोकस कर लिया है
जी।
उन पे मैंने जो नॉर्मल बच्चे से ज्यादा टाइम दे दिया है। इसके अलावा मैं क्लासरूम में और क्या कर सकता हूँ कि उस बच्चे को वो मेंटली वो डेवलप हो जाए, उनके बराबर में आ जाए?
जी
क्या
जी
एक्टिविटीज हम लोग कर सकते हैं? और गवर्नमेंट की तरफ से भी ऐसे बच्चों के लिए कोई प्रोग्रामिंग्स होते हैं क्या कि हम उनके, क्योंकि बेसिकली आपको ज्यादा नॉलेज है कि हमारा मेवात ऑलरेडी बहुत
जी
पिछड़ा
जी
हुआ
जी
क्षेत्र
जी
में
जी
काउंट होता है पूरे इंडिया में।
जी
तो वहाँ काफी बच्चे हैं, पेरेंट्स हैं, और मैं कुछ प्राइवेट संस्थाएं हैं। आपने अगर गुड़गांव में काम किया तो खुशबू सेक्टर 10A में है, जो इन्हीं
जी
बच्चों पे काम
जी
करती है, उनके साथ भी सर मैंने काम किया है। तो उन पेरेंट्स को भी हम गाइड कर सकें कि गवर्नमेंट की जो ऐसी हेल्प है, कोई अपॉर्चुनिटी है, क्योंकि वो ज्यादा पढ़े लिखे नहीं होते तो उनके पास पहुंच नहीं पाती हैं कई स्कीमें। तो इस टाइप के अगर कोई हो तो बताएं। और क्लास में इनके अलावा हम क्या कर सकते हैं कि वो बच्चे समाज में और घर में डेवलप हो जाए? देखो, और काउंटिंग और मैंने अल्फाबेट तो सिखा दिया, उसमें तो बराबर आ गया, लेकिन और बहुत सी चीजें हैं जिनमें उनको हमें बराबर लाना है, उनमें तो जा ही नहीं पाए, क्योंकि वो जब गांव में जाएगा, घर में जाएगा, तो काउंटिंग अल्फाबेट काम नहीं आनी है। मेरा
जी
अपना
जी।
ये मानना है सर।
बहुत अच्छे। देखिए जय भगवान जी, आपका सबसे पहले अप्रिशिएट करते हैं आपके सवाल के लिए। आपने खुशबू वेलफेयर के साथ भी काम किया, और आपके मन में जो सवाल है वो बिल्कुल जायज सवाल है। तो मैं अमृता जी से कहूंगा कि बीइंग द स्पेशल एजुकेटर, ये राइट पर्सन है इस बारे में रिप्लाई करने के लिए। लेकिन मैं अपना एक पहलू जरूर कहना चाहूंगा कि हमारे जो हमने इंडेक्स बना रखे हैं सोसाइटी के अंदर, जैसे मैम ने हाइट के लिए बोल दिया कि अगर कोई 5 फुट 3 इंच हाइट वाला है तो वो तो ठीक है, उससे 6 फुट 3 इंच वाला तो उसको तो हम लंबू बोल देंगे, और 5 फुट से छोटे वाले को छोटू बोल देंगे। क्यों भाई, सब है तो बराबर के इंसान है, हम क्यों किसी को लेबल करते हैं? इसी प्रकार से अगर हम अपने इंडेक्स जो बना के रखते हैं कि हर बच्चे को साइंस भी आनी चाहिए, उसको इंग्लिश भी आनी चाहिए, हिंदी भी आनी चाहिए, सारी लैंग्वेजेस आनी चाहिए, मैथ्स भी आना चाहिए, आप देखेंगे हमने ऐसे इंडेक्स बना रखे हैं कि वो धीरे 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 करके बच्चों की संख्या कम होती रहती है। प्राइमरी एजुकेशन के टाइम पे अगर 100 बच्चे भर्ती होते हैं स्कूल के अंदर, 10वीं पास करते 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 50 रह जाते हैं। 50 बच्चे हमारे डिफिकल्ट इंडेक्स की वजह से बाहर निकल जाते हैं उस एजुकेशन सिस्टम से। तो हम लोगों को ना केवल सपोर्ट देने की आवश्यकता है, बल्कि इंडेक्स भी, सारा कुछ पढ़ाना जरूरी है, पढ़ा दीजिए, लेकिन इतना नहीं कि इतना स्ट्रिक्ट कर दें कि उस फिल्टर में से, उस बोटल नेक में से बाहर ही ना निकल पाए। तो ओवर टू अमृता जी।
सी क्लासरूम एनवायरनमेंट में तो डेफिनेटली हर तरीके के बच्चे होते हैं, और आप जिसके बारे में आप बोल रहे हैं, जितना मुझे समझ में आ रहा है, हमारा जो एजुकेशन है वो बहुत ज्यादा अल्फाबेट्स, नंबर्स, ये सारी चीजों के ऊपर हम बहुत फोकस करते हैं, एंड वो बहुत जरूरी है। पर उससे पहले बहुत सारे प्री रिक्विजिट्स भी होते हैं जो बच्चे को समझना बहुत जरूरी है। जैसे कि क्या बच्चे को पता है बड़ा कौन सा है, छोटा कौन सा है, गीला कौन सा है, ठंडा कौन सा है। ये सारे कंसेप्ट्स के बारे में भी हमें ध्यान देना बहुत जरूरी है, क्योंकि ये सब चीजें हम बहुत अपने नेचुरल एनवायरनमेंट में हम ऐसे ही सीख जाते हैं। पर हमारे जो इंडिविजुअल्स है, हमारे बच्चों को ये सारी चीजें सिखानी पड़ती है। तो हमें ये सब समझने के लिए ऑब्वियसली आप अपने अंडरस्टैंडिंग के हिसाब से आपने जो किया वो डेफिनेटली बहुत अच्छा कर रहे होंगे। पर उसके अलावा उसके जो प्री रिक्विजिट्स, क्या क्या चीजें वो नहीं कर पा रहा है, उसके ऊपर हम काम करें, और वो हमें आगे जाके फ्यूचर में वो हमें ही हेल्प करने वाला है, हमारे बच्चे को हेल्प करने वाला है। जैसे आपने अभी डेफिनेटली बोला कि अल्फाबेट्स एंड नंबर्स हमारे फ्यूचर में वो इतना इंपॉर्टेंट नहीं होगा जितना हमारे जो डेली लिविंग एक्टिविटीज है वो इंपॉर्टेंट होगा। अगर हमने अल्फाबेट्स, नंबर सिखा दिए पर बच्चे को टॉयलेट जाना ही नहीं आता है, तो हमारा फोकस उसके ऊपर भी होना बहुत जरूरी है कि बाकी सारे कौन कौन से स्किल्स है जो बच्चे को नहीं आता है, उसके ऊपर भी हम फोकस करें, सो दैट वो आगे जाके वो इंडिपेंडेंट लिविंग करे। बिकॉज इन दी एंड हम जो पढ़ाई लिखाई, ये सारे एजुकेशन, सब कुछ जो कर रहे हैं, हम किस लिए कर रहे हैं? सो दैट हम फ्यूचर में कुछ अपने लिए कुछ बने, हम इंडिपेंडेंटली हम अपना पैसे कमाए और अपना घर खरीदे, या हम इंडिपेंडेंटली रहे, एक परिवार संभाले। यही हमारा मेन फोकस है। तो दैट्स व्हाट आई वांटेड टू ऐड।
बहुत अच्छे से मैम ने बताया। तो जय भगवान जी, प्लीज कृष्णानु सर से मेरी रिक्वेस्ट।
मेरे ख्याल से उन्होंने ये पूछ रहा है कि अगर उनको कोई स्पेशल ट्रेनिंग मैनुअल अगर मिलेगी इसके ऊपर, तो उससे उसको कुछ सहायता हो सकता है। तो इसी बच्चे के लिए ऐसे ही आप पूछ रहे थे कि कोई ट्रेनिंग मैनुअल अगर होगा।
अल्फाबेट आ गया, काउंटिंग आ गया, लेकिन टॉयलेट्स करना नहीं आया, तो ये जो अदर एक्टिविटीज हैं ये मैं कैसे क्लास में डेवलप कर सकता हूँ?
तो इसका एक
कैसे
मैनुअल अगर होगा तो
मैनुअली मैं इम्प्लीमेंट कर सकता हूँ?
वो
क्या क्या
implement हो
एक्टिविटीज हैं आपने पढ़ा और रिसर्च किया, और ज्यादातर बच्चों में जो रहता है मेंटली डिसेबिलिटी
अच्छा, देखिए बिल्कुल सर ने बड़ा अच्छे से क्लेरिफाई कर दिया, जय भगवान जी। तो हम लोग ये रिस्पॉन्सिबिलिटी लेते हैं कि एजुकेशन डिपार्टमेंट को एक मैनुअल बना के देंगे, जिससे कि प्राथमिक जो टीचर है, प्राइमरी टीचर्स हैं, क्योंकि सबसे ज्यादा वहीं पे ही ये बच्चे नजर आएंगे। फिफ्थ क्लास तक के टीचर्स ही इस बात को आइडेंटिफाई कर ले, सबसे आवश्यक यही है। तो हम ये इस टाइप का एक मैनुअल इकट्ठा करके एंड वी विल प्रोवाइड इट टू एजुकेशन डिपार्टमेंट।
Primary needs should be actually recognized at first, primary needs, and then the education mean not only the ABCD but the real primary needs for expression.
मैनुअल, वेरी राइटली सेड सर, एक मैनुअल हम बनवा के ये आपके पास पहुंचवाते हैं। जी जी, एक बार इनका एक सवाल और पूछते फिर आप। नो डियर,
थैंक यू सर, आपने जो सजेस्ट किया कि मैनुअल हम गवर्नमेंट को भेजेंगे,
हम ना एससीईआरटी के माध्यम से आपके पास भिजवाएंगे। एनसीईआरटी का मैनुअल रहता है।
मैं बेसिकली मेवा डिस्ट्रिक्ट की बात कर रहा हूँ, मैं अदर डिस्ट्रिक्ट का ज्यादा वो नहीं। अदर डिस्ट्रिक्ट में लगता है कि मैं गवर्नमेंट टीचर हूँ, तो वहाँ अदर डिस्ट्रिक्ट्स में तो इतने बच्चे नहीं, हमारे मेवात में जरूर भिजवाना सर आप।
नहीं, बहुत अच्छे जय भगवान जी, आपके जज्बे को सलाम। ये बात सही बोल रहे हैं जी। मैं हमारे वो थी यहाँ पे एनबीआरसी के साथ लगभग 10 साल पहले की बात है, दिल्ली वाला एक प्रोजेक्ट शुरू किया था। तो उस दिल्ली के प्रोजेक्ट के अंदर, बैठिएगा प्लीज। तो उस प्रोजेक्ट के लिए हमें हमारा ये प्लान था कि हम असेसमेंट करेंगे, और हमारी वो थी राशि वाज क्लिनिकल साइकोलॉजिस्ट। राशि को हमने कहा कि राशि ने नया ज्वाइन किया, और उनको मैंने बोला कि ये आईक्यू टेस्टिंग करनी है, हमने आईक्यू टेस्टिंग के लिए कैंप लगाएंगे। तो कैंप लगाया, तो उन, क्योंकि क्लिनिकल साइकोलॉजिस्ट रिसर्च में काम करते हुए और पब्लिक से काम करते हुए दोनों में बहुत फर्क है। आप जब एम्स में देखते हो तो हजारों मरीज आ रहे हैं, तो शी वाज एक्सपेक्टिंग कि 2-3 बच्चे आएंगे, हम आराम आराम से करेंगे। जब हम वहां पे गए तो वहां पे ट्रैक्टर ट्रॉली भर के, कहां पे हमने ताउडू के पास एक गांव में कैंप लगाया था जी, ट्रैक्टर ट्रॉली भर भर के इतने सारे बच्चे जो सर्टिफिकेट बनवाने के लिए आए हुए थे, इतनी ज्यादा आवश्यकता थी। और उन्होंने कहा कि जी आजादी के बाद पहली बार यहाँ पे डॉक्टर आया हमारे गांव में तो क्योंकि गांव के अंदर कभी कोई जाता ही नहीं था। तो दैट वाज एनबीआरसी इनिशिएटिव, ऑलमोस्ट 10-15 इयर्स बैक की बात है सर। तो एंड शी सेड सर मैं तो यहाँ पे दो किट लेके आई हूँ, 2-3 बच्चे सोच के आई थी, ये तो मुझे लग रहा है कहीं सैकड़ों हजारों होने वाले हैं। मैं कोई बात नहीं, आज तो हम करके ही जाएंगे। वो बात है कि हायर एक्सपेक्टेशन होती, कई बार हर किसी का तो सर्टिफिकेट था भी नहीं, लेकिन सब लोगों को ये था। एजुकेशन में हमने डिपार्टमेंट में मैसेज यही किया था कि जिस बच्चे के जो आपको लगता है स्क्रीन कराने, उनको सबको भेज दो। तो आवश्यकता काफी है, ये बात सही है, मेवात इज डेफिनेटली डिफरेंट। एक और पॉजिटिव मैं मैसेज मैम का बताना चाहूंगा। जी, डाली को डिजिटल कर रहे हैं, यस। तो ये जो डाली का रिसर्च बना था, उसके ऊपर भी अभी उसका एक ऐप आ रहा है, वो भी ऐप आपके साथ में शेयर हो जाएगा जैसे ही वो ऐप आ जाता है। तो उसके माध्यम से जिन बच्चों में समस्या है उनको आइडेंटिफाई कर पाए, उस ऐप के माध्यम से। वो टीचर्स के लिए ही ऐप है, टीचर्स उस ऐप को यूज कर पाए फोन में और आइडेंटिफाई कर पाए। तो वो भी आपके साथ जल्द ही जुड़ेंगे और विवाद से ही शुरुआत करेंगे।
तो ये ऐप खाली डिसलेक्सिया के लिए बनेगी। तो लेकिन हमारी उम्मीद है कि इसको हम आगे बढ़ाएंगे और बहुत सारे और न्यूरो डेवलपमेंटल डिसऑर्डर्स जो है उनको डिटेक्शन के लिए भी इसको एक्सपेंड करेंगे। लेकिन फिलहाल जो बीटा वर्जन निकलेगी अभी इसका, वो खाली डिसलेक्सिया के लिए निकलेगी।
वन मोर क्वेश्चन, जब मैं क्लास में पढ़ा रहा हूँ टीचिंग मेथड जनरली उन बच्चों के लिए मेरे को अल्फाबेट सिखाना है, तो मैं एक बच्चे को जो मेंटली डिसऑर्डर है थोड़ा, जिसको हम एसटी में काउंट कर रहे हैं, तो उनमें और जो नॉर्मल बच्चे का टीचिंग मेथड सेम रखते हैं, बस केवल हम रिपीट ज्यादा कर देते हैं कि भाई इस बच्चे को 4 बार बता दूंगा मैं, एक जनरल जो हमारी सोच है। इसके अलावा भी कोई मेथड है रिसर्च में कि हम उसको कुछ अलग सी टाइप का एजुकेशन दें, सेम चीज को रिपीट के अलावा कोई समथिंग सेकंड मेथड जिससे कि बच्चे
बड़ी सही
को...
आपका सवाल है और लोगों को भी ये बात फील हो रही होगी। अगर कोई सवाल मुश्किल है तो हम क्या बोलते हैं उसको, किसी भी, चाहे 10वीं का एग्जाम की बात कर लो, 12वीं की कर लो, कोई सवाल मुश्किल है तो टीचर्स क्या बोलते हैं, बार बार रिवाइज करो, बार बार रिवाइज करो। और वो भी बोलते हैं रसरी आवत जात है सिल पर परत निशान। तो बार बार रिवाइज करने से लर्निंग हो सकती है। तो डेफिनेटली अगर आप एक बच्चे पे ज्यादा ध्यान देंगे, उसको अच्छे से वो चीज रिपीट करवाएंगे, तो उसकी लर्निंग हो जाएगी, ये बात है। एक और पॉजिटिव नोट मैं बताना चाहूंगा मैम को, शेफाली मैम को शायद याद ना हो, एक बार 10-12 साल पहले की बात है, इनके यहाँ पे एम्स में एक वर्कशॉप थी। आई वेंट देयर फॉर टू पार्टिसिपेट इन दैट। उसके अंदर उन्होंने ये कहा कि जो उनके वो थे स्पेशल एजुकेटर थे, वो कहते जी हमारा ये बच्चा मैथ्स में बहुत अच्छा है, आप इससे मैथ के बारे में कोई सवाल पूछो। तो अब जैसे हमारे डॉक्टर्स बैठे हुए थे, कुछ क्लिनिकल साइकोलॉजिस्ट बैठे हुए थे, उन्होंने, क्योंकि हम यही सोचते हैं कि भाई ये बच्चा, इसको समस्या है तो ये आसान सा सवाल पूछा जाए। उन्होंने पूछा, ओके बेटा बताओ 2 गुना 2, एंड द पर्सन हु वाज कोऑर्डिनेटिंग देयर वो कहते आप मेरे बच्चे की बेइज्जती कर रहे हैं सवाल पूछ के, ऐसा सवाल नहीं चलेगा, और थोड़ा मुश्किल सवाल पूछिए। उन्होंने कहा ओके 5 गुना 5, कहते छोड़ो आप, मैं सवाल पूछती हूँ। उन्होंने पूछा 6 डिजिट की एक संख्या मल्टीप्लाइड बाय 6 डिजिट की एक संख्या, बताओ बेटा। एंड ही टोल्ड विद इन सेकेंड्स, ऐसा था। मेरे पास वो वीडियो अभी भी है उस समय का। आई वॉज अमेज्ड। तो ये बच्चे, इन बच्चों के अंदर स्पेशल कैपेबिलिटीज हैं जो एक हम सब के अंदर भी नहीं है, उससे कहीं ना कहीं ज्यादा भी है। तो वही बात उतनी है कि कोई हाइट में छोटा है और कोई हाइट में जरूरत से ज्यादा लंबा है, तो कई चीजों के अंदर उनका दिमाग हम लोगों से ज्यादा भी है। तो इस पहलू को समझना भी आवश्यक है, अगर हम उनको समान धारा में लेके आना चाहते हैं, या फिर उनको समाज का हिस्सा या समाज के इंपॉर्टेंट योगदान देने वाले पर्सन में डालना चाहते हैं। मैं ऐसे साइंटिस्ट का भी आपको नाम बताऊंगा कुछ देर में, जो कि जिन्होंने इस ऐसा रिसर्च भी की है। प्लीज मैम।
वांटेड टू ऐड, सर ने भी बताया ना डाली पे इतने साल से काम हो रहा है, वो कर रहे हैं। मीनवाइल जो दूसरी वाली बीमारियों का जो है, जो ऑटिज्म है, जो डीएचडी, हाँ, एपिलेप्सी, सीपी वगैरह का एक ऐप है ऑलरेडी, लेकिन वो डॉक्टरों के लिए है यार, प्रोफेशनल्स के लिए, साइकोलॉजिस्ट वगैरह के लिए है। पर अभी हम लोगों ने कुछ शुरू किया जिसमें जो स्क्रीनिंग है, जिसमें आपके लिए हो सकता है स्क्रीनिंग टूल, जिसमें स्क्रीनिंग होता है कि जो आपके बाकी पॉपुलेशन, जो बच्चे आपके स्कूल में उसमें लगाओगे कि उसमें से पकड़े आए, तो वो टूल्स बन गए हैं, लेकिन वो ऐप में नहीं डाले हैं, उनको भी ऐप में डाल रहे हैं।
और उसके बाद डायग्नोस्टिक रूल साथ में होगा। और हमने कुछ वीडियोस बनाए, आप कह रहे थे आगे क्या करेंगे, आगे हमने वीडियोस बनाया कि उसके लिए क्या करना है, वो लिंक कर रहे हैं। वो WHO के साथ काम चल रहा था, थोड़ा रुक गया था फंडिंग के लिए, तो उसमें लिंक हो जाएगा, तो आपको वो इन्फॉर्मेशन मिलेगी। इन एडिशन सर ने तो बोला SCRT के थ्रू भी होगा, पर आपको एज अ ऐप होगा फॉर पेरेंट्स ऑल्सो, एनीबडी कैन बी यूजिंग दैट। एंड वो 7 क्वालिटीज की बात कर रहे थे, एक्सेप्शनल क्वालिटीज है। आप डेट ऑफ बर्थ बताओगे तो डे ऑफ द बर्थ भी बता देते हैं। तो ये है कि एलन मस्क को भी ऑटिज्म है, वो पहला ट्रिलियनर होने वाला है। आइंस्टाइन को, मैरी क्वेरी, आइजैक न्यूटन, मोजार्ट, इन सबको था, उस समय डायग्नोज नहीं था, रेट्रोस्पेक्टिवली देखा गया है। एंड आइजैक न्यूटन, उसको उसके पढ़ाना नहीं आता था, तो वो क्लास से सब भाग जाते थे, कहीं होके भाग जाते होंगे ना स्टूडेंट है, तो वो, लेकिन देखो हमारे पास एक दो भी स्टूडेंट आते हैं तो हम पढ़ा लेते हैं, कई बार होते हैं बंक मारते हैं, लेकिन उसके पास कोई स्टूडेंट भी नहीं आता था, खाली लेक्चर में पढ़ाता था, बिकॉज उसको है कि मुझे पढ़ाना ही है, फिक्स्ड इंटरेस्ट। आप समझ रहे, तो अभी रेट्रोस्पेक्टिवली देखा है, तो इतने बड़े बड़े जो लोग हैं जिन्होंने बहुत सारा कुछ किया है, और अभी तो एलन मस्क आप देख रहे हो, उनको भी है। तो बस ये है कि हमें उनको आइडेंटिफाई करना है। और जो एग्जांपल आप बता रहे थे क्या करना है, जैसे ऑटिज्म वालों को विजुअल क्यूज ज्यादा है, तो अगर सेम चीज आप बोल लोगे 10 बार, उसकी जगह उनको पिक्चर से दिखाओ, ज्यादा समझ आएगा। जो विजुअली इम्पेयर्ड है उनको जो अक्षर वगैरह सब है वो बड़े साइज वाले दिखाओगे तो है। आप समझ रहे हो, हियरिंग वाला इंश्योर करो उसको हियरिंग एड मिले। कई लोग, आप समझ रहे हो, कई पीछे बैठे हैं, वो अटेंशन नहीं पे कर रहे हैं, उनकी आई टेस्ट करानी है कि वो नजर ही नहीं आ रहा उनको। तो इसलिए इंपॉर्टेंट है कि पूरा इवैल्यूएशन डॉक्टरों से भी उस समय, तो आप बहुत इंपॉर्टेंट रोल है आप लोगों का, सिर्फ ये नहीं, नजरअंदाज नहीं करना।
एक चीज मैं, हाँ उधर कोई आप प्रश्न पूछना चाहते हैं, तो एक चीज मैं आप सबको निवेदन करूंगा कि ये है कि ये बच्चों को आपको सबकी तरह एक नहीं बनाना है, हर बच्चे को अलग बनाना है। हर बच्चे की अलग क्षमता है, हर बच्चे की एक अलग पहचान है, और वो अगर आपको उनका जो खूबियां है उनको आपको पहचानना है। उनको क्या कमियां है ये आप मत ढूंढिए, ये ढूंढिए कि उसको क्या खूबियां है, और फिर उससे उसका जितनी सारी और कमियां हैं ना वो सब भर जाएगी, उससे कोई प्रॉब्लम नहीं होगी। तो इसीलिए आपको थोड़ा मतलब अलर्ट रहना है और देखना है कि बच्चे का स्पेशल क्या है, वो अलग बच्चे से क्या ज्यादा करता है। ये नहीं देखना है कि वो अलग बच्चे से क्या कम करता है। हाँ, उसके ऊपर आप ध्यान दीजिए, जो वो नहीं करते हैं उसके ऊपर ध्यान मत दीजिए। अगर उनको मतलब लिखना नहीं आता है तो वो लिखना सीख जाएंगे। ऐसा नहीं है कि उसको सबकी तरह एक ही साथ लिखना चालू करना है। अगर उसको कैलकुलेट करना नहीं आता है तो ठीक है, जाने दीजिए, कैलकुलेट नहीं करिए। तो ऐसा करके आप उसको मतलब प्रोत्साहन दीजिएगा कि वो जो अच्छा करता है उसको जरा उसके ऊपर ध्यान दीजिए, उसको मतलब इनकरेज कीजिए, प्रोत्साहन दीजिए कि वो और ज्यादा अच्छा करे। फिर बाकी जो है उसका अपने आप वो कर पाएगा। क्योंकि जैसे मल्लिका जी ने कहा कि कॉन्फिडेंस होनी चाहिए बच्चे के पास, तो कॉन्फिडेंस देना आपका काम है। आप उनको कॉन्फिडेंस दिलाएंगे कि वो कर पाता है। उसको मत बताइए कि वो नहीं कर पाता है, उसको ये बताइए कि वो करता है।
और एक है उसको पॉजिटिव रीइंफोर्समेंट, पर स्कूलों के लिए पनिशमेंट नहीं देनी है, पॉजिटिव रीइंफोर्समेंट से जाना है। जो अच्छा कर रहा है उसके लिए उसको रिवॉर्ड देना है। जो पनिशमेंट इज नॉट गिविंग रिवॉर्ड, दैट्स ऑल। वो जो स्केल स्कूल में हमारे टाइम मारते थे ना, वो सब नहीं मारना है अभी। है वो नेगेटिव वाला नहीं होता, नहीं नहीं वैसे बोल रही हूँ अभी तो, खैर आई एम जस्ट सेइंग पनिशमेंट इज जस्ट नॉट गिविंग अ रिवॉर्ड, इज पनिशमेंट, दैट्स ऑल। यू कांट गिव, यू हैव टू गिव रिवॉर्ड फॉर एवरीथिंग पॉजिटिव, अप्रिशिएट, है ना, पर वो पनिशमेंट नहीं है। अब एक और चीज बताओ, प्रेवलेंस बहुत ज्यादा है, 12% प्रेवलेंस है ये न्यूरो डेवलपमेंटल डिसऑर्डर्स का हमारे इंडिया में। हमने स्टडी की थी 2011 में, इंक्लूड स्टडी 2 से 9 साल की उम्र के बच्चे जो आपके प्राइमरी में आते हैं, 12%, जिसमें 1/5 में एक से ज्यादा परेशानियां थी। तो वो आपके सामने आ नहीं रहे। गैजेट ऑफ इंडिया और एनएसएसओ में 2% आता है, क्यों? स्टिग्मा। लोग बताना ही नहीं चाहते, जब आपके घर में कोई आ रहा है आपसे लेने के लिए गैजेट, स्टिग्मा की वजह से कोई बताना नहीं चाहता है। हमने जब स्टडी किया 12% था। आप समझ रहे हैं, तो आप ही को पकड़ना है, उनको आइडेंटिफाई करना है, और जो भी उनकी क्षमता है वहां ले। हमारे कई बच्चे हैं ऑटिज्म के जो सिंगिंग करते हैं, उनको कई है जो प्रोफेशनल सिंगर से कम्पीट कर रहे हैं, प्रोफेशनल जो जिनको कोई परेशानी नहीं है। पेंटिंग्स हैं उनकी, एग्जीबिशंस लग रहे हैं विद सारे लोगों के पेंटर्स, जिनको प्रॉब्लम है और जिनको नहीं है, सबके, यू नो। तो इट हैज टू बी नॉट अ हॉबी, टू मेक एन अ प्रोफेशन। आप समझ रहे हैं, दैट इज व्हाट।
यस, हाँ, आपका सवाल प्लीज, माइक उधर दीजिएगा। आपके नेक्स्ट आपके पास ही आएंगे जी।
माय नेम इज
अच्छा।
प्रीति फ्रॉम जीजीपीएस बिसर अकबरपुर। मेरा क्वेश्चन ये है कि हमें ये कैसे पता चले कि जैसे आपने बोला ऑटिज्म जो है एक बिहेवियर के थ्रू हम बच्चे को करेक्ट कर सकते हैं, बट समटाइम्स हमें मेडिसिन भी प्रोवाइड करा के ऑटिज्म नहीं होता वो बच्चे को, पर मेडिसिन से वी कैन हेल्प देम आउट। सो हाउ वी विल फाइंड इट आउट कि मेडिसिन के थ्रू होएगा या हमारे बिहेवियर के थ्रू हमें उसे रेगुलेट करना है?
आप बताना चाहेंगे?
नहीं नहीं मेडिसिन
बैठिए प्लीज। देखिए, मैं आपको ये कहूंगा, सबसे जरूरी बात तो ये है कि हम उसको अर्ली डायग्नोज कर पाए कि उसका डायग्नोसिस क्या है। डायग्नोसिस करने तक हम इस बारे में कहीं किसी को कमिट ना करें कि ना ही तो उसके ऊपर कोई लेबल लगाएं कि इसको तो ये बीमारी है। जब तक वो डायग्नोसिस ना हो कोई हम लेबल ना करें, और साथ के अंदर ये भी ना कहें कि ये दवा दुआ क्या काम करेगी। बहुत से केसेस में क्या रहता है कि गलतफहमी बड़ी जल्दी पैदा हो जाती है, और सबसे मुश्किल, एज ए डॉक्टर मैंने आई हैव सीन लैक्स ऑफ पेशेंट, सबसे मुश्किल केस या सबसे ज्यादा टाइम किस पे लगाना पड़ता है? जो केस ठीक करना मुश्किल है, जिस जो ठीक होने वाला है, फिट्स का केस है, लेवेट्रास्टेम 500 मिलीग्राम बीडी लिखा और भेज दिया, 2 मिनट भी नहीं लगी। उस केस पे टाइम लगेगा जो केस ठीक करना मुश्किल है, और वो बड़ी लंबी चौड़ी उम्मीद लेके आए होते हैं कि शायद ये डॉक्टर ठीक कर दे, और वो यही सुनना चाहते हैं कि जी ये कह देगा ठीक हो जाएंगे। तो उनकी उम्मीद भी टूटती है ऐसी स्थिति के अंदर कि दवाई तो है नहीं इस चीज के लिए। ऐसा नहीं कहा जा सकता कि आने वाले समय में दवाई नहीं बनेगी, लेकिन झूठी उम्मीद भी नहीं होनी चाहिए। और इस चीज का कौन फायदा उठाते हैं? जैसे कैंसर के केस में आपने सुना हो, कैंसर वाले बाबा बन गए। इस तरह के बहुत सारे बाबा बैठे हुए हैं कि जी हम ऐसी जड़ी बूटी देंगे और उससे सारी बीमारियां ठीक हो जाएंगी, हर बीमारी की उन्होंने। तो इस प्रकार की चीजों का लोग फायदा भी उठाते हैं, जो बीमारियां ठीक करनी मुश्किल है, या जो इस प्रकार की स्थिति है जो कि बीमारी ही नहीं है, वो उनका व्यवहार है, उस सबको आइडेंटिफाई ना कर पाने की वजह से इस प्रकार के लोग जुड़ जाते हैं सिस्टम के अंदर, जो उसका गलत फायदा भी उठाते हैं। यस, तो मैम, यस मैम, आई वुड कम टू मैम, बीइंग ए डॉक्टर शी कैन रिप्लाई बेटर प्लीज।
कुछ कंडीशंस हैं जिसके अंदर आपको दवाई देनी होती है, तो इसलिए, और मैंने ये भी बताया था कि 12% में से 1/5 में एक से ज्यादा परेशानियां होती हैं ऑटिज्म के साथ, हाइपर एडीएचडी, मेनी ऑफ द कॉम्बिनेशंस आर देयर। तो आपको एक बच्चों के डॉक्टर को, अगर बच्चों का दिमाग का डॉक्टर है तो उनको दिखाना है आसपास में, नहीं तो कोई डेवलपमेंटल पीडियाट्रिशियन, अगर वो नहीं है तो पीडियाट्रिशियन ही से, ताकि बच्चे को पूरी तरह से देखा जाए। कोई ऐसी दवाई नहीं है जिससे मानसिक क्षमता बढ़ती है। ये फालतू की जो मेडिसिन लोग लेते हैं, 2-2 हजार के सिरप्स एंड ऑल दैट, वो सब उनके ऊपर कोई रिसर्च नहीं हुआ है, एविडेंस नहीं है। तो आप, एक चीज ये है कि दिमाग बढ़ाने की दवाइयां नहीं है, दवाई की इंडिकेशंस हैं। किसी को साथ में दौरे पड़ रहे हैं, किसी के साथ में बहुत ज्यादा हाइपर एक्टिविटी है जो बिहेवियर से, इंटरवेंशन से ठीक नहीं हो रहा है, इनअटेंशन ज्यादा है, अग्रेसिवनेस ज्यादा है, या स्लीप ऑनसेट लेटेंसी, आप बच्चे को सुला रहे हैं और सो जाते हैं ना बच्चे 5-10 मिनट में, अगर ज्यादा टाइम लगा रहे हैं सोने के लिए तो एक दवाई आधा घंटा पहले मेलोसेट दी जाती है। तो ये कुछ कुछ कंडीशंस हैं जब दवाई दी जाती है, वो देनी है, एंड उसके ऊपर भी एडिक्शन नहीं होता है, उसको लंबी तौर से नहीं होता। एंड देयर इज अ प्रॉब्लम जब हम लोग दवाइयां देते हैं, प्लीज माइंड नहीं करना, थेरेपिस्ट लोग कहते हैं आप दवाई मत दीजिए, हम वैसे ही ठीक कर देंगे, सो दे हैव नॉट एनी राइट टू इंटरफियर विद मेडिसिन, बट वो इमोशनली प्ले करते हैं, सब नहीं कुछ करते हैं, दैट इज व्हाट आई फेस। और पेरेंट्स को भी मन में वहम है, ब्लड प्रेशर की दवाई खाएंगे, डायबिटीज की दवाई खा लेंगे, शुगर की, दिमाग खाली वाली दवाई में टैबू है, अगेन स्टिग्मा है, फालतू के वहम है कि भाई डिपेंडेंसी होगा, वो समझना है। पर जिन सिचुएशन में चाहिए वी आर गिविंग, दैट हैज टू बी डन, पर हर एक में नहीं चाहिए। मेन से ऑटिज्म वगैरह का बिहेवियर वाला है, पर ये समझिए जिसकी मानसिक क्षमता कम है, वो मानसिक क्षमता को बढ़ाने की कोई कोई टॉनिक नहीं है। 1 मिनट, ऑटिज्म मिमिक्स कुछ होते हैं जिसमें कुछ व्यवहार की प्रॉब्लम्स होती है, तब हम एक स्लीप और अवेक ईजी कराते हैं और उसकी कुछ स्पेसिफिक ट्रीटमेंट होता है। तभी जरूरी है कि बच्चा सिर्फ थेरेपिस्ट या स्पेशल एजुकेटर ही ना देखे, पीडियाट्रिक न्यूरोलॉजिस्ट या डेवलपमेंटल पीडियाट्रिशियन को देखना भी जरूरी है, बिकॉज वो ये सारी प्रॉब्लम्स होती है।
बहुत अच्छी बात। तो मैं एक पहलू आपको बताना चाहूंगा। हर जिले के अंदर, भारतवर्ष के, आप ही आएंगे, भारतवर्ष के हर जिले के अंदर एक डिस्ट्रिक्ट अर्ली इंटरवेंशन सेंटर बना हुआ है, हर जिले में। हरियाणा में तो मैं कह सकता हूं कि 22 के 22 जिलों में बना हुआ है। उसमें कौन लोग काम करते हैं? उसमें एक पीडियाट्रिशियन होते हैं, उसी के अंदर एक क्लिनिकल साइकोलॉजिस्ट बैठे होते हैं, उसके अंदर एक फिजियोथेरेपिस्ट भी बैठे होते हैं, उसी के अंदर एक अर्ली एजुकेटर भी बैठे होते हैं। तो ये सब लोग मिलके एक जगह पे काम करते हैं, एंड इनफैक्ट इट इज ए टीम वर्क। तो टीम के अंदर काम करें जिसके अंदर साइकोलॉजिस्ट, मेडिकल प्रोफेशनल, साइकेट्रिस्ट, पीडियाट्रिशियन, ये सब लोग मिलके काम करें, और एजुकेशनिस्ट भी इसके अंदर रोल अदा कर सकते हैं। सब लोगों का कॉमन रोल है। आप में से मेवात में या गुड़गांव में काफी सारे स्कूल ऐसे होंगे जिसके अंदर अर्ली एजुकेटर या जो आपके स्पेशल एजुकेटर हैं वो उसके अंदर जुड़े हुए होंगे। स्पेशल एजुकेटर्स हैं कुछ स्कूलों में आपके यहाँ पे, आप में से कुछ स्पेशल एजुकेटर आए भी हुए होंगे। तो काफी सारे स्कूल ऐसे हैं जहां पे स्पेशल एजुकेटर जुड़े हुए हैं। आपका सवाल मैम प्लीज, माइक पास करिएगा। पहले आप नहीं, वो सही जगह बैठ गए अब की बार।
सर, माय नेम इज रवि फ्रॉम
जी रवि जी,
जीजीपीएस हसनपुर सावड़ू। सर, मेरा सवाल ये है कि क्लास में सारे बच्चों का आईक्यू लेवल एक जैसा नहीं होता। कुछ बच्चों की ग्रास्पिंग पावर बहुत अच्छी होती है, कुछ की उतना गेन नहीं कर पाते। लेकिन जो गेन कर रहे हैं हम बता रहे हैं, लेकिन उसके बावजूद वो एक्सप्लेन कर देते हैं तुरंत साथ साथ। बट जैसे एग्जाम टाइम आता है या फिर समझो नेक्स्ट डे उनसे कुछ पूछते हैं, नेक्स्ट डे तो आई थिंक वो बता देते हैं, लेकिन एक सप्ताह बाद जैसे हम उनसे कुछ पूछते हैं तो वो बिल्कुल कुछ भी नहीं बताते। मीन्स, लेकिन मैं बताती हूँ कि अगर उनको वैसा थोड़ा सा इंसिस्ट कर दिया जाए, थोड़ी सी हेल्प कर दी जाए, तो फटाक से सब
जी।
कुछ बता देते हैं। और कुछ टाइम के बाद तो बिल्कुल ही वो इरेज हो जाते हैं, माइंड से सब कुछ स्किप हो जाता है उनके। और एक तो इस तरीके के बच्चे होते हैं, और एक इस तरीके के बच्चे भी होते हैं क्लासरूम में कि बता बहुत अच्छे रहे हैं लेकिन लिख नहीं पा रहे हैं उस चीज को। अगर उनसे वही चीज पेपर
वर्बली
और
बता देंगे।
हाँ जी वैसे बता रहे हैं, और मैं अगर उनको कहूँ तो वो मेरी क्लास के इंटेलिजेंट बच्चे मैं उनको बोलूंगी, लेकिन वो वैसे एग्जाम टाइम में कुछ भी नहीं कर पा रहे हैं। और मेरी एक खुद की भी पर्सनल प्रॉब्लम भी है, मैं आपको बताती हूँ कि आपके साथ शेयर कर रही हूँ कि मेरा खुद का जो बच्चा है वो साइंस में उसको क्लास टीचर गूगल बाबा बोलती है कि कुछ भी पूछ लो आप उससे, कुछ भी। क्योंकि ये फोन जो है ऐसे अब बच्चे देखते रहते हैं, उनसे सीखते रहते हैं, सुन लिया मतलब बहुत कुछ है उसके ब्रेन में। लेकिन क्लास में जब वो रिटर्न होता है ना, तो उसमें उसकी वो परफॉर्मेंस नहीं आती जितना उसको नॉलेज है।
जी जी
तो आपने जो स्टार्टिंग में एक स्टोरी बताई थी कि मेरे पास एक ऐसा बच्चा आया, तो मेरे को फट से अपने बच्चे का ध्यान आया।
जी जी जी, ठीक है रवि
मुझे
जी,
तो
आपका जी
मुझे
जी,
तो
थैंक यू
आपसे
मैम,
मिलना
थैंक
ही
यू।
पड़ेगा
जी जी
शायद
जी, थैंक यू,
शायद
थैंक
से मेरी प्रॉब्लम शॉर्ट
जी
आउट
जी।
हो जाए आपसे मिलके।
मैं आपको बहुत आपने सभी के लिए देखिए सबके मन में उत्तेजना पैदा की है। आपके सवाल के लिए
और
धन्यवाद।
मैं तो सर
जी जी
स्टार्टिंग में आई थी तो मेरे को लग रहा था कि शायद ये इतना ज्यादा इंटरेस्टिंग नहीं होगी क्लास, लेकिन आपसे बात करके और आपको सुन के मेरे को बड़ा मजा आ गया।
चलिए,
और
धन्यवाद आपके।
ऐसा लग रहा है कि जैसे
धन्यवाद मैम।
बहुत कुछ प्रॉब्लम
जी
सॉर्ट
जी
आउट होने वाली है।
चलिए, बहुत अच्छे मैम। आपके इसी सवाल से एक बात सुनाते हैं कि एक जंगल के अंदर हाथी भी है, बंदर भी है, एक जंगल के अंदर तालाब है उसमें मछली भी है, अलग अलग जागर सभी जानवर हैं। तो जैसे हर जानवर अलग है, ऐसे ही हर बच्चा भी अलग है। अब हमने जो टेस्ट लेना है, हम मछली को तो ये बोल दे भाई तू हाथी की तरह वजन उठा के दिखा, हाथी को यही बोले कि तू पानी के अंदर मछली की तरह तैर के दिखा, और या फिर हाथी को बोल दे कि बंदर की तरह पेड़ पे चढ़ के दिखा। तो हर बच्चे की यूनिक क्वालिटीज हैं, उसको उसकी यूनिक क्वालिटीज के लिए तो प्रमोट करना ही चाहिए। हो सकता है कुछ बेसिक स्टैंडर्ड्स हैं जो चीज सबको सीखनी चाहिए, उस चीज को सीखने के लिए थोड़ा सा और ज्यादा ध्यान देने की आवश्यकता है। और कुछ बातें ऐसी हैं जो मैम ने जैसे बताया कि मेमोरी जो है, यादाश्त है, वो उसके कई सारे मेथड्स हैं। खाली सुनने, कई बच्चों को सुन के याद होता है, कई बच्चों को देख के विजुअल मेमोरी अच्छी होती है, किसी के इसी चीज के लिए उसको रिवीजन की आवश्यकता रहती है। तो और कोई सब्जेक्ट में कोई ज्यादा अच्छा हो सकता है, कोई दूसरे सब्जेक्ट में ज्यादा हो सकता है। तो हम लेवल ना करके यही समझे कि बच्चा मुख्य धारा में इसको लेके जाना है और इसकी जो क्वालिटी है उसको उसी सब्जेक्ट के अंदर प्रमोट करना है। इस भावना से हमें आगे चलना चाहिए। सर, आपका सवाल प्लीज, आपका सवाल काफी देर से इंतजार कर रहा है।
Thank you. My name is Lakshmi Narasimhan. I teach at Amity University, Gurugram, right here.
Yes.
Today's sessions have been wonderful right from the beginning. I had a specific question for Verma madam, but I think it's for everybody. You mentioned about social stories, and you showed a lot of pictures, and I wonder, are these resources available in Hindi and other languages of India, or they're being prepared? How can they be made available? Thank you.
Yeah, see, you have only issue with the language, only with the language.
No,
the thing you understand, what is the social story, how do you use it, and why do you use it, and how it is written, how it is like used. When you have all understanding of that, then only your issue is the language, whether it is available in the original languages, some standard stories. You may Google and find it out, मिल सकता है आपको, मगर when you know the way and the why you want to write it, how you want to write it, how it is effective, then you can do it in any language by yourself. It's just इंग्लिश में है उसको हिंदी में कर दिया, हिंदी में उसको हरियाणवी में कर दिया या तमिल में कर दिया, but you know how to do it and how to write it and how to use it with the child to promote social interaction and all. ठीक है?
Thank you.
Thank you. So nice, ma'am. So it is going to be the last question because the time is a constraint.
Hello.
आवाज आ रही है मैम? Please. Yes.
Hello. Payal Gupta from Bal Bharti Public School, Manesar. So I am taking care of pre-primary kids. तो पता है कि हम उनको डिटेक्ट भी करते हैं, अपनी काउंसलर से भी डिटेक्ट कराते हैं, बट जब पेरेंट्स को बुलाते हैं वो एडमिट नहीं करते। 2-3 इयर्स ना ऐसे ही पास ऑन हो जाते हैं, जब वो थोड़ा सा एज में पहुंचते हैं 7-8 इयर्स में, तब वो उनको लगता है बहुत ज्यादा, तब वो एडमिट करते हैं। ऐसा कुछ हो कि स्कूल लेवल पे हम एडमिट करके कोई ऐसा गवर्नमेंट कुछ हो तो हम उनको ही लिस्ट कर दें और वो उन पेरेंट्स को जाके अप्रोच करें, क्योंकि वो हमारी नहीं मानते। और 2-3 एवरी क्लास 2-3 किड्स होते हैं।
आपने बिल्कुल सही ये सही समस्या बताई, सचमुच में ऐसा होता है, लेकिन उसका कारण क्या है? आप उसके पीछे कारण पे जाएंगे तो आपको ये महसूस होगा उसका कारण स्टिग्मा है। पहली बात तो ये कि एक्सेप्टेंस नहीं आती, कोई पेरेंट नहीं सुनना चाहता कि मेरे बच्चे को ऐसे कैसे हो सकता है, कि मैं इतने इंटेलिजेंट हूं, मेरे बच्चे को कैसे दिक्कत होगी, हो ही नहीं सकते, हम तो फलाने खानदान के, हमारे बच्चे को कैसे दिक्कत हो गई। वो किससे लड़ेंगे? वो टीचर से लड़ेंगे कि मेरे टीचर को, इस टीचर ने मेरे को बोल कैसे दिया, ऐसे कैसे बोल दिया। हो सकता है 3 साल बाद वो मान भी ले उस बात को, लेकिन तब तक 3 साल खो चुके होते हैं। तो इसीलिए जो मैम ने बोला था कि स्टिग्मा को कम करने की आवश्यकता है। जो ये हमने एक माहौल बनाया हुआ है कि ठप्पा लगा दिया इसके ऊपर कि इसको तो ये बीमारी है। इंटीग्रेटेड एजुकेशन का दूरगामी प्रभाव यही है कि स्टिग्मा खत्म हो जाएगा। एक ही क्लास के अंदर दोनों तरीके के बच्चे बैठे हैं, जिसके अंदर समान व्यवहार, जो मैम ने कहा कि समानुभाव हम रखें, तो समान व्यवहार सबके साथ हो, इंटीग्रेटेड एजुकेशन हो, तो शायद ये इसको एक्सेप्ट करना आसान हो जाता है। और इसको लेबल करने की बजाय हम अगर पहचान ले, एज ए टीचर पहचान ले, एंड वी कीप ए स्पेशल एजुकेटर इन द क्लास, यस, तो वो भी इसका एक तरीका हो सकता है। मैम कुछ सुझाव देना चाह रही है, प्लीज।
ये रोज की प्रॉब्लम है, बट हमारे पास तो तब आते हैं जब आपसे पहुंच जाते हैं। आई थिंक हम लोगों को ना, वी शुड नॉट आइडेंटिफाई लेबल्स। हम लोगों को बोलना है कि बच्चा पढ़ाई में नहीं कर रहा है तो आपको दिखाना चाहिए कि उसमें और क्या है फुर्ती। सो डोंट गिव लेबल्स गिव स्टिग्मा गिव्स डिनायल एक्सेप्टेंस नहीं, वो डिनायल है, वो तो घूमेंगे, आप समझ रहे हो। और जब एक्सेप्टेंस आएगी तो वहां पे आएगा तेरे खानदान से आया, खैर वो अलग स्टोरी है, दैट्स नॉट फॉर अनदर, दैट्स फॉर अनदर डे। सो आई एम जस्ट सेइंग, सो यू डोंट गिव लेबल्स, यू से कि स्कूल में ठीक नहीं कर रहा है, प्लीज गो टू अ पीडियाट्रिक न्यूरोलॉजिस्ट / डेवलपमेंट पीडियाट्रिशियन / पीडियाट्रिशियन। यस, अगर जिसको ज्यादा ही बिहेवियर की बहुत ज्यादा प्रॉब्लम है तब साइकियाट्रिस्ट भी ऐड कर सकते हैं।
यस मैम, वी आर गिविंग, वी आर नॉट गिविंग।
राइट,
या that's okay. You say there are 3 basic environments the child is in. 1 with the family, 2nd in the school, 3rd is जब वो friends वगैरह से घूम रहा है। So the problems can be in one area only. मतलब ये बोल सकते हैं कि प्रॉब्लम इस एटमॉस्फेयर में, it doesn't mean कि टीचर पागल है या टीचर खराब है। आप समझ रहे हो? आप उनको सीधा समझाओ कि they are 3 in, it's no, I know you'll have to, it is your duty to do it. अब बोल रहे हैं ना, जब अनगढ़त है ना, स्टिग्मा है, उसकी वजह से एक्सेप्ट ही नहीं करेगा, डिनाइल में, नो, सो यू कैन जस्ट फ्लैग इट पोलाइटली, उनकी फीलिंग्स को भी सोच के जस्ट फ्लैग इट इन अ वेरी डिप्लोमेटिक वे एंड से कि 3 डोमेन्स हैं, बिहेवियर के 3 डोमेन्स हैं, बच्चे की एनवायरनमेंट 3 ग्रॉसली डिफरेंट है, फैमिली, फ्रेंड्स एंड स्कूल. So the problem can come in any one of the areas. Okay.
I understand.
You'll have to go perseverance.
Of course, I understand what you're saying, and thank you for the help. I have another question if we still have time. Well, actually, I just wanted to ask the panelists before, like you mentioned the developmental, because ma'am, you showed us one slide where there are 2 autism children which had different milestone about the development. So I just had to ask, like I encountered one person like who was checking on all the development milestone, everything, but still I suspect, like this is not a formal diagnosis, it is just on suspicion that the child is still autistic, but henceforth we have an age limit for the assessment and everything in future, but the milestone are all checked, the development are all checked, the only issues the person is facing is the social interaction, and when the social interaction and the repetition. So when I speak about the parents, because I don't focus on the social interaction, so I started talking about the repetition in speech, but the parents are not agreeing, like for them
Parents have to be told milestones are in 4 different domains. Gross motor, fine motor है, language and social adaptive. वो सिर्फ चलता चलता है, सुनता है, is the milestones for them. They are not understanding the social adaptive and language are also domains. Yes. So you first have to know yourself कि इतने domains में है, फिर उनको समझाना पड़ेगा कि आपके वो domain में lacking है, तो एक विषय है, so that we should take your child to the maximum potential. उसमें कोई चीज की कमी है तो वो identify की जाए. Positively बोलेंगे तो they will go.
Okay, ma'am. Thank you.
So how you put this in front of the parent, very crucial. अगर आप बोल देते हैं कि भाई इसका तो ये प्रॉब्लम है, ध्यान नहीं देता, भागता है, दौड़ता है, ऐसे करता है, डॉक्टर के पास ले जाओ. एज अ पेरेंट विल थिंक, बाप रे ये क्या हुआ, डॉक्टर के पास जाएगा, मेरा बच्चा तो ठीक ठाक दिखता है. सो हाउ यू पुट ऑल दिस in front of the parent really matters, और बच्चे का टाइम सेव करता है, आपका लाइफ सेव करता है, पेरेंट का लाइफ. सो you have to say it in a very like ma'am said, polite way, positive,
subtly मतलब चल के ही बताना है, मतलब
ma'am,
कि ऐसे करता है,
can we
not
do
only complaining all the time.
Excuse me, can we do something from education department, like parent orientation in all these things should be mandatory? They should be aware about all these things. Yeah,
teachers should also be aware.
Teachers are aware.
बिल्कुल मैम, एक्चुअली
Teachers are aware. As we are facing in our private schools, we are facing these problems. Parents are highly educated. Yeah, we do that, sir, but they are not coming for the sessions. They're not accepting that their child is having such kind of problem, and the way you people are not strict, that's why they are doing
Problem
that.
word को हटा दीजिए, no
Yeah.
problem.
ये बात सच है मैम, ये बात सच है. चलिए खैर, मैं बिफोर क्लोजिंग द सेशन आई वुड जस्ट टेल यू ए स्मॉल स्टोरी. एल्बर्ट आइंस्टीन की मैम ने अभी बात की थी. तो आइंस्टीन जो थे, आइंस्टीन का सभी ने नाम सुना हुआ, शायद जितने टीचर्स हैं सभी ने नाम सुना होगा कि he had been one of the person जो सबसे ज्यादा इंटेलिजेंट पर्सन जिनको माना जाता है, जिन्होंने बहुत सारी रिसर्च में योगदान दिया. तो शुरुआत में उनकी एक स्टोरी ये ऐसी है कि वो शुरू में बोलते नहीं थे, वो चुपचाप रहते थे. तो वो बोले नहीं, और क्योंकि कोई बच्चा नहीं बोल रहा तो उनको उनके घर वालों ने या फिर जो समाज ने उस समय पे जो सामाजिक ज्ञान था उसके माध्यम से उन्होंने घोषित कर दिया कि ये मंद बुद्धि है, इसको कोई ज्यादा दिमाग सा नहीं है. वो चुपचाप सा बैठे रहते थे. तो जैसे हमारे साथ रहता है कि समानुभाव बड़ा आसान, कहना आसान है, होता नहीं है. तो उसको भी कहते इसको क्या बोलना है, इसको इसकी साइड में रख दिया. और वो बोला नहीं, 4 साल का हो गया और अभी तक एबीसीडी नहीं बोल रहा, किसी से बात नहीं कर रहा. वन फाइन डे उसको गरम सूप दे दिया, एंड द मदर सेड कि पी लो, ड्रिंक इट. एंड ही सेड, डोंट यू थिंक द सूप इज टू हॉट टू ड्रिंक? तो दे वर सरप्राइज्ड टू लिसन कि यार ये तो 4 साल से बोला ही नहीं आज तक, सीधा ये लाइन ही, पूरी लाइन ही बोली है. तो सच किड्स कैन बी डिफरेंट, दे आर डिफरेंट. यस, हाँ.
टीचर का रोल है उसमें. स्टोरी ये है कि वो टीचर ने लिख के उसको एक पेपर में दिया कि मदर को जाके दीजिए. उसमें लिखा था, बाद में बताऊंगी क्या लिखा था. उसने टीचर के मदर को दिया, तो मदर से उसने पूछा क्या लिखा है, तो उसने बोला, उसने लिखा है योर चाइल्ड इज टू ब्राइट, हम उनको पढ़ा नहीं सकते, आप घर में ही पढ़ाइए. मदर टॉट द चाइल्ड, एंड उसके बाद के वो आइंस्टाइन बना। और उसके बाद उसने जब मदर मर गई थी बाद में उसने देखा, उसमें पता है क्या लिखा था? योर चाइल्ड इज क्रेजी एंड डोंट कम टू स्कूल फ्रॉम नेक्स्ट डे, अंडरस्टैंड। एंड वो माँ ने उसमें उसको बोला कि यू आर वेरी ब्रिलियंट एंड जीनियस, तुम्हारे लिए अलग से घर में पढ़ाया जाएगा। इट वाज द वे द पेरेंट्स हैंडल। सो मतलब उसे टीचर ने गलत किया, पर यहाँ टीचर का बहुत इंपॉर्टेंट रोल है, क्योंकि ये पेरेंट्स समझ ही नहीं रहे हैं। और आइंस्टाइन ने भी ये बोला है कि if you assess a fish by the ability to climb a tree, the whole life the fish will come keep on thinking कि वो पागल है। आप समझ रहे हैं? तो जिसकी जैसे आप सर ने एग्जांपल दिया था वो एनिमल्स का, तो वो आइंस्टाइन की सेइंग है, वो मेरी एक स्लाइड्स के प्रेजेंटेशन में भी एंड में। सो यू हैव टू अंडरस्टैंड कि हर बंदे का क्षमता है। आप एज टीचर्स उनको आइडेंटिफाई करें जहां वो कम है और उसके मैक्सिमम पोटेंशियल तक लेके जाइए, ठीक है? उधर जब एलन मस्क ट्रिलियनर बन सकता है तो पता नहीं कौन क्या होगा। मेरे को एक पेरेंट ने बोला रिसेंटली कि वो एलन मस्क की मम्मी ने क्या किया मुझे बताओ। मैं फैमिली काउंसलिंग करती हूँ ना, तो ग्रुप में बिठा के मैं भी वही अपने बच्चे के साथ करूंगी।
I have to add a very important thing here कि ये आइंस्टीन की मदर was one very special mother who changed things like that, पर सब पेरेंट ऐसे नहीं होंगे। एंड ऑल्सो आपके क्लास में जो बच्चे होंगे सारे आइंस्टीन नहीं होंगे, सारे एलन मस्क नहीं होंगे। सो बी रेडी फॉर द एवरेज इन ऑटिस्टिक और स्पेशल चिल्ड्रन स्टूडेंट, लाइक आई एम एवरेज पर्सन। तो एक ऑटिस्टिक है इसका मतलब ऐसा नहीं कि सब में सेवेंथ स्किल होगा, सब मैथमेटिकल जीनियस होगा, इट्स नॉट दैट। सो ये बात भी हमें ध्यान रखनी है। ये जो एग्जांपल्स है ना, मैडम क्यूरी, आइंस्टीन, ठीक है, बहुत अच्छा है, काफी इंस्पायर करता है, पर हमें ये नहीं सोच लेना कि हर एक बच्चा इज गोइंग टू बी आइंस्टीन।
मोस्ट ऑफ देम हमारे जो ऑटिज्म के बच्चे, मैं लास्ट 35 ईयर्स से ऑटिज्म क्लिनिक रन कर रही हूँ। वेस्ट में कहते हैं 40% में दिमाग की समझ कम होती है, 7 तो लेस देन 2% होता है, ठीक है? तो 7 जो बोल रहे थे एक्सेप्शन, लेस देन 2% है, बट आई एम सीइंग इन इंडिया मोर देन 70% में दिमाग में समझ कम है। ऑलराइट, so that is what the where we stand and try.
So ultimate goal is their inclusion, their happiness. उसी पे हमें काम करना है, एक उनको सारा राइट एज अ इंडिविजुअल सिटीजन की तरह।
Fine, ma'am, fine. Please.
Ultimate, I am telling one thing, that it is our duty for that particular person, mother, to tell something that yes, my son is autistic. I am telling you all that now in this stage, I am a proud mother of a person with autism. My only son is autism. Now I have that particular grip that I can say I am a proud mother of a person with autism. So I am telling you all that it is our duty to say all the person like that. And really I am a proud mother. It is not I am proud mother of a person with autism.
बहुत अच्छे मैम ने देखिए बड़ी अच्छी बात पर्सनल तरीके से बताई है। हम आपके कमेंट पे आएंगे। हम लोगों का उद्देश्य देखिए, बहुत सारे लोग ऐसे हैं जो शुरू में सवाल पूछने से झिझक रहे थे। हमारा उद्देश्य यही था टू क्रिएट ए स्पार्क, एक ज्योति जला पाए, हम सब लोग एक दीप जला पाए और वो दीप इतना उजाला करे कि समाज में फैला हुआ अंधियारा दूर हो जाए। जी प्लीज
सर, मैं दो बात कहना चाहता हूँ। मेरा बचपन का नंबर 1 क्या है, मेरा बचपन का तजुर्बा रहा है। जब मैं 24 साल का था, 24 साल में मतलब बचपन ही से स्टार्ट हुई थी लाइफ, तो बड़े बुजुर्गों से बहुत कुछ सीखा मैंने और उनसे सीखता रहा हूँ। और आज मैं स्कूल में कोई बच्चे परेशान होते हैं या कोई छोटी मोटी प्रॉब्लम होती है, जैसे ईसीजी करते हैं मशीनों से, मैं वैसे ही करके बता देता हूँ 1 मिनट में सारी पूरी बॉडी। और डिस्लिप सर्वाइकल साइटिका नस ब्लॉकेज, ये मैं बिल्कुल ठीक करता रहा हूँ अब तक। मेरा चैलेंज है, जो डॉक्टर नहीं समझ पा रहे, 8-8, 9-9 महीनों पर चल रहे हैं, मैं उनको बिल्कुल ठीक करता हूँ। मेरा अनुभव रहा है अब तक। मुझे 30 साल हो गए ये काम करते करते, 30 साल से मैं ये काम करता रहा हूँ जी।
जी जी धन्यवाद। आप पे भी आते हैं जी प्लीज। कोई नहीं, आपका आखिरी सवाल होगा जी, उसके बाद में वी विल क्लोज द सेशन।
अस्सलाम वालेकुम। आपका जो प्रोग्राम है बहुत अच्छा है और बहुत कुछ सीखने को मिला। मेरा एक सवाल है,
पहले तिब्ब की किताब किसने लिखी और वो कौन से देश का रहने वाला था और कौन सी जबान में वो किताब लिखी गई? मैं आप
किस विषय की जी?
तिब्ब डॉक्टरी की किताब है, जो साइंस की, जो जो
इंडिया से शुरू हुआ था सर।
है जी।
इंडिया से शुरू हुआ था ना।
मैं
साइंस की किताब बोल रहे हो ना?
हाँ,
मेडिसिन
साइंस की
वगैरह
किताब है,
सारा
उसमें
शुरू हुआ, संहिता
डॉक्टरी की किताब है।
हाँ, डॉक्टरी की किताब हिंदुस्तान से शुरू हुई थी भैया, बीसी में है वो।
हाँ, सुश्रुत संहिता, चरक संहिता भारतवर्ष से शुरू हुई थी। तो भारत की जो समृद्ध संस्कृति है, जिसने पूरे दुनिया को जागरूक किया है, उसका हम सब लोग हिस्सा हैं। और कहीं ना कहीं जिन्होंने किताब लिखी उनको भी किसी रूप में किसी टीचर ने ही पढ़ाया होगा। तो चाहे वो उस समय पे हम कोई गुरु बोल देते थे, लेकिन टीचर का जो रोल है वो बहुत ज्यादा सम्मानीय है, सम्मानजनक है। हम सब लोग मिलकर इस उद्देश्य से काम करें कि हमने एक समान इक्वल सोसाइटी का निर्माण करना है। इस इक्वल सोसाइटी में सभी के लिए जगह हो, और जो बच्चे हमारे पास पढ़ने के लिए आ रहे हैं, खाली उद्देश्य हमारा उनको कोई 10वीं पास कराने का, 12वीं पास कराने का नहीं है, बल्कि उनको भारत का एक संपूर्ण नागरिक बनाने का योगदान दे पाए, जिसके अंदर उनको सब कुछ सिखा के भेजे, खाली सिर्फ डिग्री या नंबर तक सीमित ना रह पाए हम। जी,
थैंक यू सर।
तो इन शब्दों के साथ सर, यू वांट टू से समथिंग? सो इस सेशन को खत्म करना चाहेंगे, और ओवर टू द ऑर्गेनाइजर, डॉक्टर प्लीज, डॉक्टर सौरभ।
सो एक फोटो खींचेंगे और उसके बाद लंच।
अच्छा जी,
मुझे लगता है ये पैनल डिस्कशन 1 घंटे का ना हो, शायद 2-3 घंटे का होता। बहुत लोगों के मन में सवाल बच गए हैं, मेरे खुद के मन में भी 2-4 सवाल बच गए हैं, पर कोई बात नहीं, अगली बार।
फोटो एंड लंच।
लेकिन आप सभी का धन्यवाद जो अपने समय से निकाल कर आप यहाँ पे आए। एज अ टोकन ऑफ थैंक्स, मैं डॉक्टर सौरभ से रिक्वेस्ट करूंगी कि हमारे पैनल मेंबर्स को a small token of thanks. I will.
फोटो बाहर खींचेंगे सबके साथ।
And Dr. Shanu. Thank you so much to everybody. And we'll proceed for the group photo and then lunch afterwards. Can we have a photo? If you wish, we can have photo here. जितने लोग हैं प्लीज आ जाइए स्टेशन। आप सभी के पास एक फीडबैक फॉर्म होगा, वो भरते हुए जाइएगा, कोई भी ले लो। तरन्नुम, इफ यू कैन प्लीज क्लिक अ फोटो। हाँ, वो हम ले लेंगे आपको। तरन्नुम विल क्लिक प्लीज, आप हाइट वाइज थोड़ा सा शेयर। मैं नहीं आ रही कहीं पे भी। सभी लोग आ रहे हैं। I think I have captured enough photos. फोटो तो ले लेते हैं वैसे तो बहुत ना पड़े सही है
That's important, and you will not understand till you know the clinical side of it. Yes, I don't want to understand the techniques, but I need to understand how you approach them so that that can be integrated. And I think one thing is also there with me recently, last couple of years, we have two first authors with equal weightage, two corresponding authors, so that people don't want to share the research because one wants the corresponding. Even in like we have importance that whether how many are your first and corresponding authors in your promotions and all everywhere, and everybody wants. So you can always have now in most of the journals, good ones, you have first with equal weightage authors and you can have two corresponding. So in that way, two or even four people can have equivalent kind of representation when you do collaborative work. And even in patents, you can have joint. So it is not that you are going to lose on your research and you're not going to have a competitor because a clinician will not take over a scientist work and scientists can't take over clinician work. So it's always best. And I think 1+1 becomes 11, not 2. So I think that's why we have to look forward and try and make some change in their lives. And I really thank for the opportunity, and obviously the opportunity was given by Kishanu when he was there. So it's really nice. Thank you so much.
I think those who are standing outside the door, please come inside. सबको बुला लो अंदर. Anyway, so the as you have already heard multiple times, Saurabh has said about it. See, what I felt, feel even now, and felt at that time, is that our idea of research excellence, which you are all, excuse me, which you are all bred with, is actually a thing of the past. So if you think that if you publish a paper as a first author in Nature and Science, your life will be made, then you are wrong, absolutely wrong. Because you can actually look around, you can actually survey how many of those Nature Science first authors did anything in their life in any meaningful way. You will find it a very different story. So yes, it is good within a small community if you publish in Science and Nature and Cell, and you will get a high for some time, and after that everybody will forget, and you will also forget. So therefore, the PhD is not only about publishing in Science and Nature. That should come as a byproduct, but PhD is about contributing something substantive, and that substantive something should help the society. And none of us did that, to be honest with you, and me included. None of us did that, and therefore we really don't know how to do it. Very few of us can actually do it, and those of us who actually do it are regarded much beyond their tenure and the boundaries in which their peer group exist. So the idea is to excite all of you to think out of the box. Just not think about publishing your next Nature paper, but think about how whatever knowledge you want to gather, whether that knowledge has a value. So I'll tell you one story. This was the advice given to a great physician's advisor for his PhD thesis. And this physician was Charaka. So when Charaka went to his advisor, Shushruth, for his to decide what to do for his PhD. So his advisor told him that you go and find out whether there is any plant in this earth which is not useful for human being. That was his PhD problem. So Charak went through the entire Himalayan, sub-Himalayan region, Kashmir and the other areas, Hindu Kush and many other areas. He traveled extensively for 10, 15 years, and he came back with a simple answer. He said, I did not find a single plant which is not useful to human being. So this is what his research is, and the outcome of that journey was the treatise which helped to form the materia medica of Ayurveda. Okay, so therefore this kind of research, of course, is not very easy to do, and always requires an extraordinary caliber and effort, and not all of us will be becoming Charaka or Shushruth for that matter. But at least we can dream and we can try. And to do that, we need to also open our mind first. We should not only sit in a talk to find out what I can take to write on my paper, okay? Because that's our training. That's how we are trained to learn something and then to put it on a piece of paper, whatever we have learned. Learn, memorize, and reproduce. you have to come out of this cycle from learn, memorize, and reproduce cycle to learn, assimilate, and produce. And that is what is very important. And it's very easy to say, but it's very difficult to do. I haven't been able to do it in my 35 years of career. And I hope one of you or some of you will do it. And if you do it, then some way I will succeed with you. Okay. With all that, we will now get back to serious business. So shall I start? I introduced myself already, so let me start. I think this one has a unique property. Okay, so what I will do now in next 15 minutes is I'm going to tell you about a story, a paper that we are going to write. And after all that discourse, I'm still going to tell you how to write a paper. But it is interesting the discovery by itself, and I think it has some potential. And that's what we will discuss in next 15 minutes. All right. So I'll tell you about these two molecules. I'll tell you about this motor protein, which you have heard from me multiple times here. And I'll tell you about this lipid molecule, which is inositol. And I'll tell you how phosphorylation of an inositol can actually trigger a mass migration of vesicles towards the synapse. And I'll tell you how that is actually programmed during the development for the synaptic reprogramming of the brain. Of course, this is going to be in the context of Drosophila larva, but this is a fundamental understanding, and therefore I believe that it will have some implication in all animal nervous system. Okay, so this is nothing new to you. So I prepared this slide for the layman, but I just wanted to show you that this beautiful picture that Arpan and his colleagues made in the lab. This is the DTI image of the human brain. And these I want to also show you this neuron, which is going to be the centerpiece of our talk today. So this is the nervous system of Drosophila larva. It's very simple. It has those and what you see here in green are the sensory neurons. Come forward and sit here. Lots of. So this is the sensory nervous system of the fly, and this is the central nervous system of the Drosophila larva. So Drosophila larva does a lot of complicated functions. It has to search and find food. It has to detect the right kind of food. It has to go away from the predators, and it has to also sense the toxins and poisons to keep it alive. And also it has to eat for the first 3 days of its life, and after the 3 days of its life, it has to walk out of the food and form a pupal casing, and then after that pupal casing, this entire nervous system is going to be degenerated and reprogrammed and remade entirely as an adult brain. So this is what happens in this kind of insect, which we call holometabolous insect, like butterflies. So the larva is a feeding and behaving egg embryo kind of thing, and then out of that, or maybe more equivalent to a placenta, and out of that then comes out of the adult, which is there in some embryonic bodies, embryoid bodies called imaginal disc, and that those imaginal disc then grow and form the adult body. So essentially, what you see here is the placenta, and the embryos are inside in small distributed tissues. The only thing that doesn't change much, I mean, doesn't get completely degenerated, is the central nervous system. The central nervous system is reprogrammed and rearranged, and grown preferentially in certain part, and shrunk preferentially in certain other parts to shape the adult nervous system. And that's why studying how the central nervous system gets reprogrammed is very critical to understand how the adult nervous system is built. And as you know, as of today, the Drosophila is the first insect or first complex brain that has been fully visualized. So we have the 1 nanometer scale resolution image of the Drosophila brain, the entire brain. So every synapse and every molecular structure of the adult Drosophila brain has been now documented, compiled, and put into a database where you can visualize it. So you can see every neuron, every connection. In addition to that, the fly brain is also fully functionally visualized. So we know every neural circuit and what is their function, how they correlate to the output of the function. So this is the first of its kind, and the mouse brain is on its way, and hopefully in another year's time, the mouse brain will be fully visualized. So that is where we are going, and hopefully in some day we will visualize our own brain also. Okay. So today I'm going to tell about the other aspect of the brain, which is how these synapses are reprogrammed and pruned in a developmental context, and how the small signaling systems actually uh, trigger these developmental changes. And this is the typical synapse that you see here, which has these uh, areas which we call the docking zone, vesicle docking zone. The thing is in green, and you see these vesicles which come and dock and release the neurotransmitter, and there is a mitochondria, and then this is recycled in that process. Now this synapse structure is actively maintained by movement from the cell body, and this movement has to be fully choreographed and controlled so that the synapse is constantly supplied with lipid, proteins, and other nutrients, including the major macronutrients. And this process happens through the axonal transport. So understanding how the axonal transport happens and how is it regulated plays a very critical role in maintaining the homeostasis in the adult nervous system. And if this breaks down, and also there is a constant communication going on from the two ends of the neuron to control this process. And if this breaks down, then the everything, there is a domino effect that happens, and that ends up into a neurodegenerative disorders. But in between this extreme neurodegeneration and the homeostatic function, there is a lot of spectrum and a lot of gray area, and that is what we are trying to understand using the axonal transport modulation in the nerve. And the logistical challenge is the following. So the longest neuron in the axons in the adult brain in our head is more than 100 meters long. In one estimate it is 1,000 meters. So this is a 1,000-meter long axon which has to communicate with its nucleus. uh, the synapse which at the end of 1,000 meter has to communicate with the nucleus in real time. Okay, so if you are forming a memory, then the synapse has to expand its contacts, and to expand the contacts it has to stimulate the nucleus to express the RNAs for those proteins that make the synaptic contact, and it has to bring those proteins back into the synapse so that the extra contacts are made, because there is no enough space to store those proteins in the synapse in any form, and there is no evidence that it is stored. So how does this the nervous system manages these logistics? That's the question that I got fascinated with, and I wanted to answer that question. So how do the neurons solve the logistics problem? Now, typically, this logistics is managed by these kind of proteins known as motor proteins. And there are three of its kind which goes towards the synapse, and one of its kind which comes back from the synapse. And these motor proteins usually hydrolyze an ATP and then takes 1 nanometer step at a time, 8 nanometer steps at a time, and covers this distance. So imagine covering a distance of 1,000 meters using 8 nanometer step at a time. And typically the duty cycles of these motors are about about 100 to 200 hertz, which is about 160 100 to 200 steps per second, per minute, I'm sorry. So therefore it's a very in that sense a very slow process, but yet it is one of the fastest kind of process that we know in our physical world. And this happens through this microtubule network, which is heavily decorated with other proteins. So imagine a road full of potholes and obstacles and ruminating cows, and you have to walk through that, and the motors always do it throughout their lifetime. So this is no wonder, I mean, this is really a wonder rather, that how does we actually survive, and how does our nervous system actually work. Okay, and then do all these wonderful things and create so many wonderful things. So you can take out these motors. This is their work, and this is how it looks like, single molecules that you can see moving on microtubule, and it's very easy to do it in vitro, and you can measure these steps. And this process is also regulated. And this process is regulated at multiple steps. It regulates at the cargo interacting stage, and it also regulates at the motor interacting stage, motor microtubule interacting stage. And if these regulations break down, for example, there is a host of kinases and phosphatases that control this regulation. And this, if this regulation breaks down, there is a whole variety of different disorders that starts happening. So these are some of the examples, not all of them, but we now know a lot of such disorders which are caused due to dysfunctional transport in the nervous system. And for the last 26 years, I've been trying to look for this, the biblical holy grail of, how does the motor bind to the cargo? And we eventually end up identifying several such proteins which directly interact with the tail domain of the motor, but we still don't understand how it interacts and how does these proteins magically find affinity with each other at one point and then magically find quite repulsive of each other at some other point. So that's the whole logistics, that the motor has to bind the cargo in place of its manufacturing, and the motor has to deliver the cargo at place of its function. And usually at the place of the function, the cargo will have higher concentration than the place of its manufacturing. So how do you sort of reverse this chemistry of interaction between two proteins? Normally, the chemistry would drive the interaction when there are more, reactants together than there are less reactants together. And this process, we now recently, learned, and that is what I'm going to tell you a little bit about how does this happen. So it started with this identification that if you knock down kinesin 2 motors, the synapses become big and bloated, but they lose the contact sites. They lose those, the vesicle docking sites. You may not be able to see it here, but you might, if you concentrate a little bit, you will see a dark patch, which is the docking site. And you also have a dark patch somewhere here, as you see, arrow is shown here, but there are no vesicles. So here we have multiple such vesicles, but you see these very few vesicles. And so we were wondering why the synapses get so bloated, and why these vesicles disappear from the synapse. So to understand that, so this project started with a master student called Deepti Rai, and what she did was very painstakingly look through the entire larval stages from the beginning to the end, and she studied the density of synapses at every developmental stage. It was a very painful process because you have to dissect, you have to first stage the larvae and age them correctly, and then dissect, fix, stain, image, and then quantify. So what she found was, in a nutshell, is that there is this, larval nervous system goes through this reprogrammed restructuring. So initially, you see the density is a bit high, and then at a certain stage, the density comes down, and then again the density goes up. So this particular process was quite excited us quite a lot, and we saw that even this is the estimate of a protein called Brook pillow or ELKs, which makes the vesicle docking zone on the presynaptic terminal. So you see the amount of protein goes down, and then the amount of protein goes up. So we thought that this will be a very interesting way, the model to look at how the programmed synaptic restructuring happens in the adult brain. And then Asmita and the Simanti and others continued that, and they showed that, after a lot of search and miss, they showed that this particular molecule called RAB4, which is a small GTPase that is known as a traffic cop of the cell, which recruits the motor protein and drives them to their destination. Their level has an anti-correlation with the level of the synaptic protein. So as you see, when the synapses are more and Brook pillow is more, there are the RAB4 is less, and vice versa. So this suggested that maybe driving RAB4 towards the synapse could actually help to reprogram these synapses. And this was also done by Swagata earlier and showed that artificially, if you drive the RAB4 towards the synapse, synapses disappear. So then Kamal and Simanti did the correlation and found that indeed during this developmental period, the amount of RAP4 traffic goes up during the stage when the synapses are low, and the traffic comes down towards the synapse when the synapses grow back. So this shows that a very direct correlation between the axonal transport and programmed synaptic reprogramming. So then the question is, how exactly the cell manages to time these events, and what is the trigger? So usually the trigger is always a signal, which means that there will be some kind of a chemical ligand which will be secreted, which will bind to some receptor, and that receptor on the plasma membrane will signal the cell inside to drive this traffic. And then I'll have to tell you a very long story. So this is the preamble with which Kamal started his work, and he used this assay, which is the tracking the vesicle movement inside the axon. So what you see here is the bunch of segmental nerve axons, and what you see in various colors are different vesicles. And as you see, these movements are not always linear; they are quite saltatory. And they are quite indeterminate. So which means that some vesicle you will see goes one direction first, then reverses and comes back, and vice versa. But some vesicles you will see continuously move in one particular direction. So the cell body of of this nerve lies on this side. This is the side where the cell body is, and this is the side where the synapses are. So looking at this, you will be wondering, how does the cell actually manage to move anything to any destination with so much chaotic traffic that is going on? So then Kamal looked into this process, and I'll quickly skip this. This is the idea of RAP4, which recruits the vesicles. And again, after a few trials and error, he found out that if you stimulate these neurons with insulin, human insulin, at a physiological concentration, then these vesicles appear to move faster. And if you eyeball these two movies back and forth, you will probably appreciate what I am saying. You will probably see that the insulin stimulated nerve has more agitated movement inside this nerve. And then this has been quantified, and indeed we found that insulin stimulation does increase the anterograde movement of the vesicles, but the anterograde movement is not too much. There was only 10 to 15% increase in the anterograde vesicle movement, and there is a 10 to 15% decrease in the retrograde vesicle movement. And so this suggested that insulin can just tweak it a little bit, just give it a little push, and that push can make a 10% change. And Kamal also found that what happens after insulin stimulation is that the vesicles that are moving at a faster speed, at 2 microns per second in the median, are more than the vesicles which are moving at slower speed. So what I showed you earlier was the flux data, and this is the actual speed data. And the run length also was turned out to be higher along with this process, that insulin stimulation increases they run more longer than normally they will run in one direction. So then we tried to do this genetically. We found that again, if you knock down insulin receptor, you get the same effect. You get the reduction of the anterograde flux and increase of the retrograde flux, and the reduction of the small, the faster moving vesicles go down, and the slower moving vesicle population relatively goes up. And then we phenocopied the insulin stimulated data using insulin constitutively active, insulin receptor constitutive active. So which is kind of a mimicking the situation of insulin stimulation. And we did that same thing with the inhibiting the insulin signaling by using a PI3 kinase inhibitor. So all that suggested that indeed there is a physiological requirement of insulin that can stimulate the anterograde movement of these RAP4 vesicles. Now insulin signaling works in various ways. It has many different arms, and the most of them are slow arm, which makes the cell grow and divide, and that takes days. But there is a faster arm of signaling that happens rapidly by the direct phosphorylation of the lipid kinases called PI3 kinases. And insulin receptor is like all other tyrosine receptor kinases, are also kinases that can phosphorylate tyrosine residues. So they phosphorylate this, the PI3 kinases, and these PI3 kinases then phosphorylate the inositol lipids. So Kamal found out that in this case, insulin receptor phosphorylates VPS34 and activates the VPS34 activity, which makes phosphorinositide 3-phosphate. And this phosphorinositide 3-phosphate can be monitored using a biosensor called FIV-GFP. So then Simanti looked at how whether these RAP4 vesicles also have PI3 phosphate on them. And what you see here is the green-red co-localization of the biosensor of PI3 phosphate and the RAP4 M-cherry. And as you can see that the RAP4 vesicles also have a lot of PI3 phosphate on them. But you will also see here that the vesicles which are really bright green are not moving. Okay, and I'll come back to that later. And so because this the biosensor that made with FIV GFP domain will also mask the the head group of the phospholipids, and which will prevent its legitimate binding partners to access this phospholipid. So if you have too much of this biosensor on this vesicle, then it will act as an inhibitor of this process. So this suggested that the phospholipid localization on this RAP4 vesicle is a trigger for its faster movement. And this is just the quantitative data to show you that yes, if the relative amount of the phosphoinositol increases, the speed also increases within a certain limit, and it doesn't depend on the level of the RAP4 on this vesicle. So this suggested that the phosphoinositol is the driver of this faster moving vesicles. And we could also prevent that by reducing the adding the inhibitor of VPS34 in this process. So therefore increased speed of vesicle correlates to increased phosphoinositol 3-phosphate. Now these phosphoinositides act as a pin code in the cell. Okay, so typically the cell has these various compartments, lipid bound compartments, which requires these lipid bound compartments to be identified by different factors. And this phosphoinositol is the way the various molecules can identify these compartments, and this is called the phosphoinositide code of the cell. So what you see here is this various compartments of the cell, which is marked with different combination of phosphoinositides. And the proteins that bind to this VPS34 marks only the PI3P domain. And so we first thought that kinesin 2 could be the motor for this process, because there was some literature which suggests that RAP4 recruits kinesin 2, but we ruled it out. And then we came back to this particular motor called KIF16B. So KIF16B has been is a kinesin family motor, or called kinesin 3 family motor, and it has a PI3 phosphate binding domain called PX domain in its tail, and it can directly bind to PI3 phosphate. And most interestingly, the mutation in this KIF16B gene has been identified with intellectual deficit, congenital intellectual deficit syndrome. It's called ID syndrome, and therefore the KIF16B is a very interesting candidate for that matter, and it has been also shown to bind to RAP14, which is a RAP4 ortholog. So therefore we looked at this KIF16B ortholog in Drosophila, which is KLP98A, and these movies are a bit hazy because this is a endogenous level of the KLP98A directly tagged with GFP, and you can see that these RAP4 vesicles also have some green dots, and this is endogenous, and this is the overexpressed condition. So in the overexpressed condition, of course, you see a lot of these the red vesicles have a green dot on them, but usually the green dot is very small on the moving red vesicles and very large on the non-moving red vesicles, again for a similar reason. And then we found, when we knocked down the KLP98A, we found that the anterograde flux of RAP4 reduces, the high speed movement are also reduced, and we also found that it abrogates the insulin stimulated movement. I'm done here. Insulin stimulated movement in the cell. So therefore the next question, wait a minute, sometimes now it is not moving. So therefore, we came to the conclusion that VPS34 activated under insulin stimulation will recruit KLP98A, and then that recruitment will make this movement go towards the synapse. Now the final question is, does it really happen in the natural scenario? So far whatever I showed you is a stimulated artificial scenario where we have created these conditions to drive the process. So therefore, to understand whether it happens naturally, we need to go back to the same developmental system and ask whether during this programmed synaptic pruning, we do see an increased KLP98A and VPS34 activation. So look at the lower panel first. So this is the quantification of the PI3 phosphate sensor in the axon, and its level in the axon, and you see that the sensor level goes up in the axon during the process where the RAF4 vesicle moves faster towards the synapse, and when we see less amount of synapses. And that same thing phenocopies with the KLP98A level also. So therefore, in the developmental situation also, when naturally the synapses are pruned, we see an activation of the VPS34 signaling and the RAF4 movement towards the synapse. So that ties up the story from the beginning to the end and suggests that the developmentally regulated programmed insulin signaling can help to restructure the synapses. Now, what is the implication of this process? Okay, so insulin signaling in the brain, early endosome, and autophagy, these are all linked together. So PI3 phosphate and VPS34 is required to start the autophagy and recruit the autophagosome components on the lysosomal pre-lysosomal vesicles, and then those pre-lysosomal vesicles will migrate back from the synapse to the cell body to get autophagosed. Okay, and the KIF16B has this genetic connotation also. Now, most interestingly, the insulin signaling, which has been mostly studied in adipocytes, is responsible for, as you know, for diabetes. But more than diabetes, insulin signaling is also equally important for the nervous system function. Insulin signaling is known to stimulate the anterograde axonal transport through kinesins, and insulin signaling is also known to induce Alzheimer's like signaling. Okay, and therefore these three things is not very clear how it tied up. And as I show you here, that we observed in Drosophila that the RAF4 vesicle transport towards the synapse and the synaptic stability are inversely correlated. So this is the tie-up that we are trying to build together and synthesize this whole process, that the mechanism by which the insulin signaling may work in the brain is through this process. Okay, but how does the RAF4 enrichment at the synaptic compartment, presynaptic compartment, induces synaptophagy is something that we have to study in future. Okay, and this is the final data that I am going to show you. Here it was shown that if you lower the insulin signaling in the adult brain, then the adult remain more alert, and their learning ability remains intact with aging. So this is just one data in Drosophila, and we still don't know what happens in the humans. With this, I thank you very much for your patience, and I have to acknowledge all these wonderful people who did the work for this. Thank you.
Anterograde insulin signaling in the synaptic
anterograde transport induced by the insulin signaling.
Right. So the insulin signaling will also promote retrograde transport, because you need to have a communication between the cell soma and the axon terminal. So what is the
so we only studied the anterograde transport of the RAF4 vesicles, right? And that is stimulated by the insulin signaling. Now you are absolutely correct. Insulin signaling should also promote the retrograde signaling, but it should not promote the retrograde signaling of the RAF4 vesicles. So what we see is that the retrograde movement of the RAF4 vesicles are reduced, but we don't know what other is activated. Most likely it is known that dynein mediated transport of autophagosomes are induced during the synaptophagy. So quite likely that this same insulin signaling through VPS34 could promote the retrograde transport of the autophagosomes. But unfortunately, we could not find any good marker. The lysotracker doesn't work in the axon.
So the autophagosomal vesicles are not acidic yet, so they are not marked by the lyso tracker. And we tried few other autophagy markers. They did not work. Of course, we have to try much better to find it out, but that's a good question which I don't have an answer.
You said last interesting data when the insulin signaling was shut off. I mean,
right.
Negatively regulated. Then the learning ability was
learning
restored.
ability is retained for a longer period.
So
normally the flies age
Right.
within 15 days the learning ability start declining, but in this case they found up to 30 days the learning ability remains intact.
So that is a little bit of conundrum opposite what you think, because a lot of role insulin signaling has
No,
to
that
promote.
gels nicely with these, right? So what I showed you is that
Yes.
insulin signaling reduces the number of synapses, and what they showed is that suppressing insulin signaling can promote the synaptic activity, maintain the synaptic activity.
And is that phenomena is transcriptional output dependent, FOXO
So
or DAP16?
nothing is known. Nothing is really known. So what you're talking about, yes, insulin signaling has many facets.
Right.
So what I showed you is the immediate effect of the insulin signaling. Sure. Now there will be a long-term effect which will be triggered by the AKT and MAP kinase pathway. And those long-term signaling will do two things. One, in case of the neural stem cells, it will promote cell proliferations, and in case of the existing neuron, it will promote the the more homeostasis. It will promote more lipid biogenesis and all that. So neural growth and so on. So those are the slower processes which will be triggered by the insulin signaling. Now, another question that we still haven't answered, and I have a gut feeling that it might be an interesting issue. The question is, where do you signal with insulin? If you signal with insulin receptors, we found localizes both in all three places, in the cell body, in the axon, and in the synapse. So insulin receptors are present in all these three places. Now, insulin receptor stimulation at the presynaptic compartment is known to recycle the AMPA receptor, and it has been implicated in the learning process. Okay, now, insulin signaling in the axon is probably going to stimulate the RAF4 vesicle transport, and insulin signaling in the soma is probably stimulate the homeostatic process. Now, how do you sort of control the insulin receptor in these different compartments of the cell is another big question, and which nobody has really thought through. Yeah.
When the insulin signaling is impacted, is it really impacting in the brain also, in the particular in terms of diabetes patients?
Yes. So hyperinsulinemia, in case of hyperinsulinemia, which is type 1 diabetes, where the genetic disorders causes that process, these patients are prone to early dementia.
Oh, okay.
It induces early dementia.
Okay, I think if there are no more questions, we'll move on to our next speaker, Anindya.
Thank you.
And I know we're running a little behind, but we'll catch up during the tea break. But please be back here at 4:30 so that we don't get pushed back.
First slide. Yeah. Thank you very much. I would like to thank again Saurabh, Shefali, and Krishanu for encouraging us to do these events every day. We started last year, and thanks to Krishanu also to inspire us to think about the society. And really, we are for last few years we are thinking and made efforts to go out of our comfort zone and talk to clinicians, and we have a couple of projects going on. It's nice to see this progress. And also I would like to thank again Saurabh and his team and other my team members for actively organizing this event, and our poster of this events is organized by one of the students, Dikshali. Thanks for the encouragement. And today, so I'm just, since I think we are running out of time, I'll try to give a very simplified view of our lab's work. And since I think two of the students will be talking, I will just try to keep things simple. So as the theme of the conference is society, just I want to start by saying that, you know, there is, we understand there is a societal need to understand neuroscience and put neuroscience in the perspective of solving the societal problem. You know, theme of this conference is, you know, early developmental disorder, autism, and all these things. And we are talking about it. And, you know, at the midst, you know, late adolescent phase, there is a huge increase, surge of, you know, young, you know, adolescents living with anxiety, mental health disorder, and all these things, which also begs our attention of neuroscientists to, you know, look into this problem in the context of neuroscience. And of course, we all know that, you know, at the old age, we are having all these issues of neurodegeneration, dementia, locomotion deficits, and all this problem is complicated by the fact that, you know, that nervous system has a poor regeneration capacity, because the nerve cells cannot divide in the adulthood and replace the lost neuron, which is going to be very relevant for this session, because Saurav said this is a session for nerve regeneration. Here looking at this person called Deepa Malik, who had a multiple surgery at the spinal cord, and she was paralyzed, and therefore all the seamless communication we are talking about, Krishan was talking about, is there a pointer? Talking about the axon and axonal transport, all this communication process is blocked, therefore the neurotransmitter release does not happen, and we left with paralyzed life, right? And this problem has been very challenging when I started thinking about working on this problem, because the nerve cells in the central nervous system does not regenerate at all, which will be also covered by my co-speaker in this session, Ish Venkatesh. And there are multiple challenges in the nervous system regeneration. Even we talk about the peripheral nervous system regeneration, carpal tunnel syndrome, and all this brachial plexus. In the late stage, this, you know, axon, all the axon can regrow, but their pathfinding and the regrowth accuracy is lost. And of course, in the central nervous system, you look at the axons after injury, cannot initiate the regeneration program. It seems the problem is very hard, because somehow the central nervous system neurons are rather evolved not to regenerate. That's why we have a long lifespan, but we cannot regenerate our CNS. And it was a hard problem. People are working on it, and people are trying to find out, way to promote regeneration. One of the example I will give is that when protein synthesis was upregulated in these neurons, one could promote this regeneration of CNS broken axons despite their inhibitory environment. Kind of, this was inspiring to see that, you know, one could understand the intracellular processes within the neuron, so that one can help promote regeneration. So when I was thinking about this problem, also not just promoting the regeneration, one can promote regeneration. This is another example where RGC neuron, after receive crush injury, although you can promote their regeneration by playing molecular trick, then these axons cannot go in the contralateral side. So that leads to the, you know, imperfect regeneration, lack of functional restoration. So it's, when I was started thinking about this problem, I thought about, you know, why not use organisms which with a low, you know, shorter lifespan. They have to, organisms like worm or fly, where they have shorter lifespan and they cannot replace, really, they don't divide their neurons, there is no stem cells, and, you know, 3 weeks of their lifespan, they are navigating through the tough environment, twisting their, you know, body muscles. Perhaps they know it better how to do it, and we know that many of the lower organisms have better regeneration of the injured nerves. So, and it was also inspired by the fact that the system was very nicely developed by Sidney Brenner and his colleague to understand how brain circuit, how the connection between the neurons leads to the, you know, behavior. And his lab actually figured out how the neurons are connected at the level of electron micrograph and know the connectomics. One could understand kind of how these connections are utilized to give rise to locomotions or other kind of behaviors, olfactory behaviors. Here we are looking at work by Martin Chalfie, where he works using a neuron called touch neuron, where you can touch the worm with an eyelash and it kind of shows a response. And this behavior is given by this circuit which controls this behavior, and the mechanoreceptors are identified, and which are the neurotransmitter, how they develop, how they maintain this circuit, all understood by work from his lab. And so I thought that why not use this system to understand regeneration process, then we could start thinking about the genes controlling the circuit repair. So here is the first experiment, the same neurons I was talking about used by Martin Chalfie's group, responsible for giving rise to this gentle touch sensation behavior. So these neurons are growing on the periphery. It's like the peripheral nerves. So one could use 2-photon laser, and because the worm is transparent, many of you know that it is easy to use this kind of optical techniques, tools to kind of break these neurons. You could use the 2-photon laser and make a small saver. What you see is that these axons again starts regrowing. Although that movie is not playing. So anyway, this movie is supposed to show that these axons can regrow and kind of regrows towards the original synaptic area, and one could use this technique kind of very systematically figure out that how axons regrow. And the first discovery we and others made was a signaling cascade involving dual leucine zipper kinase, which is very critical for initiating this process. If you remove one of any of this component in this pathway, what you see is that all the nervous system develops perfectly, but they cannot initiate the regeneration program. There is absolute stop. So this MAP kinase we discovered later on found to be also very critical for the nerve cell axon regeneration in fly and mouse system. So that's what one could see, that what we understand using worm could be also important in the other system. So once we started our research at NVRC, we asked a question that whether we have a gene that initiate the regeneration program, whether this is important for functional recovery. What are the limits? What are the other limiting factor that kind of prevents the functional recovery? How the functional recovery happens? So the way we do this experiment is kind of using we have the microscopy. You can look at these neurons, not only just look at the neurons, you can also look at the synaptic proteins. And as you are seeing that, with laser, one could break these neurons, and you have these behavioral assays at various time points, and you can ask what happens after the axons are broken. And we can also image at various time points to look at that how axons are regenerating. And because these two neurons, which are very important for the sensory functions of the whole organism level, so you could basically relate the axon regeneration of single axon regeneration event to the whole organism behavior. So we could basically ask how the axon regeneration events are related to the synaptic function and the behavioral recovery. And recently we started also looking at the synaptic the partner neurons, whether the communications will be restored with the physiological techniques. Although you cannot do electrophysiology, what we do is we use this optogenetic techniques where you express channel rhodopsin in the presynaptic neuron, and postsynaptic neurons has the GCAMP, which reports the calcium activity, and we stimulate this presynaptic neuron and record the postsynaptic activity using confocal microscopy, and look at how the activity will be affected. Here is one movie, I think it shows that. So there is a electrical synapse and the chemical synapse. What you are looking at, that through the electrical synapse, the postsynaptic neurons are activated. That's what GCAM trace is telling. And with the chemical synapse, which is the inhibitory neuron, there is an inhibition post-stimulation. And after the injury, that activity is lost, telling that the activity between the pre and post synaptic is lost during the after the axotomy events. So thereby allows us to look at the correlate the regeneration with the physiological techniques. So when you look at the behavior, you see that at 24 hours time points, the behavior is very kind of not significantly changed, telling that there is no recovery. But at 48 hour, you see that there is a huge variation. Some of the events are kind of giving rise to functional restorations. So it allows us to ask that whether we can correlate the regeneration events with the behavioral recovery. And when you look at the various events, you see that many of the events are kind of following different kind of trajectory. It's not like the original axon. So some of them are going straight, some of them are making multiple branch. A large fraction is going to the ventral nerve cord where the original synapse was there, and also enriching the synaptic protein again. And also recently, actually, this experiment shows that when the ventral when there is the ventral targeting events are the one which is the gives rise to functional restoration, and this experiment actually connects it to the physiological level. When the ventral targeting happens, the inhibitory circuit is completed, and if there is not growing with the ventral targeting, when they are going randomly, when there is no actually activity restored. So relating the regeneration events with the in the imaging experiments. So also we have seen that although the worm can restore their behavior at the early stage, even it's a peripheral nerve of worms, as worm ages, the restoration is declined. So telling that even simple so telling that the behavioral recovery is also age dependent, and we have looked at in details axon regeneration parameters are also declined as the worm is aging. So the telling that then that's sort of giving us a parallel that in human also we see the regeneration is age dependent, in worm also it's age dependent. So what we did is use this paradigm to ask that whether we can switch this regeneration clock to promote regeneration, which is I think the main idea of the field. In the adulthood, one could promote regeneration. So I'll give you a few examples for the interest of the time. So one is we found the LET7 microRNA, which is very critical for the developmental timing. When the LET7 is not there, the worm remains at a young worm, like kind of a larvae, late stage larvae. That they don't become adulthood, because LET7 is very critical for this transition. So this, if you do this experiments in the LET7 mutant background, you see it promote regeneration despite their old age. They will kind of regenerate not only anatomically, also behaviorally. So the LET7 microRNA is one of the critical factor we found out. And Krishan was talking about insulin signaling. The insulin signaling is very critical for controlling the lifespan. In lack of insulin receptors extend the lifespan, and we found that, and that lifespan extension is dependent on the activity of the insulin signaling within the gut. But what we found that insulin signaling is very critical for within the neuron for controlling this navigation process. In the absence of the transcription factor DAP6 in FOXO, the rewiring is not perfect, therefore also the functional restoration does not happen. So this transcription factor kind of controls the expression of the guidance factor within the neuron and outside the neuron muscles, so therefore controlling the regeneration process. So for the interest of the time, I'm sort of skipping many of the data. So after we found about the LET7 discovery in the worm, what people found is that same kind of manipulation of LET7 microRNA one can do in the mouse retinal ganglion cells, which has a central axon. One could promote regeneration anatomically as well as at the functional level. Telling that LET7 microRNA discovery we did is also kind of can be applicable to the other system. So to summarize this work, we have sort of seen that regeneration, we can study regeneration after axotomy at the not only anatomical level, also the functional level, and the regeneration is kind of declines with age, and we saw that various factors one could manipulate, let's say when microRNA, insulin signaling, and also there will be a talk by Shivram who will show that physical exercise, the swimming exercise also promotes regeneration at the old age. So just to switch gear, how much time I have? 4 minutes? I'll finish in 4 minutes. So when we talk about regeneration, we are only talking about axon regeneration. But one has to think that the axons, these circuits are developed because of the connection. Axons and dendrites go hands in hand. We are not worried about axon, but recent work from various people shows that when the axon injury happens, the signaling within the surrounding environment kind of from the glial cells sort of kind of chops up this surrounding dendrites and the axon both, sort of creating an environment that this circuit is injured. Sort of this begs our attention to look at the dendrite regeneration. Also, when you talk about regeneration using stem cells, those stem cells can form neurons in the adult environments. Those has to again rewire. That is there is a challenge. One has to think that dendrite regeneration will be also need to be recalled there, and one is to understand. We thought with this inspiration we started working on dendrite regeneration. There is another neuron called PVD neuron which has a very elaborate dendrites. This throughout their body, these dendrites are extended with a very stereotypic hierarchy of primary, secondary, tertiary, and quaternary branches. These branches are interacting with the muscle and the epidermis so that it senses the force in the environment, which acts as a proprioceptive neurons. And yes, this connection between the muscle and the neurons are mediated by protein complex. We know from the work from Kangshan's lab and few others lab. And what we found was, along with this paper from Kangshan's lab, that there are two functions of these neurons. So the dendrites are very critical for the proprioceptions, and the axon is very critical for this neurons also senses large force. When the student poke this worm with the platinum wire, the worm hits, it run away. That the force is sensed by this PVD neuron, and also the trajectory of the worm, which we call proprioception, this sinusoidal movement and the curvature is maintained by these dendrites. So it's kind of a polymodal neurons, and if you cut the dendrites, only proprioception is affected. If you cut the synaptic connection, only the harsh touch. So it serves as a multifunction neuron, and with utilizing its differential neurites. So we thought that what will happen if the dendrites are injured? When we make a small injury using laser, what it does is that these dendrites are kind of quickly regrow and try to catch up and fuse with the distal end. But when we make a big injury with multiple laser shot, so what we see, the dendrites are again started branching in adulthood. So we're kind of trying to rewire again. And we asked a basic question. I remind you that we found that axon regeneration is dependent on this DLK MAP kinase cascade. We asked a simple question whether the dendrite regeneration would also require DLK MAP kinase cascade. So what we found is, in the absence of DLK MAP kinase cascade, the dendrite regeneration can happen, both at the functional level, at the behavioral level. So the dendrite regeneration does not require DLK MAP kinase cascade. In the same neuron, when you do axotomy, that axon regeneration is dependent on DLK MAP kinase. So telling that the same neuron has DLK MAP kinase for the axon regeneration, the dendrite regeneration would perhaps require some other mechanism. So that is the question. So I want to talk the end with. So we have a system where we look at the axon regeneration and dendrite regeneration both, and axon regeneration depends on DLK MAP kinase cascade, and the dendrite regeneration perhaps requires some other mechanism. So a talk from Pallavi will kind of tell us a very interesting story of the discovery of a different mechanism which is important for dendrite regeneration. I just want to thank all the people in the lab, including the people whose work I presented, and thank you very much. Yes.
signaling is regulating the axonal transport, anterograde transport, and in your case, it's regeneration. It also involves significant amount of transport. So one is happening in developing neuron, other your stuff is happening in the adult neuron. So what is in this unique in your case in the insulin signaling that it specifically promotes regeneration?
So one common theme I can say that because the requirement of axonal transport, the axon regeneration very requires active axonal transport. The after axonal injury, not from our lab, some work from Zhigang Heng's lab and Mark Hammerland show that mitochondria needs to be actively transported near the growth cone. That is a kinesin dependent. So you can relate the right. So the growth cone related stuff will be signaling component, and there is a common theme. And the other thing he said that if you shut down this long term thing like maybe the transcriptional thing, somehow the nervous system circuit rewires in a way some of the benefits you get. Learning ability, when he talked about that, it restores the age dependent learning ability. You can think it's kind of restores the functional nerve regeneration better. That way two things are common here.
When it forms the synapse, the neurite is stable. So that is the end of the growth. So when you have to grow the synapse, grow the neurite, you have to recover the synapse, and then the neurite can grow. So these two processes are not opposing each other. These are actually the same system. So if you promote RAP4 movement, to promote membrane recycling, to promote the neurite growth, and that neurite growth would require you to recover the synaptic contacts, because otherwise the neurite cannot grow. So recovering the synaptic contact and neurite growth are part of the same process.
So we'll have just 15 minutes break instead of 30 minutes.
Any
to
question you
time.
have? Yes. We'll discuss here.
And also our next speaker, Ish Venkatesh, needs to go to the airport. So we really have to start on time. We'll be back at 4:30, really at 4:30.
Yes.
We can continue? Okay, so then
we'll continue this session, this talk, yeah.
Okay, just one second, I have to turn off the yellow. Perfect. Thank you. Thank you everyone for sticking around and helping me with my logistics. I think it makes complete sense to go right after Anindya. Okay. Since I work on a very similar topic as well, I also thank Anindya, Saurav, and NBRC for having me over. So I work on spinal cord injury and regeneration in the spinal cord system. Superman is here for a reason. I don't know if you guys know Christopher Reeve, if you're old enough to remember that he played Superman. I like starting with his example because he was an avid horseback rider and he's taken his horse for multiple tournaments. And for almost 16 years, he's worked with this same horse. But I didn't put the accident, but right after this, what happens is his horse just sort of collapses in 3 seconds. He had an upper cervical injury. And just like that, the epitome of physical strength was reduced to someone who had to be kept alive on a wheelchair. And he remained on the wheelchair till his death, but he became a advocate for spinal cord injury research. And I think it just speaks to how instantaneously someone can suffer a spinal cord injury. And right now, there's absolutely no treatments except rehabilitation and physical therapy. And this is a problem that was noticed by Kahal back very back in the time. So this is a transection of a cat's spinal cord. And it sort of shows you how the accident site looks like. You have the two ends and you have these axons that are attempting to sort of regrow, but they form these dystrophic growth bulbs that are just, not able to cross the injury site. And he left us with a very harsh degree, saying that all these observations allowed me to, lead me to the conclusion that the central tracts are incapable of regeneration. And this is exactly what we're trying to challenge in the lab and figure out how we can actually make them capable of regeneration. And this is a global burden. We have cases that are piling up every year. And I think there is hope because majority of the neurons in nature actually are fully capable of regeneration and repair. So there's an evolutionary and developmental divide. And you're seeing here from lapre all the way to mammals in young ages, in up to a week after birth in mice, if there's a spinal cord injury, they fully recover behaviorally, functionally. They mount a very successful response. But the same thing when you look at a little bit older, like Anindya mentioned, there's a divide now, right? So up until reptiles, they're capable of repair, but after that, there's a dramatic shutdown. And this happens in a very specific developmental window, like I mentioned. P8 is when this happens. And after that, no matter what you do, these axons just don't regenerate. And it also, like I said, its lack of regenerative ability comes up late in evolution, it comes up late in development, and even then it's only restricted to the CNS neurons because the peripheral nervous system, like he mentioned, is fully capable of repair throughout adulthood, right? So I'm here to throw a little bit of light into why this happens. So we work with layer 5 of the motor cortex, corticospinal tract. In humans, this controls the fine motor ability. So to appeal to the younger generation, when you're texting, your corticospinal tract is firing. So it's important for all kinds of fine motor control. What it does is it crosses over at the medulla and innervates opposite sides of the cord. And during P0 to P8, what's happening is the axons are linearly growing. They're actually laying the information highway, your spinal cord. And at this age, the process is driven by a group of genes that I'll refer to as pro-growth genes throughout my talk. They have complementary functions, everything from remodeling to transport, guidance, macromolecule synthesis. And so we're talking about 800 odd genes that come together at very specific times to pull off this axon elongation during development. Now, sitting on top of this cascade, like the engineers that sort of control the process, are transcription factors that we call pro-growth transcription factors. They're fewer in number, and they sort of, decide who gets made, how much they get made, when they get made, so on and so forth. And the reason young neurons are so good at repair is because these transcription factors are on. As a result, the genes are on, and as a result, the repair ability is on. But at P8, now it's shifting its phenotype to collateral sprouting. So it's now starting to form synapses and connections. And like Krishanu just mentioned, those two sort of cancel out each other. So when a neuron has formed a connection, it stops growing, right? So at that stage, what happens is these transcription factors are turned off. As a result, all of the downstream processes are shut off, and it sort of goes into this dormant growth state. And if you look at the ability to respond to injury, those species that reactivate these pro-growth transcription factors, they successfully recover from injury. And those that fail to do so permanently become incapable of repair. So what I've been trying to do in my career is see if we can reboot the growth capacity by turning back the clock, to back to development by manipulating these developmental transcription factors and supplying them back to an injured adult neuron and see if we can promote growth. There's good news and bad news. The good news is the process works relatively well. So just to orient you, you'll be seeing a lot of mouse spinal cords. You're looking at a sagittal section of the cord, and the red arrow marks the site of injury. In green is the axons coming down from the brain. And you'll see that our system is extremely refractory to growth, which means they just don't grow past the injury site. Not even a single axon makes it past without any sort of gene treatment. But with a treatment like KLF6, you do see some long distance axon growth that's past the injury site. And to orient you to a slightly different model, this one's a regeneration model, this is a plasticity model. This injury is called a pyramidotomy, where we go in and just injure one half of the pyramid at the medullary level. What that does is the uninjured axons from that site start sprouting to make up compensatory sprouting into the injured site. And this naturally happens. And we supply our gene treatments and see if we can push the ability of this natural sprouting. And we do notice that when we give a cocktail of KLF6 along with its partner, developmental partner NR5A2, we do restore quite a bit of the, to developmental levels. And while this is good news, I want you to sort of pay attention to the numbers. We get about 3 millimeters of growth with the very best gene treatment in 3 months. That's how much developmental axons grow in a day, right? So if you have a human being with a lumbar 5 injury, if you do the math, which I've done, it'll take them 25 years to go from any sort of regeneration in the current system. So while I think it's there's hope, I think we also have to find ways to boost this phenotype, which is what I've been working on. And one of the things I've heavily paid attention to is considering the idea that transcription factors don't work in isolation, and there's likely a network of factors that work together. So a mouse peripheral neuron, which successfully regenerates, recruits groups of factors, and it's doing both turning up of some factors, but also turning down of some factors, and it does this simultaneously to achieve regeneration. The same is true for frogs, flies, fish, worms. All of them recruit groups of these factors. But when you look at the central nervous system, we know of only 3 factors that promote any sort of CST regeneration. So it's pretty much still a black box as to what are the full set of factors that are involved in promoting developmental growth. So that is what I set out to do in my lab. One of the main directions of our lab is to figure out, decode, somehow deconstruct who is driving axon growth during development, and then ask the question that if you supply this back to an injured neuron, if that is sufficient to induce regenerative axon growth. In a very simple framework, that's what we are trying to do. And so when I started my lab in CCMB is 2.5 years old, so we are relatively still new. So I asked my postdoc Manoj and my first PhD student to tackle a question that we've been trying to answer for quite a while, which is we've always known that between P0 and P8, somewhere the regenerative capacity is shut down. But what we've never known is exactly when this happens. Is it a very slow process? Does it happen P7 night? Does it slowly happen from P4? And that dynamics was not mapped out. And we had started generating some single cell data sets that allowed us to directly answer this question. So what we did was we lined up from E14 all the way to P28, closely spaced intervals of neurons across development. And we simply just asked in a way of RNA velocity mapping, trying to map the loss of regeneration by looking at certain regeneration markers and how they progress. And then importantly, what you can do is you can identify these violet points that are anchors in the process where there is a significant cell-fate transition that's happening, and ask who is driving that cell-fate transition. And when we did that, we noticed that we pulled up a lot of transcription factors that were actually not on our radar at all. These were nuclear receptor family. And although they are very well studied in pretty much all of biology, and they're quite well studied in the context of axon growth during development, but for whatever reason, it was not, as a family, was not on our radar. So Yogesh did the basic question. He wanted to see, we have an atlas in the lab, an RNA-seq expression atlas, where we go from early stages before axon growth, during axon growth, and then an adult RNA-seq data set, so that we can sort of see differentially how these genes are expressed across age. Is there any pattern that suggests that they might be involved in coordinating regeneration? And we noticed that they sort of clustered or fell into 3 buckets. There were some that are coming on very early, like the NR2 F1 and F6, which comes up even before growth is starting or attempting or peaking. And then there are some that come up right around the time when axon growth happens. And of course, there's a bunch of factors that seem to come up after axon growth is established. And so when we started looking into literature by pulling up mouse PNS data sets and zebrafish data sets, what we found was evidence to suggest that these are all reactivated by regeneration competent species in response to injury. And these are what is usually turned down in data sets. So we started sort of working with the hypothesis that we've probably narrowed down on a set of genes that are acting as primers of growth, some that are drivers, and some within the same family that seem to be putting the brake in older neurons on halting growth. So this is the hypothesis that Yogesh is working with. I'm primarily presenting his work, but it's a huge team effort, as you'll see. So our hypothesis is that if you deliver combinations of these factors, and I'll tell you how we narrowed down to the exact combinations, if you reintroduce these factors, that should restore regenerative capacity, and if you CRISPR knock down these factors, you should see some more regenerative capacity, and the combination of the two will probably give you the best phenotype. That is our working hypothesis. For lack of time, I'm not going to go into all of the techniques, but just some of it. We have a nice in-house AAV production platform where we can make these viruses. One of the bottlenecks for us is, since we're talking in tens of genes, we can't order and buy these commercially. So Yogesh has established a very nice platform to make the assays. I'll be showing you some in vitro growth assays, both neuroblastoma cell lines that are differentiated to neurons and also primary neurons. And we have two different injury models, like I showed you, pyramidotomy, and this is thoracic crush, where you can see that axons that are labeled sharply stop at the injury site. And this is how we check with our gene treatment, can we manage to push them past. We also do single cell genomics to start teasing apart the molecular mechanisms. And we have some detailed behavioral assays that I'll show you as we get to. So once we had this cocktail of factors, the first question we asked was, let's find out which factor is binding or regulating most pro-growth genes during development, right? So if you were starting with something, you want to hit that at the network in a maximal way. So a factor that was predicted to bind or regulate almost 60% of our 800 odd genes was a factor called NR2F6, which is a close binding partner of NR2F1, which is more well studied in development. And so we just put a simple experiment, AAV experiment, where we apply AAV NR2F6 to neurons. And then we ask if there is significant growth. And actually, we saw that consistently we were able to see a lot of growth. Now, when we started off, NR2F1 was our bet. And we've done experiments. I'm not showing you the data, but we've overexpressed NR2F1, we've overexpressed F6, we've overexpressed combinations. And all three times, this one factor alone is able to do the job of multiple factors. So, and if you Google NR2F6 and axon growth, nothing comes up. It's sort of a relatively unstudied factor. And then several months later, after we managed to get ethical clearance for our experiments, we pulled off our spinal cord injury experiments. So it's a straightforward experimental design. We inject layer 5 of the motor cortex. We load it up with our virus, AAV, and it has a GFP label so that you can also see the axons. And then we expose the thoracic region of the cord, and between T8 and T11, we do a crush injury with our forceps, which immediately leads to instant hind limb paralysis. And then 12 weeks later, we do GFAP staining to make sure that the astrocytes are there at the injury site and it's a strong enough injury. And then we see if our gene treatments make sense. So this is just to show you the controls. Like I showed you before, if the white arrow points at the site of injury, GFAP signal is in blue, and they just stop right there. And we were sort of taken aback to see all of the regeneration that we were seeing in the NR2F6 animal. And it's we are still in the process of replicating this, counting this and everything. But so far, 8 out of the 8 animals, we saw complete proper regeneration restored, which itself was very happy news for us. The other important aspect is promoting regeneration is one aspect, but translating that into any sort of behavioral recovery has always remained a challenge for the field. And for reasons I think Anindya has rightly outlined, these guys are coming back into a environment which is filled with immune cells that are trying to attack them from regrowing. The local circuitry is completely misfired, right? So we were, so what we're attempting, so until now in the field, usually what happens is does a mouse fall or not, does it walk or not? We've sort of had these binary readouts, and most factors don't promote any sort of walking. So what I started doing in the lab along with Manoj, who's a brilliant programmer, is to write our own pipelines to start tracking these mice as they're walking, especially track them on some critical body anatomical points, so that even if you're not seeing a mouse walk, we can at least measure the extent of gait improvement that we're seeing, at least start to put together a framework. So we allow these mice to just, this is right after injury, 2-3 days. So you can see it's just sort of dragging its hind limbs, and that is usually the phenotype we see most. But with F6 animals, what we noticed was significant. For the first time, so with the control animal, you can just see it just doesn't have the strength to sort of stand up or lift itself up. But with the treatment of NR2F6, these animals are able to raise their hips, which is sort of the first step towards walking. They're at least able to hold themselves. So just again, the control animal remains like this. It uses its forelimbs to sort of drag itself forward, rarely engaging its hind limb. But with the treatment, we saw that these animals are at least attempting to stand up, and we've quantified the hip raise. Both NR2F1 and F6 seem to do really well with this. And like I mentioned, we're interested in seeing what extent of behavior we're restoring and how we can improve on this. So, for instance, if you were to, this is showing you compressed frames of like lots and lots of videos of these animals walking. And you can see that an uninjured animal, there's a nice sort of movement for the toe to go from attempting to walk, coming into this, and then walking. And this continuously happens. And if it's an injured animal with no gene treatment, the angle always remains like that. It never attempts to walk. And with our F6 treatment, we are noticing that for the first time, even if it is not going the full swing, it's at least attempting to come until there. So we feel like the neurons are trying to mount a behavioral response. And I'll tell you why this is exciting. Like I said, in the history, we only know few transcription factors that promote regeneration in this cell type, which is CST, which is very refractory. Almost none of them have shown any improvement in behavior. So for the first time, we seem to have a factor that is showing regeneration and some sort of behavioral improvement that we can actually build on. And in those lines, along those lines, I have a team of people, my third PhD student, Rutuja, two postdocs, and Pratikya. They're developing ways that we can combine this gene treatment with stem cell implantation, again, what Anindya was talking about, to sort of get rid of the inhibitory environment at the site of injury and replace it with a more permissive substrate, something that's going to welcome these axons as they're regenerating and allow them to functionally integrate into the circuit. And so we have iPSCs that are getting differentiated into different motor neurons. And this is all very preliminary, but I was just excited to show you. So you can actually see that if you inject iPSCs in the cord, there are some cavities that don't get filled, but they're starting to sort of crowd in on the injury site and fill it up. So we are following this for a long-term study to see what happens. And the goal is to actually differentiate them into very specific type of motor neurons before implantation, so that we can get the kind of sculpting of the circuitry that we want. We're also combining that with optogenetics, also what Anindya introduced to you. And we're also excited about these peptide hydrogels that have recently come up in the field. One of the biggest problems is the extracellular matrix is so severely damaged post-injury. So people have found that self-assembling peptide hydrogels that are embedded with some of these molecules can mimic an environment that is very pro-regenerative. So we're attempting this as well. So we're doing everything we can to make sure that the axons that are coming through are able to sort of functionally translate into behavior. Meanwhile, fundamentally, I'm a molecular biologist, so I love digging into mechanisms of action. Like I said, with NR2F6, we didn't know more than we knew, right? So we didn't know whether it's acting as a transcriptional activator, whether it's acting as a repressor, who are the downstream gene targets, and does it target gene promoters? Is it targeting enhancers? Is it upregulating developmental genes, or is it upregulating some novel regeneration associated genes? So those were some of the fundamental questions that we want to ask. So we do single cell RNA-seq in the lab. Actually, the schematic is missing. So essentially what we do is, the beauty of single cell genomics is in the same animal, you can have NR2F6 plus F1, and also neurons, and by random permutation combination, some neurons are going to take up just the control, some of them are going to take up F1, some of them will take up F6, and we can sort of parse it out like that bioinformatically and figure it out. And for the first time, we noticed that most of the transcription factors we've identified are activators. But in this case, we noticed that 3,500 genes were repressed, and only around 400 genes were upregulated. So there was 8-fold more downregulation than there was upregulation, hinting that it's likely a repressor. And we looked at the kinds of genes that were getting upregulated, and with some network analysis, we did notice some of the usual candidates, which is a sanity check to make sure we're on the right track. So nervous system development genes, a couple of them were going up. Some of the known pro-growth transcription factors like SOX11 were upregulated. But interestingly, the main suite of genes that showed up were a lot of metabolism genes. So it seems like it's gearing up for some energy intensive process by turning up metabolism. We also noticed that when you look at the downregulated genes, there's 4 to 8-fold downregulation of immune response genes, sort of, probably stopping the immune response from going forward. And interestingly, this is also something we didn't expect. A big chunk of the genes, around 2,000 odd genes, were genes that are slowing down translation in all ways. So it seems like the neuron is shifting its metabolic reprogramming to go from only translating what it needs relevant to regeneration. At least that's the hypothesis we're going for, and turning off everything else. And this is actually not new to our system of regeneration. When I was reading up on this, many neurons, when they go into regenerative phenotype, try to dampen their translation to sort of go into an energy saving process, which is what I think we are also noticing. And Yogesh is now doing RiboSeq, which will tell us the global translatome and tell us whether this is matching up, what the transcriptional consequences are matching up at the level of the proteins. And meanwhile, a simple experiment for us to confirm that it's actually acting as a repressor before we run away with this. So what Yogesh and Meghna did is they pulled up the F1, F2, and F6, which are highly conserved in structure, and they identified the potential amino acids responsible for the repressor activity, the repression domain, and then they just chopped it out to make a domain deletion experiment. And what we noticed was, this is your control just to train your eye. This is your NR2F6. Usually we see these long neurites. when we did the domain deletion of these 9 to 10 amino acids, we noticed that the growth phenotype is now abolished. So essentially, it's back to control levels. So likely suggesting that this repressor domain is important for the activity of NR2F6. So we now have some new knowledge that NR2F6 is acting as a repressor. It's slowing down eukaryotic translation and upregulating metabolism genes to shift into that state. And we also, I'll be wrapping up, just 2 more slides. We also did an experiment cut and run, which if you've done ChIP-seq, it's like a version 2.0 of ChIP-seq. It gives you better resolution because there's less background. And Katha was interested in knowing where in the genome, we've always had this question, when you reintroduce these transcription factors, do they go back to their developmental targets or do they go somewhere new? So she sort of wanted to check that. And interestingly, we found out that around 15% promoters were only bound by NR2F6. Majority of the genomic regions that are soaking up NR2F6 are actually enhancer regions. More than 80% of the regions are enhancer regions. And it seems like we're hitting on a regeneration specific gene program because we didn't see a lot of the developmental targets being bound by NR2F6 in an injury context, which is something we've always predicted, but they've never tested it. And so genome browser screenshots are showing you that wherever NR2F6 is binding, either in the promoter or the enhancer, it's filled with these cis regulatory elements annotated by ENCODE, suggesting that there is some sort of long range gene regulation that is happening, because what you're seeing is islands of enhancers, put proximal and distal enhancers surrounded by wherever NR2F6 is binding. So Deepta in the lab started optimizing Hi-C experiments. Hi-C, what it allows you to do is it maps genome-wide what are the 3D contacts present across every gene. So we just got the data sets back on our before injury and after injury data sets, and we have our F6 data set we're analyzing. And the idea is to see whether when NR2F6 is bound to these enhancers, do we see 3D genome reorganization, which is something that peripheral neurons do routinely. So it will tell us whether there's any overlap in how they seem to reactivate the growth transcription program. And lastly, to finish off, I told you that some of them are repressors that we intend to knock down with CRISPR. So this is showing you Meghna and Chopin have been working in the lab to develop our CRISPR system. So when you overexpress NR1D1, which we predicted is a repressor because it's only present in adult neurons, so they almost become bald. We don't see any neurites at all. And if you CRISPR knockdown, then we start seeing this re-establishment of neurites. So I think just like we have a cocktail of activators, we also seem to have a cocktail of repressors that we are targeting in a separate project. Somewhat of the same framework, but instead of overactivating the activators, we would knock down the repressors and then see what that does to gene expression. With that, I just want to zoom out. I've told you today about a family that's simultaneously acting to promote regeneration in young ages or growth while acting as a brake in older neurons, and walked you through some of the approaches that we're taking to test this hypothesis. With that, I want to thank my terrific team. These guys do all the hard work that allows me to come and tell you all the fun stories we're doing, and also CCMB for its fantastic support system that allows even a young faculty to set up a good time. Thank you so much again to the speakers for inviting me, and I'm happy to take any questions.
So there are a couple of questions. One is that, is this protein is expressed in response to injury?
So it's a developmental factor that is turned off after development. In species that are capable of regenerating, like zebrafish retinal ganglion cells, even the peripheral nervous system neurons in mouse, they're re-expressed upon injury.
And like, for example, if you try from P0 to P8 at different developmental stages, at particular developmental stages, if you capture, okay, if NR26 is expressed, then there will be possibility for regeneration.
So you're asking me if
like, for example, one particular so what is very cool in your talk is that the vertebrate actually lost the ability to regenerate. And these particular proteins, like a nuclear receptor family of protein, holds the key that possibly, I'm not very sure.
I would love to keep going.
And then probably if we can express this protein, we can actually promote the regeneration.
Yes, that's the hypothesis.
Right, this is the hypothesis. Yes. So if the hypothesis is true, in that case, first thing first, it should be expressed in response to injury.
It's expressed in response to injury,
And
not
if,
in.
and if it is,
Uh-huh.
then at what developmental stages?
Okay, you're asking me about younger neurons. If they're injured, is NR2F6 expressed in that age?
Right. And
upon
then
injury?
CNS versus PNS.
PNS. So doing a spinal cord injury on young pups is something that's very tricky. We haven't yet done it here. There are actually, with the exception of a couple of papers, there's not even genomic data sets profiling young CNS neurons. All we know is the regeneration happens, but nobody's actually checked to see what goes up or down. So that's a great idea. We can check, establish that and check. I can only tell you that in non-mammalian species and in peripheral nervous system in adults, if there is an injury, many of these factors seem to go up. Yeah, that's the yes.
It's very nice talk. So NRF you are talking about.
Yeah.
So it's sort of, if you, the way you are doing is you are expressing more because of the
Yes.
overexpression, and then
Yes.
it's promoting the regeneration.
Yes.
And remind me, what was the story with the KL? There are a bunch of KLFs.
Right.
Some of the KLFs are inhibitors, some of the KLFs are promoters. Am I
Exactly.
correct?
Yes,
Have you looked at your NRF in the context of this? For example, have you tried co-expressing NRF with the other promoters? There are a bunch of other
Absolutely.
transcription factor which shown to promote the regeneration in this assay system, right?
Right, right. It's the ideal question. Actually, we now have injections of KLF6 plus NR2F6 going on. One thing that is interesting in that context is when you turn up NR2F6, one of the first genes to go up is KLF6. So we actually think it's upstream of KLF6 and could be triggering the expression of KLF6 itself. So we're trying both in just combining NR2F6 plus KLF6 and also doing KLF6 overexpression in an NR2F6 knockdown background to see that if you don't have NR2F6 around, would KLF6 still be able to do what it does during development? And so those combinatorial experiments are all being tried now. What is cool is after the KLF discovery, which was 2009, right? We've not seen any family that simultaneously acts as an activator or repressor. So I think like the KLF family, we might be onto something that is both simultaneously activating and repressing.
There are a few other transcription factors like SOC, correct me, like
Uh,
SOC.
SOX11.
Yeah.
SOX11 is also there, a different family. We haven't yet started trying with SOX11. There's a particular reason because SOX11, while it improves regeneration, it makes behavior much worse. In a follow-up paper, it was shown that the long distance regeneration we see, it doesn't translate to behavior. In fact, it makes it worse. For that reason, I've still not gone back to SOX11. We could always try that as well.
Thank you.
So I think in one of your earlier slides, I remember seeing retinoic acid transcription factor. So have you thought, number one, combining it with retinoic acid treatment or just expressing the transcription factors? And then coming back to the SOX, the second question, coming back to the SOX aspect. SOX has been known to be an initial promoter and then it can again shut down proneurology later on. So have you thought about a transient treatment?
So actually that's a good question. So coming to the retinoic acid part, RARB we've tried in combination, RARG we've tried in combination. So far we've not hit on a cocktail that is doing better than NR2F6 by itself, but RARB by itself promotes some regeneration. We have a project going on where we're trying to see if we can, because one of the problems with retinoic acid based factors, it's endogenously it is unstable quite a bit. So we're trying to do some small molecule treatment to see if we give a sustained supply of retinoic acid, how would that affect the activity of these factors? That is ongoing. Coming to SOX, so this idea of like keeping it on forever is something I'm also not a big fan of. So now what we're doing is we're trying to go back into a system where we're using flip excision vectors, because the growth promoting genes and the synaptic, plasticity related genes are both at odds with each other. If you continuously have these things on, you're never going to sort of get the kind of behavioral improvement that you want. So we're trying to turn off the growth program to see then what happens to behavior using these sort of transgenic mice, but it's all ongoing. It's just something.
Hello, great work, and it's very interesting. So I have one question that you are giving this NR2F6 before the injury is performed. So is it possible that if you are giving it after the injury, that also might lead to restoration? Also, it might give the temporal when this factor is coming into play.
Absolutely. See, it's a question that I think is the right question because clinically speaking, preloading a system versus giving it after injury. So we've always been thinking about this. So one of the things, the reason why we do it this way, I'll walk you through the logic. I think it's to give the best shot for these axons to regenerate because we've seen that this system is so refractory to growth that initial screening, if you start putting that model, then almost none of our factors are going to make it. But now that we've identified something that seems to work, the immediate question is, if we give it 1 day after injury, two days after injury, 1 week after injury, can we still restore? So one of our project associates is doing a chronic study on NR2F6 to sort of map out that dynamics and see what happens.
So as I've understood that this factor, it gets downplayed after the embryonic stages goes and in the normal context.
Yes.
So are there like other studies and other models in which, I mean, in the invertebrate systems or where it still is active or its orthologs are still active and they are playing a role in the regeneration?
As far as I've seen, most of these nuclear receptor factors, at least the ones that we've pulled on, seem to get turned off early on during development. The time of when it gets turned off changes, but pretty much they're all gone in some stage of development. We've not seen anywhere where it remains constant. They all just seem to be re-upregulated in response to injury. But one of my students had a question: if you keep it constitutively active and never shut it down, what would happen? So she's trying to put a VP64 activation domain in the locus to make sure that there is a strain where it's going to be constantly active and it doesn't shut down. And then she's trying to test that hypothesis. But as far as I know, it's one of those factors that goes down and then comes back up in species that are able.
There must be a temporal regulator of this NR2F6. Is it known in the
Yeah, it's a chicken and egg problem. You know, who's above NR2F6 itself? That also we're trying to look up and figure out. So far, I don't have a clear answer. It seems like we don't know much about who is regulating NR2F6 itself. Yeah, so hopefully next year, if I am around, I'll give you some insights.
Sure, looking forward to it. Thank you.
Thank you so much. Yes. So usually a consequence of growth is that the activity, all the transcription factors that seem to work, in some level they seem to turn down neurotransmission and activity because those two don't go hand in hand. The minute the neurons found a target, it stops linearly growing. So for that reason, I think the synapse genes are usually turned down in a successfully regenerating environment. Here we haven't started testing what happens if, with local activity increasing. One thing we're trying to do is put a electrode below the site of injury to see if those regenerating axons are forming functional synapses. And then with that system, we could ask, you know, to keep the activity stimulated, does it seem to pull more axons or does it hurt your case? That's something we could do. Thank you.
There was an interesting observation from angry requirements of the they are stimulating the postsynaptic
Okay.
attracting this
Growth cones
in the RGC system.
So this is what yes, I've seen that paper, and Supraja's paper is telling the opposite, that you know postsynaptic activity prevents regrowth in RGC also from Andrew. This is you're talking about Supraja. Okay.
Stimulate the postsynaptic neuron with optogenetic canal rhodopsin.
And that seems to promote
the regrowth and guidance at the level of function.
Yeah, I mean same paper we're talking about. Yeah, I mean this both evidence. So Frank Bradke's Brett Hilton, I think he's the one who showed the opposite. So there's two camps of thought that.
Supraja is the one who is working. He has a lab.
Lab now, yes. Oh, that's fantastic. I should get back to that idea and see if we can test that in the system.
Great. I think we had a awesome first session. And let's thank our speakers today. We'll take a tea break right now. So, minute break. So by 5 o'clock, we'll start already.
हेलो हेलो ठीक है ना। और कम और जोर बोलूं क्या? थोड़ा सा बोल दूंगा, अभी तो मैं ट्राई कर रहा हूँ। ओके। अभी जब वैसे भी थोड़ा सा हो सकता है। Yeah.
Okay, everyone, please settle down. We're going to get started.
Hello, am I audible? Yeah. So good afternoon, everyone. So now I'm thanks to organizer for giving me this opportunity to talk about my work. My work also focuses on understanding the mechanism of axon regeneration and how to improve it. So I basically work on the MPK signaling that controls axon regeneration. So before the session, like the previous session before the tea, people have elaborately talked about the problem about spinal cord injury and the problems of regeneration. So I'll not go on details, like you already appreciate that there is a like requirement to understand and improve the axon regeneration in the central nervous system. So along with all the efforts to understand or improve the regeneration by manipulating the genes or molecules, so one of the approach which has been found to be beneficial or promising is the rehabilitative therapy, which involves physical exercise. So as you can appreciate, in the previous speaker gave the example of Christopher Reeve. So after he fall down from the horse riding accident, so it has been found that after 5 years of swimming experiment, he was able to regain 40 to 50% of the functionality by the rehabilitation paradigm. So that suggests that the rehabilitation paradigm is beneficial in improving the functional restoration. Not only the Christopher Reeve, there are now documented evidence that shows that after going through a rehabilitation paradigm, it promotes the functional recovery. Also, some of the evidence shows that it improves the axon regeneration. But the understanding is still at very nascent stage, and what is the mechanism involves in like correlates from physical exercise to axon regeneration is still not known. So that is my interest, where I want to understand how this mechanism is elucidated. So for that, we are using the C. elegans gentle touch sensory neuron, which Anindya described elaborately. So to use the C. elegans neuron, we need a exercise paradigm. For that, we are using the swimming exercise paradigm. As you can see, when we put generally the worms grow in the solid medium. When we put in the liquid medium, they goes in a severe muscle activity through thrashing, which mimics the mammalian exercise features. That's like suggest high energy consumption, post exercise fatigue, and the muscle fats get utilized. So this suggests is a established swimming exercise paradigm, which we combined with the PLM action regeneration model, which Anindya talked about, and we are trying to elucidate what is the mechanistic understanding and how exercise affects the action regeneration in this context. So our paradigm starts like this. So we use the worms PLM, which are marked in GFP neuron. Then we give a 2 laser shots in around 50 micrometer from the cell body, and then we assess the behavioral assessment. Then we divide into 2 groups, one is control and one is swimming group. Then at 24-hour again we assess a behavioral assessment and see what kind of regeneration response is there, and then we calculate a recovery, which is the behavior at 3-hour and 24-hour, the ratio. So when we did that, so as you can see in the panel, so this is the 3 different life stages I am showing. So there is a significant behavioral improvement after regeneration while we do the swimming. So that suggests that a single 90-minute session of exercise improves the functional recovery and action regeneration significantly. So we found out that the metabolic sensor, which is the AMPK, that regulates or monitors the cellular energy status, that is important for mediating this effect. As you can see in this second graph, where AK2 loss of function, which essentially says that the AMPK is non-functional, the functional improvement which was seen in the wild type case is not there. So that suggests that AMPK is required for mediating this effect. So that we have published in the summary that AMPK and how exercise in this article. But the question remains, right, how AMPK is activated, and is there the direct activation of AMPK will improve? If AMPK is a direct mediator of exercise, if we activate AMPK directly, then it can mimic, or it should mimic, the exercise effect. So for that we use this paradigm. So where we have seen that when we, before putting the worms into the swimming well, if we just briefly use a levamisole treatment so that they don't swim in the swimming well, they just stay stationary, that does not give a functional improvement. So in this context we use the drug treatment which is called AICR, that directly activates the AMPK. So if we put AICR in the swimming well, so essentially the worms is not swimming, but the AMPK will be getting activated. So we tested this paradigm in the behavioral context. As you can see, out of 3 concentration we tested, 1 millimolar treatment gives enhanced significant functional improvement, and that is absent in the AK2 loss of function mutant, which suggests that the AICR treatment mediated improvement in functional recovery is through AMPK, and this holds true for in the adulthood also. And the anatomical regrowth length, as you can see, is also increased significantly. So that suggests that directly activating AMPK through a drug can improve the functional recovery significantly and can be a promising approach to follow up. So now the question comes, like, after swimming, how AMPK gets activated? So there are a couple of mechanisms by which AMPK gets activated. So AMPK gets phosphorylated at 309-172 position by predominantly 2 kinases. One is the liver kinase B1 and one other is the calcium kinase 2, which in turn phosphorylates AMPK, and that activates the AMPK for mediating its downstream effect. So when we tested this, we found that in the loss of function of PAR4, the improvement due to the swimming exercise is not observed, and that suggests that PAR4, which is the liver kinase B1, is important for mediating this effect through the AMPK. And as you can see, when the AMPK is constitutively active, there is an increased functional recovery, and that does not get hampered by the PAR4 mutant. So that suggests that PAR4 is acting in the upstream of the AMPK. And the PAR4 activity requires a scaffolding protein, which is called axin, which is in the worm, is called PRY1. When we knock down the PRY1 in the neuron, you can see that does not give the improved behavioral improvement after swimming exercise. So this, in summary, we tested the AXL1 and CKK1 also. We did not saw the effect. So this suggests that in the swimming exercise through PAR4 and PRY1, AMPK gets activated. Now the question comes, what is in the downstream of AMPK? Like AMPK regulates multiple transcription factor and enzymes to maintain the energetic balance. So we selected few candidates. One is the CREB, one is the histone deacetylase and FOXO transcription factor, and one is the PGC1 alpha, which is another transcription factor, which are relevant for exercise and axon regeneration. So out of these, as you can see in this graph, when the DAP16, which is the FOXO's ortholog, and MDT15 is the functional ortholog of PGC1 alpha is absent, we did not saw a functional improvement compared to wild type. So this suggests these two are involved in the downstream process. And when the AICR treatment was given, the DAP16 mutant and MDT15 mutant does not show a AICR treatment mediated improvement also. That suggests they are acting in the downstream of AMPK. So to summarize the downstream factors, the finding is that the swimming exercise activates the AMPK by PAR4 and PRY1. And in the downstream, these two transcriptional regulatories are somehow mediating the effect to the axon regeneration. So now the question came, like previously, by the rescue experiment of AMPK, we have found that AMPK is required in body wall muscles and PLM neuron. Now with this finding, there are two transcriptional output. One is DAP16 and MDT15. Now we wanted to understand how these tissue specific interaction in respect to this signaling is working. So for that, we used tissue specific RNAi approach. So where we knock down DAP16 and MDT15 specifically from muscles and neurons. As you can see, when the muscle specific DAP16 and neuron specific DAP16 were knocked down, so the effect of swimming was not observed. While MDT15, when muscle is knocked down, still the swimming mediated enhancement is observed. But in the neuronal MDT15 RNAi, that does not show up. So it summarized that DAP16 is required in muscles and neuron, but MDT15 is specifically required in neurons for mediating these effects of swimming exercise. So now, like this is a behavioral assessment we are doing to correlating the axon regeneration. So we delve details to understand what are the anatomical aspects of regeneration is getting affected or getting regulated by the DAP16 and MDT15. So I'll be done. So in the context, like, so first we check the regrowth length, so it's in the muscle and neuron specific RNAi. You can see in the neuron specific RNAi, both DAP16 and MDT15 does not show the enhanced regrowth at early time point compared to control. Similarly, we also saw the ventral targeting, which Anindya showed that which were correlated to the functional recovery. And we found that in the neuronal RNAi context, DAP16 and MDT15, those are required for enhancement of in the ventral targeting, which is caused by the swimming exercise. So this suggests that the neuronal in the neuron, DAP16 and MDT15s are regulating anatomical factors to which is correlating for the enhanced functional recovery. So in order to see how is the really DAP16 is active after swimming, so we used a CRISPR reagent tagged with MK2 in the DAP16, which shows the localization of DAP16. And when we compare the DAP16 with swimming and without swimming, you can see in the control condition, there is very basal level diffused localization of DAP16. But after swimming, the DAP16 is highly localized towards the nucleus. So this suggests that the nuclear localization suggests the activity of DAP16, which is quantified in here. And similarly, in the PLM neuron also, we saw a increased activity, increased localization of DAP16, and that is responsible, like that is required AAK2, because that in the AAK2 absence, the improvement was not observed. So this suggests that swimming improves or promotes the DAP16's nuclear entry, and that is dependent on AMPK. So this correlates with the behavioral and axon regeneration aspect. So now I'll summarize the models till now. So there are two tissues which are interesting, and in the exercise there are two arms which like activates AMPK through PAR4 and PRY1, which then in turns activate DAP16 in the body wall muscle and DAP16 and MDT15 in the neuron, which then correlates or integrates towards enhancing the functional recovery. So in the neuron, we have found that it is required for improved axon regrowth and ventral targeting. But there are certain questions still needs to be addressed to understand clearly. But the interesting part is that if we directly activate AMPK through AICR, so all this signaling integrates and triggers, and which enhances the functional recovery, so which can be followed to have a better strategy for therapeutic intervention. Okay, so this we just submitted to bioarchive, so if the details experiments are in there. Okay, thank you. I'll happy to take questions.
Any questions? We have 2 minutes.
Yes. Yes, so it has been shown that AMPK directly phosphorylate DAP16 at 6 specific serine threonine sites, which are responsible for probably the activity enhancement. So like AMPK directly phosphorylates DAP16, and that phosphorylation improves the cellular localization. Yes. So it has been shown that generally the AKT phosphorylation to the DAP16 that prevents the nuclear entry into the nucleus, but this AMPK mediated these 6 sites of phosphorylation. So that's we are currently doing the experiments like the AICR mediated activity, like activating the AMPK and the DAP16 localization, that will tell the question. So currently just is the swimming and the AMPK mutant data suggests that because in the AMPK mutant so we are generating that strand which the specific phosphorylation site will be blocked. So that will say the conclusively the answer.
Okay, I think we'll move on to our next speaker. Hello. think we can have time for maybe one question while I ask the next speaker to get set up.
Mohua Mole.
So while we're waiting, so our next speaker is Mahua Malik, who is a fellow at the National Institute of Biomedical Genomics. And her talk is going to be on connexins, potential targets for modulating neuroinflammation in demyelinating diseases.
Hello and good evening, everyone. So first of all, I'm really very happy to be here. And I thank all the members of the organizing committee to give me this opportunity and present at NBRC Foundation Day Symposium. So I'm Mohua. I'm coming from another DBT institute in IBMG, which is very unique in having a building in the form of DNA double helix. And we have a very small group of people who work on neurodegenerative or neurodevelopmental conditions at NIBMG. And my research interest actually focuses on proteins called connexins. So unlike the previous speakers who focused mostly on nerve regeneration, so this talk will be focusing on the glial cells. So what are connexins? Connexins are basically proteins which form intercellular channels between different kinds of glial cells. So these are called as gap junction channels. So as we know, in the brain, the major macroglial cell types, particularly the astrocytes and the oligodendrocytes, they extensively communicate with each other over distant brain regions through these gap junction channels. And these gap junctions comprise of proteins called connexins, which is a very huge family of proteins. There are about 21 connexin isoforms in humans. And each cell type, what is very interesting is each cell type has a particular subset of connexins. So like the oligodendrocytes, they have connexin 47, 32, 29, of which 47 is the predominant connexin expressing in the CNS. Similarly, astrocytes have connexin 43, which is the most predominant connexin expressed in the astrocytes. Now, so this is showing the basic structure of the connexin. And each of these monomers, they oligomerize to form a hexamer, which is called a connexon. So this connexon can express at the cell surface in the form of a channel, which is called as a hemichannel. This communicates with the extracellular milieu. But under normal physiological conditions, these hemichannels mostly dock with the hemichannel of an adjacent cell to form a gap junction channel, which allows passage of very small molecules like ions, amino acids, which are mostly of size less than 1 kD. Now, why we are so interested in connexins? So recent studies shows that a lot of neurodegenerative conditions, connexins are impaired. For example, different kinds of neurodegenerative diseases like Alzheimer's, other forms of dementia, epilepsy, stroke, as well as different kinds of autoimmune diseases like multiple sclerosis, NMO, and Barlow's disease. So all these diseases, connexin function is deregulated. Not only that, mutations, the recessive diseases, Mendelian recessive diseases where mutations in connexins are found, they have severe demyelinating condition. So because of this, there has been a growing interest among the pharma companies to develop a lot of therapeutic strategies targeting these connexins in different neurodegenerative conditions. But one important thing is these connexin proteins, other than their canonical gap junction function, recent literature suggests that they are also found to moonlight in other subcellular compartments like the mitochondria, the nucleus, the terminal nanotubes, and other places where they might play a very important role, which is not that well studied. So unlike the current therapeutic strategies, which mostly target the hemichannels, what happens is in the disease condition, the hemichannels are overactive. So the current therapeutic strategies mostly try to target these hemichannels so that their activity can be reduced. But it is not well known if you target the hemichannels, how the connexin activity or how the connexin function in the other cellular compartments or even the gap junctions are affected. So further understanding is required in this field in order to develop better therapeutic strategies targeting these channels. So now in that context, we are mostly focusing on two different connexin proteins. One is connexin 43, which is the predominant astroglial connexin. Another one is connexin 47, which is the predominant oligodendroglial connexin. And these actually heterocouple among themselves, forming connexin 43, connexin 47 gap junctions, which are present between the astrocyte and oligodendrocyte. So currently, my group actually focuses, we have two main projects where in one project where we are looking into the role of oligodendroglial connexin 47 gap junctions in the development of chronic demyelinating lesions, which happens following a viral infection. And in the other project, we are trying to decipher the role of connexin 43, which is the predominant connexin in astrocytes in context of Alzheimer's disease. So my student Pushpita will be talking more about her findings in this project. So in next few slides, I'll be mostly showing you some very recent data which we have generated on the oligodendroglial connexins in the neuroinflammatory demyelinating disease model. So as a model, what we are using a mouse hepatitis virus, which is a murine beta coronavirus. So what we do, we inject C57BL6 mice at 4 weeks of age intracranially with this virus. And following this infection, it induces a biphasic disease. So there is an acute stage where the viral infection persists, where the viral infection is there, and this is actually characterized by massive inflammation in the brain and spinal cord. Now, following 10 days post-infection, the viral infection is cleared from the system. This blue line, as you can see here, although the viral mRNA persists. And as the viral clearance happens, demyelination sets in, which peaks at around day 30 post-infection. So using this model, we are trying to understand the demyelinating pathology in the spinal cord and how that demyelinating pathology actually correlates with the alterations of connexin 43 in the astrocytes and connexin 47 in the oligodendrocytes, and whether these connexins actually have a role in the chronic expansion of the demyelinating lesions. So what we have done is we have collected tissues from the spinal cord at day 5, 15, and 30 post-infection, each of which marks different stages of this disease. So day 5 is acute stage, day 15 is a stage which marks the clearance of the infectious virus particles, and day 30 is a virus-free stage where there is peak demyelination. And we have looked at the different regions along the length of the spinal cord to understand how connexin actually associates, the connexin levels or expression of the gap junctions associates with the severity of the demyelination. So when we started the work, the general understanding with this model is that there is prominent demyelination along the length of the spinal cord, and that there is an overall downregulation of connexin 47 in the oligodendrocytes. So, so we started to look at whether the demyelination profile is uniform along the length of the spinal cord. So that's why we looked at all the three regions separately. And the data which I'm going to show today is only on day 30 post-infection, but we have done all the time points, day 5, 15, and 30. At 5, there is no demyelination. 15, we do see some demyelination, but most prominent demyelination we observed at day 30. Now, when we looked at the different myelin protein levels, what we observed is that the thoracic cord region shows the most prominent downregulation of all the myelin protein levels. In the lumbar region, we could not detect any significant alteration in any of the myelin protein levels which we have checked. And in the cervical region, there were some myelin protein levels which were downregulated, but not all of them. So from here, we concluded that the thoracic region showed the most consistent downregulation of the myelin proteins. And also, we labeled all the sections with a myelin stain, which is called a fluoromyelin, which you see here as the red stain. So what you see here is a spinal cord cross section. This is the gray matter area where all the cell bodies are. And this is actually the white matter area, which is supposed to brightly fluoresce with this fluoromyelin stain. So this is the mock, as you can see here. So this is the in the infected tissue, and here we have images from the cervical and the thoracic region. As you can see, the demyelinating lesions are much more prominent, or the area of the demyelination is much more higher in the thoracic regions compared to the cervical region. So this actually showed us that the demyelination is not uniform along the length of the spinal cord, but there is regional variation along the length of the spinal cord, and the thoracic cord region has the most prominent demyelination compared to the anterior cervical or the posterior lumbar region. So now we took this as a tool to find out whether connexin alterations are actually associated with this severity of the demyelination. So this is a representative image from the mock tissue. And as you can see, so this is stain for connexin 47, and CC1 is a very well-established marker of mature oligodendrocytes. So as you can see here, so all the CC1 labeled oligodendrocytes stain show nice punctate staining of connexin 47, which actually line the periphery of the cell. So these actually show or represent the typical gap junctions which are present in these oligodendrocytes. And this is the picture shown from the gray matter area as well as the white matter area. So when we looked into the spinal cord sections, this is the cervical region images taken from day 30 MHB infected tissue. What we found is that there is a severe reduction of this connexin 47 labeling in the gray matter, in the normal appearing white matter, as well as the demyelinating lesions. So as you can see that not all CC1 labeled cells do have the CX47 leveling. So there was a dramatic decrease in connexin 47. So this we found to be for the gray matter region and the normal appearing white matter region, we found this like the decrease was similar for both the cervical and the thoracic region. But what was very interesting to see was for the cervical region. When we looked at cervical region, the DM lesions, when we looked into the DM lesions of the thoracic region, we found that there was a complete loss of CX47. So you look at the cervical region DM lesion compared to the thoracic region DM lesion. So if we also quantified the percentage of CC1 positive cells which were positive for the CX47 gap junction staining, and as you can see from this graph, the DM areas actually showed more profound loss of connexin 47 gap junction expression in the thoracic compared to this cervical region. So remember, the thoracic region has a greater amount of demyelination compared to the cervical region. What was even more interesting was in the mock tissue, we found that all the CX47 labeling was restricted to only the CC1 positive cells. We could not find any CX47 punctate gap junction labeling outside of the CC1 labeled cells. But when we looked into the infected tissue, we found that CX47 gap junction puncta were also present in a number of cells which were negative for CC1. So we also quantified these CC1 negative cells which were positive for CX47. And what we show here, so we have the quantification here. So what we show here is that these CC1 negative CX47 cells were more prominent in the cervical region, which has lesser amount of demyelination. So clearly this data showed that connexin 47 expression in different subsets of oligodendrocytes is different, and that actually correlates with the extent of demyelination. So obviously the next question was, so there is a population of CC1 negative cells and there is a population of CC1 positive cells. So what we as cells are gaining gaining the expression of CX47 gap junctions. So we wanted to see if the proportion of these different subsets of oligodendrocytes were differing in the spinal cord region. So we very painstakingly actually quantified. So we did serial sectioning of the spinal cord regions for all the cervical, thoracic, and the lumbar regions. And we stained them, we double labeled them with two different markers. One is OLIC2, another one is CC1. So OLIC2 basically is a pan oligodendrocyte marker. It labels all the oligodendrocytes, whereas CC1 labels only the mature oligodendrocytes. And from this, we actually identified three distinct populations of oligodendrocytes in the spinal cord. So what we found is there was one population which was positive for OLIC2 and negative for CC1. So this population basically marks immature or precursor cells. Then there was a population that was double positive for both the markers, which is a mature population. And interestingly, we found another population of oligodendrocytes, which were positive only for CC1, but were negative for OLIC2. So when we quantified, what we found is that these OLIC2 positive CC1 negative cells, the CC1 negative cells, which had a higher expression of CX47 gap junctions, those cells were found to be increasing. Whereas all the CC1 positive cells, which were actually had a loss of CX47 gap junctions, those populations were decreasing. So we wanted to delve deeper into this, actually to find out how the different clusters of oligodendrocytes are varying. And so recently we have done a single nuclei RNA sequencing on the spinal cord tissue. So this is just showing that overall data. And so with the single nuclei RNA sequencing, we have been able to identify all the populations of cells, so oligodendrocytes, neurons, microglia, and OPCs, T cells, and ependymal cells. But we looked more closely into the oligodendrocytes. So when we looked into the oligodendrocytes, we found that these oligodendrocytes are actually having subclusters of, like, there are 8 subclusters, 8 or 10 subclusters within this, actually the total population of oligodendrocytes. And all these different subclusters do not have the same trend actually. So as you can see here, like some of, so these ones, they have a decreasing trend, like MFOL, MOL, OPC2, and COP2. COP2. So these are committed oligodendrocytes. These are oligodendrocyte precursor cells. These are myelin-forming oligodendrocytes, and these are mature oligodendrocytes. So these populations of oligodendrocytes were found to be decreasing, whereas there were different other clusters of oligodendrocytes that were supposed to be increasing. So this clearly showed us that the oligodendrocyte population was not homogeneous, and there was actually a distinct regulation between the subclusters, how they are regulated, how the proportion of these subclusters were varying in the control versus the infected tissue. Right. So further, what we have done is, so we actually grouped these cells. So we took this all these cell states of oligodendrocytes, grouped them into one, and these cell states, and grouped them into another set. And we did a pathway analysis of differentially expressed genes. What was very surprising is the increasing trend, the populations of cells which showed an increasing trend. In those population of cells, all the inflammatory pathways were coming up. So the oligodendrocyte populations, you remember the oligodendrocyte populations which showed a higher expression of CX47. So from a crude analysis from the IHC staining, what I've shown you is that the CC1 marker, which is a marker for the mature oligodendrocytes. So cells which were lacking CC1, they had a higher expression of CX47. So from this analysis, what we are finding is that the population of cells which showed an increasing proportion, those population of cells were showing the inflammatory signatures, whereas the population of oligodendrocytes which had a decreasing trend in the proportion, they had signatures of decreased oligodendrocyte neuron interaction. So I will, so just to summarize this, so what we have actually shown is that following a viral infection during chronic demyelination, the loss of connexin 47 gap junction is not uniform between all the oligodendrocyte cells. So there is a clear distinction in the gap junction expression profile between the precursor cells and the mature cells. And the relative proportion of these cells also vary with the infection. And the proportion of cells which show an increasing trend, they bear the inflammatory signatures, whereas the proportion of cells which show a decreasing trend, they have reduced neuron oligodendrocyte interaction signatures. So with this, I would like to thank my students actually who did the work. So the work I presented is done by Shobik and Gayatri. Pushpita will be presenting her work. And as I mentioned, that we have a small group of people who are working on different neurogenic diseases. And along with that, I thank all other members, my collaborators and mentors who have actually helped with this work and shaping up my research career. Thank you.
So can I, before we take one question, can I ask Deepti to set up the next speaker? Deepti.
keep this anywhere. You can keep this anywhere, right?
So that we don't know. So what we know is that virus mRNA circuits within the cell type. Here we are seeing that there is a difference in the severity of the virus among the lymphocyte particles. Virus protein is not there. So if we have to look at virus protein, we have to look at so the acute infection stage, when the infected virus is there is protein. So once the virus clears out, then the evaluation sets in. So the phase that I have shown to you is the base versus post infection when there is chronic evaluation, but there is only viral RNA, there is no infected virus. So what we are saying is that maybe the persistence of viral RNA is also not uniform. It depends on what type of the viral RNA acts as suppressing some other. So with a single we also get 1 to 3. From there we can find out like which of the cell types actually retain the viral RNA. So we are trying to see like what are the cell types that have the viral RNA and if there is any difference in the proportion of these cell types or even in the level of the virus. So this data is very new. The RNA sequencing data we just got it, so we are trying to analyze it as best possible.
Okay, thank you for that. Next speaker is Deepthi, and we'll talk about the study of the role of PI3K and AKT signaling in myotonic dystrophy type 1 in Drosophila.
Am I audible? Start. Good evening, everyone. Thank you, organizing committee, for giving me the opportunity to present my work here. I'm Deepthi Chakraborty, PhD student at NBRC, and I'm doing my project under the supervision of Dr. M. Dhruva Singh. I have started my work. I have been working on this project for past 1.5 years, joining the lab. The title of my project is to study the role of PI3K AKT signaling in myotonic dystrophy type 1 using Drosophila model. So first of all, coming to the introduction, what is myotonic dystrophy type 1? Myotonic dystrophy type 1 is a genetic autosomal dominant disorder. It's a neuromuscular disorder and is characterized by neurodegeneration and progressive loss of muscle. One of the most important hallmark of this disease is grip myotonia, where the patient could able to relax, contract the muscle easily, but relaxation of the muscle is difficult, followed by drooping of eyelid, distal muscle weakness, winged scapula, desolate face or lifeless face, and even the children are found associated with complete atrioventricular block. It has a global prevalence of affecting about 2.7 people per 100,000. It's a multisystemic disorder affecting almost every system of the body, including endocrine system, visual system, cardiac system, respiratory, as well as gastrointestinal system. Some neuroscience aspect of this disease is mental impairment or mental retardation, as well as psychiatric disorders. And some MRI, fMRI scans have revealed that there is severe white matter lesions and decreased gray matter in DM1 patients. Now, myotonic dystrophy type 1 is caused due to mutation of the DMPK gene in chromosome number 19. This mutation is due to the presence of this trinucleotide CTG repeats, which are present in the 3' untranslated region of this DMPK gene. For a normal gene, these repetitions are less than 35, whereas with gene with DM1 alterations, this repetition goes from 50 to more than 1,000 repetitions. Due to this trinucleotide repetitions, it leads to the formation of toxic CUG mRNA, and this toxic CUG mRNA further accumulates and forms the abnormal RNA foci formation and hinders different other RNA binding proteins and leads to protein sequestration, which further hampers the splicing activity of this process. Splicing leads to spliciopathy, as well as aberrant protein expression of some insulin, especially insulin receptors have been found in previous studies. So my work has been divided into 3 objectives. Objective 1 is to identify genetic targets of DM1 in the insulin signaling pathway using the Drosophila model. Objective 2 is to investigate the role of PTEN in the pathogenesis of DM1. And objective 3 is to study the mechanistic role of PTEN in altering the molecular hallmark of DM1. We focused on the insulin signaling pathway because it has been already found from previous studies that insulin signaling pathway is hampered in DM1, and insulin sensitivity is decreased by 70% with DM1 patients as compared to the control one. We started with initial screening of genes which are involved in this pathway, and we found that PTEN RNAi could able to give us rescue or improvement in the eye phenotype as compared to the diseased flies, where there was enhancement. This is the diseased one. These two are the controls. So there was decrease in the eye size, presence of some necrotic patches, whereas depigmentation of the eye, which was improved in case of PTEN downregulated flies in the disease background, where there was significant increase in the eye size as compared to the diseased one. PTEN is an important molecule present in the insulin signaling pathway. It's a small molecule and it can be easily targetable molecule. That's why we selected PTEN as our modifier, and we proceeded with further experiments. First of all, we validated our line using qPCR, and we found that there was decrease in the transcript level of PTEN in the 5-day-old flies. We started with driving the disease pan-neuronally using ELAV-Galfo driver, and we found here that there was death in the embryonal and in the first instar larval stage of the fly. The survivability assay showed that there was 0%, like eclosion percent of the disease flies didn't eclosed at all. There was complete death in this stage of the fly, whereas 9 to 10 flies still eclosed and in the rescue condition. As we have seen that the flies, there was complete death of the flies while driving the disease pan-neuronally. So we drive the disease only in the mushroom body, which is equivalent to human hippocampus, and we seen the morphology of the disease flies. So there was complete degeneration of the gamma lobe in the disease condition, whereas there the gamma lobe is still intact in the some of the gamma lobe is still intact in the rescue flies. Now what we did, we drive the disease in the muscle of the fly, and we checked for the lifespan of the flies. We found there was a great improvement in the lifespan of the flies, of the rescue flies, which survived till 40 to 45 days as compared to the diseased flies, where the lifespan was 5 to 7 days. We decided to move on to further experiment to check the motor ability of the flies. So in this video, we can see that the disease flies could not able to climb up to 8 centimeter mark, whereas the rescue flies could able to climb up the wall. And we did this experiment with both male and female flies, and we found significant rescue between the disease and the rescue one. Now, as we have found that there was improvement in the lifespan as well as motor ability of the flies, we decided to check on the muscle morphology of the disease flies as well as rescue flies. So muscle biology is largely conserved from insect to mammals, and this is the sarcomere, which is the functional unit of the muscle, which is composed of actin and myosin. Actin is the thin filament, whereas myosin is the thick filament. And this group of sarcomeres forms the myofibril, whereas the group of myofibril forms the myofiber, which forms the muscle of the fly. We used phalloidin staining to mark up the actin filament, and MHC, that is myosin heavy chain antibody, to mark the M line, that is myosin heavy chain. We did our experiment and we found that yes, PTEN downregulation improves the muscle morphology. So this is the actin filament, and we found that there was decrease in the sarcomeric length in case of the diseased fly, and in the rescue fly there was improvement in the sarcomeric length as compared to the diseased. Next, we move on to check the myosin or the M line, where we found in the disease condition there was decrement in the thickness of the M line, as well as there was decrement in the mean fluorescence intensity of the M line, and it was improved in case of our rescue flies. As I have already mentioned in my previous slide, that RNA foci is one of the major hallmarks. It leads to the formation of RNA foci. So we wanted to check whether RNA foci was also decreasing in case of our rescue flies or not. So we did fluorescence in situ hybridization, and we checked for the RNA foci. And yes, there was decrease in the RNA foci in the rescue flies as compared to the diseased one, where there was increase in the number of RNA foci. Now we wanted to check the mechanistic role of P10 and how it is like how this rescue is happening. What is the molecular mechanism behind this? So from the previous studies, we found that the autophagy level in myotonic dystrophy is increased. So what we did, we stain the adult fly muscle with LAMP1. LAMP1 is lysosome associated membrane protein, which marks up the lysosome, and we found that there was significant decrease in the autophagy level in the rescue flies as compared to the disease fly, where there was increase in the autophagy level. So after doing that experiment, we wanted to check whether the ATG gene expression are decreased or not in case of rescue flies at the transcript level. We did PCR. In case of ATG1 and ATG5, we found there was a significant decrease in the ATG1 and ATG5 expression level as compared to the diseased flies. So after doing that experiment, we just wanted to understand how these ATG genes are playing role. So we started with screening of the ATG genes. We did the ATG1 RNAi in the disease background and ATG5 RNAi in the disease background, and we found there is improvement in the eye phenotype as compared to the disease flies. Whereas when we overexpress ATG1 and ATG5, there was enhancement in the disease phenotype, and in case of ATG1, the flies even did able to eclose. To summarize my work, P10 downregulation demonstrated significant improvement in several DM1 associated phenotypes, which leads to improvement in the eye, brain, and muscle morphology of the flies with CTG repeats. Our findings were consistent with various behavioral studies also and longevity studies, and we found there was reduction in the RNA foci, as well as there was decrease in autophagy level in the rescue flies. Our future directions are to study how P10 is controlling autophagy, and second one is there any interaction between P10 and autophagy genes, and we want to also investigate the disease progression at the level of neuromuscular junction. So I would like to express my gratitude and my special thanks to Dr. M. Shubha Singh, my supervisor, DC committee, my lab members, family members, and all my teaching and non-teaching staff at NBRC. Thank you so much.
Okay, so we'll take one question, and in the meantime, I'm going to ask our next speaker, Pushpita, to set up.
Yes, sir. So P10 and repeats, like P10 have been, that is the main question what we want to like. Okay, so actually P10 is one of the molecule which helps in cell cycle like regulation, and also might be it's going through any, yeah. Yeah, PI, yeah, and acetyl phosphate. Yeah.
So how does that kind of help you to
separate So that is the like main question which we want to look into it. Actually, there are studies where I think they have told that P10 is somehow in some other disease also it's controlling foci, but we want to check that how it, yeah. No, sir, not still. Mm-hmm. So we have to look into it. Just we are just onto this experiment. We are doing this experiment. We will check it further, like how it's foci and P10 is. Mm-hmm. 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 Yes. Thank you, sir. Any question? Thank you.
Okay, so our next speaker, Pushpita, will talk about amyloid beta mediated inhibition of connexin 43 anterograde trafficking disrupts gap junctional communication in human astrocytes.
Hello. Good evening, everyone. Am I audible? My name is Pushpita Shaha. Currently, I'm working under the supervision of Dr. Mohua Maulik and Dr. Maulinath Acharya at BRIC NIBMG. First of all, I would like to thank the organizing committee for giving me the opportunity to present my work today. And as my supervisor has already pointed out the importance of connexins in neurodegenerative disorders, I will be primarily talking about how amyloid beta impairs the functions of astroglial connexin 43 in Alzheimer's disease. The title of my talk is Amyloid beta mediated inhibition of connexin 43 anterograde trafficking disrupts gap junctional communication in human astrocytes. As we all know that Alzheimer's disease is an age-related multifactorial neurodegenerative disorder that affects people primarily above 65 years of age. And the common neuropathological hallmarks of an AD brain are accumulation of amyloid beta plaque, neurofibrillary tau tangle, synaptic loss. In recent years, astroglial dysfunction has emerged as a common pathophenotype alongside neuronal dysfunction and amyloid beta deposition. In a normal healthy brain, the astrocytes are the predominant glial cells of the central nervous system, and they communicate with each other through these gap junction proteins. For astrocytes, connexin 43 is the major astroglial gap junction protein, and they also communicate with other glial cells in the central nervous system through the gap junctions. Gap junctions are intercellular channels which allow the transfer of molecules less than 1.2 kilodalton. These molecules can be neurotransmitters, ions, and other small molecules which are required for synaptic informational processing and maintaining the homeostasis in the brain. It has been found that in an AD brain, these astrocytes acquire a reactive phenotype, and the overexpression of the connexin 43 hemichannels under the influence of amyloid beta leads to the exaggerated release of excitotoxic molecules which lead to neuronal death. Thus, the importance of astroglial dysfunction in AD has been pointed out. Connexin 43 is a transmembrane protein, and as we had already seen that the head-on docking of two coaxially positioned hemichannels forms the gap junctions on the cell surface. While in recent years, a growing body of evidence has shown that amyloid beta impairs the functions of connexin 43 gap junctions and also the hemichannel activity, but the mechanistic underpinnings of this process has remained elusive. And along this line, we started our work using the primary human astrocytes as the in vitro cell culture model. We treated our cells with we treated our cells with the toxic fragments of amyloid beta at different time points, and through the conventional gap junctional assay technique, we found out that amyloid beta significantly reduces the gap junctional activity in primary human astrocytes. We wanted to understand that how amyloid beta is carrying out this process, and we found
that upon treatment with amyloid beta, the connexin 43 gap junction proteins, which are present normally in a punctate manner along the cell borders, are forming intracellular aggregate like aggregates in the cytoplasmic region. And upon further co-localization studies with the cell surface marker N-cadherin, we found that not found that not only that the amyloid beta increased the intracellular localization of connexin 43, but also it reduced the surface expression of CX43. Thus, amyloid beta increased the intracellular localization with a concomitant decrease in its surface localization. We next wanted to understand that where this increased pool of connexin 43 is headed in the amyloid beta treated astrocytes. And we found that this increased pool of CX43 is sequestered in the endoplasmic reticulum that led to a significant increase in the co-localization of CX43 with calnexin. Further, we also studied the trafficking of CX43 through the secretory pathway, and interestingly, we found that the increased retention of CX43 in the endoplasmic reticular compartment was accompanied with the restricted entry of this CX43 in the Golgi compartment, and that led to a decreased co-localization with both the cis Golgi and the trans Golgi markers. Thus, from our data, we found that amyloid beta increases the ER retention of CX43 along and restricts its anterograde trafficking through the from the ER to the Golgi compartment. Next, we wanted to understand that what is causing the ER retention of CX43 in the A-beta treated astrocytes, and we asked that whether the A-beta induced ER stress is a plausible mechanism, and we found, and through a study of a comprehensive panel of ER stress markers, we found that amyloid beta induces ER stress response in the primary human astrocytes. And interestingly, this ER stress leads to the ER associated degradation of this accumulated pool of CX43, which we could rescue by MG132 treatment. We further wanted to validate our finding that A-beta induced ER stress is the underlying mechanism that is causing the ER retention of CX43. So we used 4-phenylbutyric acid in our experiments. 4-PBA is a well-established ER stress inhibitor, and we found that this increased pool of CX43, which was accumulated in the ER of amyloid beta treated astrocytes, when treated with 4-PBA, this pool was sent back to the surface, and the connexin 43 gap junctions were again organized as distinct punctate structures along the cell lining. And upon further analysis, we found that 4-PBA could also repress the ER stress induced by amyloid beta. So we found that 4-PBA reversed the ER retention of CX43 by repressing the A-beta induced ER stress in primary human astrocytes. From our Triton X100 solubility experiments, we found that amyloid beta interferes with the proper assembly of connexin 43 in the astrocytes, and we also checked that what happens upon 4-PBA treatment, and we found that this increased pool of CX43 was sent back to the surface. Thus, 4-PBA could improve the assembly of the CX43 gap junction complexes and restore the gap junctional communication on the cell surface. So from this, we concluded that amyloid beta induced ER stress increases the ER retention of connexin 43 and interferes with the anterograde trafficking of CX43 towards the cell surface that disrupts the gap junctional activity. Finally, in addition to the amyloid beta induced ER stress, we also found that amyloid beta interferes with the interaction of connexin 43 with its chaperone ERP29. And from our proximity ligation assay experiments, we found that amyloid beta reduce the interaction of CX43 and ERP29, and that is another mechanism by which amyloid beta interferes with the gap junction activity of CX43 on the cell surface. So with this, I would like to acknowledge my PhD supervisors, Dr. Mohua Maulik and Dr. Boninath Acharya, for their guidance and support, our director, Dr. Sagar Sen Gupta, and I would like to thank my student advisory committee members, NIBMG for intramural support, and DBT for fellowship. Thank you for your kind attention.
So thank you, Pushpita. We'll take one question, and can I ask Proloy to get set up, please? Thank you.
Sir, what we observed that there is an aggregate which is present in the ER. So maybe the ER stress response is causing the misfolding of CX43, and we have already seen that under the effect of amyloid beta, the interaction of CX43 and ERP29 is impaired. So maybe that's why the yeah, but yeah, sir. But sir, we haven't conducted that experiment. But one line of thought is that maybe amyloid beta is itself interacting with CX43 or ERP29 and interfering with their interaction with each other, because A-beta is a stress inducer. So that can be done by co-immunoprecipitation experiments of amyloid beta with CX43 or ERP29. Yeah. Okay. So, sir, another experiment that can be done over here, one is the in situ, we can study the in situ interaction. And also we can have like fluorescent A-beta and see that whether it is interacting in live cells. We can see that how it is interacting with CX43 or ERP29. Yeah, yeah. So if whether it is a direct effect of or not, that can be only addressed by studying the interaction of amyloid beta with either of these two targets. Thank you, sir. Oh, sorry.
I will be our next speaker, and final speaker for the day is an assistant professor at NBRC, and his talk is titled "All It Needs to Meaningfully Quantify Neural Oscillations is a Clever Dynamic Generative Model."
Okay. Okay, am I audible? Okay, so I'm Proloy Das, assistant prof, like youngest assistant prof here at NBRC. And this talk will be very different from all other talks. It's not molecular, it's not, it's definitely biology, but it's not biology in that sense. It's computational biology. So as the title says, it's all about modeling some signal that we are recording out of an experiment. And then I will show, after we figure out how to model efficiently this phenomenon, how this can be used in different, like in designing biomarkers for humans for different diseases. Okay, so first of all most of this work applies on any time series data,
but here we will only focus on MEG and EEG, magnetoencephalography and electroencephalography. And for those who don't know like where these signals are coming from, so in our brain the neocortex has like a bunch of neurons which are organized, which are organized in this very structured way parallelly, and all these actions when they fire all together, all these currents create something called current dipoles, and those current dipoles can be measured, the activity of those current dipoles can be measured outside of the brain. That is how we can either measure the electrical activity that is happening inside the brain, outside on over scalp, or even like outside of our like sorry, not outside, like in the air around our head as MEG signal. So these two signals are the most direct correlate of the in vivo processing. Like you cannot do better than this. fMRI does not give you this sort of signal. Okay, so this is the most basic signal with which our brain signals one part of the brain to another part of the brain. Okay, so now why now like where did this thing come from? So in 1924, exactly 100 years from this year, this guy Hans Berger, he was a German doctor. So he thought about doing this experiment where he put one electrode in front of one patient's brain and one in the back of the patient's brain, and out of nowhere he saw there is this. So this is the actual data that he collected. Out of that experiment he saw, oh, there is a very nice looking pattern in that activity, that electrical activity. And then he compared that activity with this 10 hertz signal, and that is how he figured out this is the very fast ever seen neuronal oscillation, okay, from a human brain. So he named it alpha wave. It was, for some time it has been known as Berger's wave, but later on we started calling it alpha wave because this was the first EEG wave oscillation that was ever discovered. Now fast forward, what happened was we found all other different frequency bands. These frequency bands in EEG are called canonical bands. And whenever you see any kind of EEG experiment, let's say Alzheimer's biomarker, they will say, oh, we looked at the alpha activity in disease population versus control population. So what they actually mean is they looked at the power in this alpha band. So alpha band is not shown here. Alpha band should be around 10 to 13 hertz or 9 to 13 hertz or 9 to 14 hertz. No one knows because it's just a heuristic. People just came up with two numbers. Okay, from 9 to 13, let's look at that frequency band, and that is how alpha band works or any kind of band works. So now the problem is all the data analysis, like the way you have to look at this frequency bands is you take this EEG data, you do something called Fourier transform. So you look at the frequency component of the data, and when you look at frequency component of the data, so these three, like, okay, let's look at only this column. In this column, all these three panels, they're the same thing but computed in different ways. So just the way it is computed gives you very different results, right? So this one is almost garbage. You can't see any feature. This one you can see, oh, there might be some alpha wave but not sure. This one shows, okay, yeah, there is some alpha wave. Now why is that? Because this problem is very difficult to get the frequency component. As humans, we are not supposed to look at any frequency of anything. We are like, the way we have been engineered by nature, we only make sense of the time series data. All our information we get from the environment is time series, right? When we are looking at something, we are just getting it at different time points. We are not looking at, okay, what is the frequency or what is the power in this frequency, that frequency. That is how MRI works, not us. Okay, so it will be very problematic if we just try to see, oh, if this is a alpha, if there is a alpha wave in this signal or not, depending on the depending on how you analyze the data. and things become even complicated here. See, there are two peaks here. So there is definitely alpha, but there were two peaks here. And then those two peaks kind of disappeared here, become just one peak. Now, which one do you believe? This one, this one. Right. So first of all, detecting these peaks are difficult. Now, sometimes there could be more than one peak. So then you are lost. Like, okay, which one you should believe? So if you can't even like believe these plots, how can you just say, oh, all this analysis method just works for Alzheimer's disease biomarker? Right. That is like a huge leap. So think of this as a microscope. This thing is basically a microscope which allows you to look at the brain's activity. Now, if the microscope is not working properly, how can you like justify all your research, right? So now, these guys, Thomas Donohue in 2020, he came up with this method to, it's called FOOF method to quantify like the power, or like if quantify the oscillation in a spectrum. So what it did was very ad hoc based method. He just looked at peaks in this profile. If there is a peak, he says, oh, there is a oscillation, and then there is a ad hoc way of kind of like setting these two bars and saying, oh, this is the oscillatory power and whatnot, and then we can basically treat this as a biomarker. Oh, so this alpha power comes from patient group and this alpha power comes from control group. Now again, this thing theoretically sounds awesome, but what will you do here? Will you consider these two as two different oscillation, or this as one oscillation, right? So all these ad hoc methods, they don't tell the whole picture. These are just approximations, and these are just like some modeling framework, and modeling, all models are wrong, right? Some are useful. Here these models are also not useful, so we have to look elsewhere. So where do we look? So this was the frequency domain. So we have to actually go back to the thing that we are based at, looking at the signal in time domain. And it turns out that there were actually lots of this sort of work in economics. In economics, people used to separate out this trend and this cycling effect of, let's say, GDP fluctuation, or what that thing called, insurance price fluctuation and whatnot. So there were, like in other fields, people try to do this, but in neuroscience, because these things are hard and neuroscientists primarily come from biology, they just thought, okay, let's go for the easy one, right? So how can we make this thing available for neuroscientists? So basically the idea here is, before that, the idea here is they actually design, so Fourier transform builds the filters very generically. So it is like one fits all approach. So instead what these guys did was, okay, let's design the filters so that let's design the filters according to some model which actually are consistent with the data. So we will actually make the filter out of the data itself. Okay, so there won't be any template. The template will be automatically learned from the data. So that way we will be more closer to the data. We will be more representative of the actual model, rather than just saying, oh, the data came from there, so we just feed some arbitrary model. Okay, so these things are called adaptive modeling. So the model, the underlying model that we will work with is very simple. So consider this thing as one hand of the clock, and here the clock hand moves counterclockwise. Okay, so every time it moves, it needs to be multiplied by this rotation matrix. So here is this multiplication, and then this clock hand can also change its length. Okay, that thing is necessary because nothing in our physical world is regular. Everything is irregular. So in order to compensate for that irregularity, we have to consider that. And now what happens is, so this clock hand is moving counterclockwise, and we just look at one projection of this clock hand on this real line. And as this clock hand actually rotates, this projection actually moves back and forth. So now if we plot that thing with respect to time, it actually forms a oscillation, right? So this is very easy modeling. If you think about it, it's very intuitive. You don't have to you don't have to know Fourier transform or anything. So if something is rotating with a fixed speed in 2D plane, its projection will make out an oscillation. So this is the mathematical model, and this Matsuda and Komaki they came up with this model in 2017, and we came up with the same exact modeling from neuroscience perspective from a dynamical model system. And now you can also generalize this for multiple observation. So let's say there is one underlying oscillation, but now you are making two observation. So one observation is along this line, another observation is along this line. So you will have two different time series corresponding to the same oscillator. So that happens in EEG data all the time because there are multiple electrodes that is recording the same neuronal activity, right? And this framework is like generalizable very easily. We can also generalize it for multiple oscillator where we have more than one oscillations. Here I'm showing M oscillations, and each of these oscillations are showing up in the observation according to this projection. So I'm just projecting all the oscillations, all the clock hand on the real line, and summation of those actually makes up the EEG signal, let's say. And now the task will be if we can decompose this EEG signal into this oscillatory component, then I can identify all the oscillations that were present in the data, right? So the way we do it, we basically write this for fixed time. Let's say we know how many oscillations are there. In that case, we write this complicated log likelihood function. This looks very complicated, but for engineering people, this is like our bread and butter. And so after you write this log likelihood, there is a way you can figure out all these parameters that define these oscillations. The parameters that define these oscillations are given in this slide. So these are the parameters. So this F, which is the frequency with which the oscillation oscillates, that frequency is one parameter. This noise is another parameter, the nuisance parameter. And sorry, this R is the noise, this is the nuisance parameter, which accounts for the measurement noise, because in EEG there is like lots of measurement noise. So you have to account for that. And then this is one important noise. This is called process noise. So this noise actually drives the oscillator. So this is basically our internal driving force that is making us alive. Anyways, so these are the parameters that we can learn from the data, and we will learn using this algorithm called EM algorithm. So this EM algorithm has these two steps, E step and M step. And why we need this specific algorithm is we know only the so basically in the problem we know only the EEG observation. We don't know the hidden oscillations. We don't know any parameter that describes these oscillations. But if we know the oscillations, we can get back the oscillation parameter. And if we know the oscillation parameters, we can also get back this hidden oscillatory states. So what we do is in each step we consider that okay, we know a guess, like we have a guess for the oscillation parameters. So with that guess we can compute, or we can say okay, we can extract what are the oscillations are there, and then once we get all the oscillations, we do maximization step. This maximization step basically says if these oscillations are explaining my data good enough, so it computes a goodness of fit, and it maximizes that goodness of fit. And that goodness of fit, once it is maximized, then we come back to the E step again. So we basically have another set of oscillation parameters. We then again compute the same thing with those parameters and then go back and forth. until we are satisfied with the oscillation parameters. And when do we get satisfied? We get satisfied when these parameters does not change anymore. Okay, so now once we have the oscillation parameters, we can decompose the signal into those oscillatory components. And so I showed this thing. This thing works for only fixed M, but we also don't know beforehand how many oscillations are present. So the way we figure out how many oscillations are present, or there is only this method allows you to do so, is the way we do that is we have this goodness of fit, right? We and once we terminate the algorithm, we can again compute the goodness of fit for that many number of oscillators. So that goodness of fit, or the negative of that goodness of fit, is plotted here. So as we feed the same model with different number of oscillators, we will see that this goodness of fit will change, and we will choose that goodness of fit which explains the data the best. Here I am plotting the negative of that goodness of fit. So whichever has the lowest value, I'll pick that one. So probably some of you guys know about archaic information criteria, which basically allows you to kind of compare two different models. So this is one way of implementing that thing in our framework. So once we know how many oscillations are present, and we already figured out what are the oscillation parameters, we can decompose the signal into those elementary oscillations. For example, this is one EEG data. So here there are two oscillations, as we can say. there is one slow oscillation, and then another one is this fast oscillation. And using this algorithm, we can decompose it as these two oscillations. So this is the slow oscillation, and this is the fast oscillation. This is the alpha oscillation. This is the slow oscillation, and this is the error. So not only we are separating the oscillations, we also separate out the background noise. So this is like this denoising comes right out of that. We don't have to do.
Second oscillation actually has another modulating amplitude modulating.
Yes, it has, but the model did not vote for that. Yeah, so that is the good thing about this method, that you can be very objective. Like, okay, I am picking up only two oscillator because of this goodness of it, right? End of story. Like there won't be any reviewer comment like, oh, you missed that, right? So this is like one way of doing this kind of oscillation selection very rigorously. Okay, so now more examples. So where do we actually use good? Where do we see any good use of this type of data analysis? So this is one of our preliminary data from my postdoc lab back in Mass General Hospital and Stanford University. So here we collected just resting state data from these three different population: young adult, and and cognitively normal elderly, and elderly person who is already amyloid positive, right? So if we just look at alpha activity by looking at the power within this 8 to 12 hertz, we see there is no trend. There is just no trend, because young is this one, alpha beta negative is the healthy aging. So it should have somewhere between, like its alpha should be somewhere between young and alpha beta positive, right? And why we are not seeing that thing is because this 8 to 12 this span actually misses this peak. So that's why this alpha activity that we are summarizing here is a wrong estimate of its alpha. But if we can if in our method we can actually see this is the alpha, and this alpha does not satisfy this 8 to 12 hertz frequency band strictly, but still it is the alpha. So if we consider this as a alpha, we can see there is a clear trend. So with amyloid positive patient, their alpha is even decreasing. So that we have to do, that we are doing right now. Collected lots of data. So we will go on doing that later on. So this is one example. And then we can actually look at multiple oscillators. So this is very quickly, this is, no, we don't expect anything. This is just our primary observation. So we have to basically, this is just exploratory. We have to look into the crux of it, and that is why we need like lots of data collected, because you have to look at the population, right? So we just build the data, the analysis method, and then we will go for the data analysis. Okay, so we can also generalize this thing for multiple observations. And if I show you the results, so there is this method called independent component analysis, which is analysis method directly borrowed from machine learning people to kind of like try to separate out this oscillation, and it does a very poor job because it doesn't have that underlying oscillation model. If we introduce that underlying oscillation model into that framework, we can separate out these oscillations really well. And why that is important is, so this is one real data example where we compared the resting eyes closed data to the REM sleep data to check if the activation that is happening in the brain in these two conditions are similar or not. Because both of the cases we see the alpha, the posterior alpha. And from these figures, this oscillation components, you can see the components, the distribution of the components are slightly different. So this one looks something like this, and this one also looks something like this. This one doesn't have any corresponding ones. This one also doesn't have any corresponding ones. And also we can compute something called what is the, like, how correlated all these activities are within the whole brain. And if we see that during REM sleep, all the alpha activities are much more coordinated than when we are awake. So in alpha, during sleep, we are actually using the alpha in a very focused way rather than when we are awake. When we are awake, we are in a very ready mode. So our alpha needs to be everywhere. But when we are sleeping, the alpha is very focused because we are just resting. Right? And then we can also use this framework to characterize oscillations which are transient, because in neuroscience, human neuroscience, these transient oscillations show up everywhere. This is one example where it shows up sleep spindles. So this is how this normal sleep spindle data looks like. And this is and you see this spindles which are like activities around like 15 hertz. They show up like for a couple of seconds, and then they dies down again, they show up, and so on and so forth. And it has been shown in Alzheimer's, the sleep spindle density changes between the Alzheimer's with the disease progression. As the disease progresses further, the sleep spindle activity becomes less denser and denser. Okay, so we need to characterize basically to follow up with that. We need to better characterize how to detect these sleep spindles. So we can use our framework to detect sleep spindle, and also during aging, it has been shown across the literature that the alpha decreases. Okay, but if you show this figure, the alpha probably does not decrease, it just becomes fragmented. So our alpha, instead of like staying stronger all over the time, it just becomes transient, kicks in for some time and then dies out for some time. The x-axis is time always. X-axis is time and y-axis is the frequency. This is spectrogram. Okay, so we can actually model all this thing using something called switching oscillator. So in this switching oscillation model, we consider two different models, where in one we have two oscillation, let's say, and this one we have only one oscillation. This is the spindle. So let's say this is the slow wave, which is predominantly seen in the sleep, and this is the sleep spindle activity. And the other model only has the slow wave activity. and there is this switching state which kind of switches between these two states depending on the data. Okay, so this is the pictorial depiction. So within this model we can actually segment out. This is the same figure earlier. We can actually segment out. So these are the segmentation we are getting from this switching state. So this switching state, when it is on, it says that there is spindle, and when it is off, it says there is no spindle condition. So we can like automatically segment out this spindle activity without even telling there is spindle or any properties of the spindle. It just, the algorithm just needs to know there are two oscillations. There could be two oscillation or one oscillation. We don't even say look for spindle within this frequency band or anything. So this can be custom made for any individual. Okay, it's completely data driven. So this can be made into your mobile phone and your sleep spindle can be tracked very easily. There is no intervention needed. So this is one example, and then this is another example. Same data for multiple patients, and we can do it very very efficiently, meaning like consistently. So if we do it on multiple trials, multiple days, multiple night sleeps, we have the same. We can detect the same density, whereas all the other methods, they kind of fluctuate all around the place. So that this is the other one, and that's it. So all these methods are available on my website, on GitHub. So this partner lab MEG is from my previous lab, and we deposit all the developed code so that whoever is working with this sort of data can use our methods really easily. So with that, I'll just close my talk. Thank you. It decreases in all type of dementia. Oh, that difference, I am not familiar with any that type of study. They always like most of this data collected separately, and there is like, because of the different ways they formulated or like figured out the sleep spindle density, they are not comparable with each other. So that is the problem. So we are trying to standardize the method so that they can be compared across different yeah. I did not get your question. So yeah, can you repeat it once more? Yes. Yeah, these two oscillators. Yeah, these two oscillators. Yeah. Oh, I see, I see. So that we cannot say right out of the bat, but probably there is some phase coupling happening. Okay, phase amplitude coupling happening between the slow wave and this spindle activity, or like whatever kicks in that spindle activity. So probably when it gets phase locked to something, like when this slow wave and let's say probably the alpha one or the beta oscillation gets phase locked to something, it starts one sleep spin. Okay, but that is like a pure conjecture. We don't know yet. It's very hard to study all those things because EG data is very noisy. So, and there is just not that many tools available to deal with this kind of problem. Oh no, spindle. Yeah, spindle happens in only couple of electrodes. It's very focused. So probably you will see spindle in 2 or 3 channels, like simultaneously, and it happens at different parts of the brain. So it's not a global phenomenon.
Thank you, everyone. So this is the last speaker of the day. And so the only thing I will like to request tomorrow, please be here on time, pretty please, with a cherry on top, extra sprinkles.
So just a couple of announcements. One is that today dinner will be at 8:30 instead of 8 o'clock, and it will be in the cafeteria. Okay. So you inform your friends in the hostel to come over, and even they have not registered, there'll be plenty of food. Number one. Number two, tomorrow breakfast will be at cafeteria at 8 o'clock in the morning, and be here by 8:45. And we really have to start by